A very big hello to all my wonderful champions out there. Let me see if at all I am audible as well as visible. Yes. Hello Ganesh. Hello Ganesh, hello Arun, hello Stranger Things and everyone out there. Children, we'll just wait for some time so that everyone else. Hello Riddhi. I hope my dear children, good evening Sanjeevani. I hope all my dear children are doing really good. Yes, so make sure you share this video link with all of your friends. Listen to me very carefully. All these questions that I'm discussing over here are super important questions, and they have been curated, as in I went through all the model question paper. You saw the video for the fixed question. One video will be still going to be released. From that, the questions which have, or the concepts which have most probable chance of coming, I have taken them over here. I'm going to solve them for you guys over here. Make sure you listen to me super carefully. Yes, and we'll rock biology. Hello Simran, hello Ganesh, Ganesh, and Shravan Kumar, Madhuri, Tanish, Viresh, Indra, everyone, hello. You're looking pretty. Thank you so much. Thank you guys. Okay. So I think we are good to go. So I'll tell you, MCQs will be very less. More of the, 2 marker, 5 marker, 3 marker questions which are there, they will be in more amount. But yes, MCQ will be there here and there in between. So let's get started. Everyone ready? So one rule is there. Whenever you are done with the concept, just give me a thumbs up. That's how we go to the next question. Okay? Sup ma'am, I'm all good. Thank you for asking, PG Sam. And Vaishnavi, hello. Pratiksha, hello. Riddhi, thank you for your efforts. You're most welcome, dear. Pushpa, good evening. And by the way, I hope everyone have sent the video link to all the children who are studying biology. No one is selfish, right? Everyone wants your friends also to score good. So definitely share this video link. Let's make it 100, more than 100 this time, and let's make it the best use of it. Okay. So I do not want to waste your time. Let's go get into it. First thing, you do not have a choice. Do you have a choice? No, you do not have the choice. You have to give your best. You just have 1 day's time, and we are going to give our best. Starting with the first question that we have goes like this. Let's see who all able to solve this. Statement number 1. Formation of fruit without fertilization is known as apomixis. Formation of fruit without fertilization is known as apomixis. Statement number 2. In some species of Asteracean grasses, seeds are formed without fertilization. You can give me your answers like A, B, C and D, as in 1A, 1B, 1C, 1D and things like that. Rathod, good evening. Ganesh Rathod actually. Good evening dear. Radhika, I'm sure. Okay. Vibha, yes, we can start. Come on. 1A, Sanjeevani says the answer for this question is A. Indra says it is C. So I'm getting questions like A and C. What about others? Shobha, I am going to finish this class by 11 p.m. and I'll make sure everything important is being covered over here such that you can grab that 70 on 70. Okay? Both of them are correct. Okay? Ishan says D is correct. Hello Aditya. Hello. Okay. Okay, so let's go ahead. Over here, formation of fruit without fertilization is called apomixis. Wrong. Listen to me very carefully. Here you have the ovary, Baba. Inside the ovary, what do you have? You have the ovule. Ovule, when fertilization takes place, here you have the egg cell, egg and the male gamete meets, and that's how fertilization happens, right? So when fertilization happens, I will call this ovule as what? I will call this ovule as seed now. And what do I call the ovary? I'll call the ovary as fruit. Now, apomixis is the formation of seed without fertilization. But over here, it is written fruit without fertilization. That is wrong. So, its first statement is wrong statement. Yes. Next thing, statement number 2. In some species of Asteracean grasses, seeds are found without fertilization. As in, basically, these two guys are the examples of apomixis. Is that true? Yes, obviously, these are the 2 examples of what you call apomixis. Statement 1 is false, statement 2 is correct, which is why your statement would be statement 1 is incorrect and statement 2 is correct. Your answer is going to be, my dear children, D. It is going to be D. So who all gave me the answer as D? So let me check it out once. Let me see who all got the concept. Oh my God. Kirtana, hello. Ishant gave me the correct answer, says D is the correct answer. And Kirtana also gave me the correct answer. Prajwal gave me the correct answer. And yes, Kirtana, Prajwal, anyone else? Hello, Chetan. Okay, so those of you who are getting your answers wrong, Should we be sad about it? No, you should be happy. Why? Because now you know the correct answer. Now you got an understanding that I can do silly mistake over here and probably I should not be doing that. Yes, ma'am, you rock. Thank you so much. You also rock, my dear children. So Arun, Chetan, Kumar, everyone, Madhuri, everyone, Pushpa. So all clear? If it is clear, can you just give me a quick thumbs up? All my champions who feels that this question is super clear, just give me a quick thumbs up. We'll go to the next question. Siddharud, hello. My dear children, forgive me if at all I am not able to read out your names. Sometimes it goes like this. Yes, ma'am. Thank you, Chetan, for telling it's clear. What about others? I'm waiting for the thumbs up. Yes, perfect. Arun, thank you, Sanjeevani, dear. Okay. Next question that we have over here. Listen to me. Sexual, Chetan Kumar, thank you. Varsha also, thank you. Listen to me. This is the questions which are there from sexual reproduction flowering plants that you should not be missing. These are the 2-marker, these are the 3-marker questions. I hope you guys know in 2-marker category, according to your model question paper, these 3 concepts are super important. Pollen grain, okay, true and false fruits, seed related concepts. They have more chances to come. That does not mean the other questions will not be coming in. Every question will have the probable chance of coming in, but you have to make sure all these questions are being sorted. Now I am going to discuss the things that I have written over here. The things that is being mentioned in yellow, I'll be discussing about that, since I said there are more chances for them to come in. Next thing, 3-marker. This is super easy. You can grab easy 3 marks from this chapter because you are going to get 1 diagram from this chapter. 1 diagram can give you 3 marks, so do not miss out on diagrams. We will be seeing that also over here. Hello, ma'am. Good evening. Hello, Mansi. Chetan, for everyone else, is my video getting stuck or is it okay? And let's go with the first concept. Why is apple called as a false fruit? Any idea why is apple called as a false fruit? Dear children, is the video getting stuck or is it okay? It's okay. Thank you. Okay, perfect. Okay, guys, so why is apple called a false fruit? Because, listen to me, a true fruit means what? Here you have the ovary, here you have the ovule. Ovary becomes the fruit. Ovary is going to become the fruit. That is how it usually works in. Right? Now, in the case of, exactly, Chris, it is not developing from the ovary. Exactly. Over here, in the case of false fruits, the thing is that there might be some other parts of the plant which is also involved in the fruit formation. For example, I see that there is a thalamus over here onto which the ovary is resting, and then you have the ovule. Suppose this thalamus is also contributing to the formation of fruit. I will call this fruit as what? False fruit. Now I'll call this as false fruit. You get it? In the case of apple, thalamus is something that is what we are eating. So when we eat crunchy apple, so right over here, I have an apple. Okay, so this particular apple that you see over here, the thing that we actually eat is the thalamus. It is not the ovary. The ovary is there. Do not be worried. The ovary is there. Where is it? Inside. So this particular picture is also important. Now this picture is given in your NCERT. Please know the labelings of it. It might come, we never know. So this is the thalamus, the outer portion. Over here you see the seed. Just outside the seed, this particular portion is what? Your ovary. The hard portion that you have, no, it's the ovary. Now if you dissect the apple like this, if you cut the apple like this and look at it, it will be looking like this. The outer portion thalamus, here you have the seed. Here, my dear children, you have the innermost layer of ovary that is known as endocarp. Then you have mesocarp, and the outermost layer will be known as basically exocarp. That is how it is going to go. Floral parts involved in fertilization. Exactly. Perfect, Shobha. Perfect, Ishan. Perfect. Okay, so Pushpa also, everyone is doing absolutely great. So I hope this concept is super clear to you. Can you guys give me some other examples of false fruits? So it is not just apple, there are some other examples for false fruits which you should be knowing. Any idea what are the other examples which are there? Any idea about the other examples for false fruit? Cashew. Simran says cashew. Varsha says strawberry. Chetan says strawberry. Okay. Madhuri strawberry. Sanjeevani strawberry and cashew. Exactly. Prajwal, Shobha, Ishant and Mansi. Madhav, Deepashri, Inchara. It's baby girl. Paru. Oh my God. So many people are there. Yes. The answer for that is strawberry as well as cashew. And in your NCERT it is written that both apple as well as cashew is developed basically from the thalamus. That is the other part of there. So I hope this particular concept is clear. If it is clear, just give me a thumbs up. We'll go ahead. Okay, so someone said, I do not know. It's okay. Now you know that, right? Now you clearly know that. So let's go ahead with the next concept which is out there. And by the way, whoever is watching this video, make sure you click on the like button, okay? And also the subscribe button. Over here, one more thing, some people have said banana. Listen, banana is not an example of false fruit. It's an example of Parthenocarpy. That's a whole different term, okay? In Parthenocarpy, what is happening? Basically, in Parthenocarpy, I usually said the ovary turns into a fruit when fertilization happens over here. Suppose there is no fertilization, the egg and the male gamete did not meet. Still, my dear children, the ovary will transform into fruit. In that case, if these guys did not meet, do you think here the ovule will become seed? The ovule is not going to become the seed, which is exactly why I'll say the ovule is not becoming the seed, and over here the ovary is turning into a fruit. That means this fruit is not going to have any seed. Seedless varieties of fruits are going to be formed with the help of something that is known as Parthenocarpy. And thank you for giving me the thumbs up. Let's go ahead with the next thing. So over here I have the picture. Can you guess what is this particular thing? Can you guess what is this particular thing which is labeled over here? This is strawberry. Which part of the strawberry is this? Anything? Ma'am, please be fast. Okay, I'll try my best to be more faster than this. Any idea? Peara. This particular part, this is strawberry, okay? I'm telling you this is strawberry, just tell me what is this particular part. Okay, Sanjeevani says thalamus. Okay. Rupashri, thalamus. Okay, why not you says thalamus? Exactly, my dear children. This is actually thalamus. I told you the thing that we actually eat in the strawberry is nothing but the thalamus. Let's go ahead with the next concept out there. So, JJ, talk about albuminous, non-albuminous seed and the pollen grain. So, let's go ahead and talk about the next 2-marker thing that can probably have a chance to come. That is albuminous seed and non-albuminous seed. Listen to me carefully. What do you mean by albuminous seeds? Albuminous seeds means you might know that there is endosperm being formed and there is zygote being formed, right? At the end of double fertilization, you get both of these things. The endosperm forms first, develops first, such that it can nourish the zygote to form what? Form the embryo. This is how it usually goes. Now, sometimes when the embryo is formed, it says that I do not want the endosperm out there. I will have my own nutrition unit. I'll be making my own cotyledons. I'll make my own cotyledons out there. You get it? So there are some type of plants where the embryo over here, the embryo formation over here is going to utilize all the endosperm up there. Why? Because they wanted to form the cotyledons. In such type of plants, you will not see any endosperm left behind. No endosperm is going to be left behind. These type of seeds are known as non-albuminous seeds. No endosperm will be left behind over here. It is going to be completely utilized. So endosperm is completely used during the embryo development out there. In some, the embryo feels that I have to look for the endosperm also, little bit endosperm, let it remain. So in some cases, it retains some endosperm even after seed maturation. That is albuminous seeds. Now listen to me. Over here, in the case of albuminous seeds, as endosperm is there, what does it do? Provides nutrition to the embryo from the stored endosperm only, do not have any other choice. But rather, in the case of non-albuminous seeds, nutrition is stored inside the cotyledons and not in the endosperm. So here it is cotyledon that plays a major role. Here it can be endosperm and also cotyledon, both of them actually, both of them. Now examples are super important for you. Do not miss it, okay? Do not miss it by any chance. Examples of wheat, maize, barley and castor. Over here, 3 of them are monocots. Some might think that castor, being a dicot, it has 2 cotyledons. How can it be coming under this particular thing that is albuminous seed? How can it still have some endosperm left behind? Because dicots usually do not have it. But this guy has it. So please remember, this guy is the odd one out. Castor is also an example of albuminous seeds. Here you have other examples such as bean, pea, groundnut. Most of the dicots which are there, dicots means the ones that have 2 cotyledons which can be broken like this, they are dicots. Most of them, they come here in this particular category. Castor is an exception, comes in that particular category. I cannot say that all dicots are non-albuminous. Okay. Did you get it? If yes, if you got it, give me a thumbs up. Everyone who got it, just give me a quick thumbs up out there. Hello, Irana. Hello, Jajappa. Nice name, by the way. Hello, Kushi. Okay. Ma'am, if we do this much, is it enough? If you do this much, it would be helping you really well for scoring 70 on 70. That is what I believe. Okay? Yes, obviously you have to go ahead and solve the other questions also, but yes. Deekshit, it will be ending at 11 p.m. To be honest, yes, perfect. Thank you, guys. Now listen to me. The next thing that I have to discuss over here is about the pollen grain. About pollen grain, you can get questions. So what about pollen grain is something that you need to know. The structure of the pollen grain is the first thing. It can be asked in a 2-marker question also to draw and show. Okay, so either to draw or to explain something related to pollen grain, it can come. So listen to me carefully first of all. The pollen grain, what is the first name that comes into your mind when you talk about pollen grain? It is known as the male gametophyte. What do you call it? It is the male gametophyte, my dear children. It is not the male gamete, it is a gametophyte. It has the gamete. It has 2 cells. One cell which is there, it has irregular nucleus. Irregular nucleus. This is a very big cell, which is why I will call this as vegetative cell. Very big cell, vegetative cell. Inside the cytoplasm of the very big cell, you have a small chunnu cell out there. This has an elongated nucleus. What do you call this particular cell as? Any idea? Any idea, guys? What do you call that small chunnu cell as? Chunnu cell. Oh my God. Tell me. Miss, for maths question paper, questions were not repeated from model question paper. I am not saying questions are going to be repeated from model question paper. Okay, questions might not, concepts might. You can get related concepts from it. Okay, vegetative and generative cell, exactly. So this is going to be the generative cell. This is going to be what? The generative cell out there. This generative cell is later going to divide into 2 to give us what? 2 gametes. 2 gametes, okay? So if at all a cell, pollen grain is released like this, I'll say it is in 2-celled stage. And basically only 40% of angiosperms, shed the pollen grains in this cell stage. Now the rest, what about the 60% of the left of the angiosperms? They are going to, sorry. Just a correction over here, it's basically ulta. 60% of them, okay, majority of them are going to release in 2-cell stage, and rest 40% of them, my dear children, are going to release in 3-cell stage. Ma'am, how 3-cell? Basically, this generative cell is going to form the gametes. So how does it divide it? Basically, it goes like this, and you have the 2 cells out there. What type of divisions are being undergoing over here? The divisions are mitotic divisions. What divisions are there? It is mitotic divisions that are taking place up over here. It has its own plasma membrane. Outside it, it has a continuous layer. What do you call this as? Intine. Intine is continuous, and my dear children, it is made up of Priyanka Chopra. What do you mean by Priyanka Chopra? Pectin as well as cellulose. And outside it, there is a discontinuous layer. What do you call this discontinuous layer as? It is known as exine, and exine is made up of sporopollen. It is made up of sporopollen. This is super important. This is the most resistant material out there. Even if you put alkali, acid, anything, okay, what is going to happen? It is not going to be destroyed. And this is the exact reason that the pollen grains can be stored as what? Fossils, my dear children. Super important. And the whole thing, you call this as gametophyte. Understand that the vegetative cell which is there, you also have another name for vegetative cell. It is also known as tube cell. Why? Because the pollen grain later on forms a pollen tube. And the tube, cytoplasm and everything is coming from the vegetative cell only. Fine? Okay? Yes, if you listen to the class, definitely you are going to score really well. Okay. How many mitotic division? Listen. First of all, the microspore is there. The microspore is there. Microspore undergoes 1 mitotic division. Okay. 1 mitotic division, you get 2 cells, that is vegetative and generative. Again, the generative cell alone undergoes the 2nd mitotic division to give the 2 male gametes out there. Okay. Perfect. Hasifa, good evening, dear. Fine. Now, listen to me very carefully. I have told you, I am telling you again, here are few other questions also which are given over here. Make sure you practice it. It is also important. Now, coming over here in the 3-marker section, diagram-based questions are gonna come. It is short gonna come. So, if at all you do not want your, 3 marks to lose, make sure you practice each of these diagrams out there. Tomorrow morning, sharp at 10 a.m., you are going to get a video on how you can make the diagrams as well. Okay? Fine. So these are the diagrams. All the important diagrams have been put up over here. Practice it. Next thing. Now, sexual reproduction in flowering plants, the 5 marker questions that have probable chance of coming is from the concept double fertilization, the gametophytes, the male and the female gametophyte out there, outbreeding devices, they have more chances to come. Okay, and over here we will be solving some application-based HOTS question as well. And starting with the first question out there, double fertilization is the unique feature of angiosperms, and the products of this double fertilization are zygote and primary endosperm nucleus. Okay, now in context of this, when a hexaploid plant is pollinated by a tetraploid plant, find out the ploidy or zygote and PEN. My dear children, listen to me carefully, it's important. First of all, understand the concept of your double fertilization. Suppose this is going to be your female gametophyte. What is your female gametophyte? We saw the male gametophyte, pollen grain. Female gametophyte is your embryo sac. It has 3 cells at the top. What do you call these cells as? You call these cells as antipodal cells. Antipodal cells. Okay, there are 2 cells in the bottom. What do you call them as? Synergids. And there is one big fancy cell out over here. You call this as what? Egg cell. That is your female gamete, right? And now in the middle you have 2 polar nuclei. These are nuclei, they are not cell. The cell is right up over here like this. What do you call this cell as? This is your central cell. It has 2 polar nuclei. What is the ploidy of each cells out there over here? Listen to me carefully. The ploidy of each cell is, this is going to be haploid, 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 haploid. What about this big cell out there? It has 2 nuclei, each of them haploid, haploid, which is why this is the only one which is diploid in nature. Rest all the cells are haploid. I hope you will remember that. Yes? Now, listen to me. When fertilization happens, there are 2 male gametes that are there in one pollen grain. One male gamete is going to come over here and fuse. Understand the logic. What do you get? You get a zygote. Okay, ma'am, what is the ploidy of the zygote? Usually we say half and half comes to form what? To form something that is known as 2n. Okay, it is known as 2n. That is diploid. Now over here another male gamete comes, it fuses with the polar nuclei of the central cell. Polar nuclei of the central cell, they are diploid in nature. One male gamete which is having haploid number of chromosome comes, diploid plus n, it becomes triploid. It becomes what? Triploid in nature. 2n plus n is equals to what? 3n. Now listen, in this particular condition, in the context given over here, hexaploid plant is pollinated by a tetraploid plant. Means the plant that is male over here, tetraploid plant, it is pollinated by a tetraploid. This is tetraploid. It is 4n in nature. Tetra means 4n. Normal cells are 4n in nature. The gametes will be what? The gametes will have only half the number of chromosome. So if I take the half of 4n, it will become what? 2n. My pollen grains which are there, the pollen grain which is there, it is having 2n number of chromosome. What about this particular thing? Listen to me carefully. It is hexaploid plant. That means it is 6n in nature. What is the ploidy of the egg cell? Half of it. What is half of 6n? It is going to be 3n. It is going to be 3n. Now listen to me. Pollen grain and the egg cell. Pollen grain and the egg cell. Sorry, that is the male gamete and the egg cell over here is going to be what? 2n plus 3n. Right? 2n plus 3n. Don't do into 3n. It is going to be over here 3n and this is going to be 2n. Plus you get 5n. You get what? 5n. This is important. This is first answer. That is for the zygote. What about the primary endosperm nucleus? So when I told you that both of them fuse, what do you get? You get a triploid primary endosperm nucleus. And now in this particular case, my dear children, you need to understand, we know the ploidy of the male gamete. Ploidy of the male gamete is what? 2n. Got it. But what about these guys? If it was hexaploid, okay, if it was hexaploid and the ploidy of each cell is 3n, the nucleus, there are 2 nucleus, both of them, my dear children, will be 3n and 3n already. Will be 3n and 3n already, this one and this one, right? Because all the nucleus are 3n in nature. Here you have 2 of them, so 3n plus 3n plus what? Your male gamete. So 3 plus 3 is equal to 6, 6 plus 2 is equals to what? 8n. It is going to be 8n in nature. That is your primary endosperm nucleus. Exactly. Zygote is 5n, Kirtana, and PEN is not 7n. It is going to be 8n, octaploid. Okay, it is going to be octaploid. So one is going to be pentaploid, other one is going to be octaploid. I hope my dear children are able to understand it. The 1914 returns. By the way, Simran Sheikh, hello. Madhav, Kesaiya, Sarah, and Alaina, Chetan, Kirtana, Pushpa, it's baby girl, Lollipop, everyone, I hope you're doing great. Yes, and also you're understanding the concept that is there. Understanding? If yes, just give me a thumbs up. I'll go ahead with the next one. So over here, I have given you the answer for the entire thing. Go through it properly and yes. Vinayak, hello. Why not you octoploid? I think there is some lag in the class, right? It's baby girl. Yes, my dear child, tell me. Chandrasekhar, ma'am, I'm not started bio. Still, can I get 70? Yes, you can. Try to get 70. We do not know whether you will get 70 or not, but you definitely can try. Go for it. Probably you might get also. Yes. Fixed 5-marker questions. So yes, fixed 5-marker questions will be sorted over here itself. Everything. Yes, ma'am, dope teaching. Thank you so much, Macha. Okay, next one. Explain the different outbreeding devices developed by flowering plants. This is another 5-marker concept that I'm expecting. It might or may not come. I do not know. I'm not sure about it. But yes, let's try it. Okay. So what do you mean by outbreeding device? Outbreeding means basically one plant, one flower is there, another flower is there of the same plant. They both are fusing. They are only tired because what you see in breeding depression, whatever toxic traits are over there, it is going to go on. Suppose this particular plant is not resistant to pest, it is not helpful in drought condition, then all the toxic traits are found to be passed on to the next generation also. We do not want this, which is why plants have come up with a new thing, that is outbreeding devices. And the first outbreeding device out there. Now for here, I want you guys to give me the answer because I see that it's me who is talking, right? So let's go. Outbreeding devices, tell me the names of the techniques out there. One is spatial separation of the things. Next thing that you have is what? One is spatial separation of all the things. Next thing that you can remember probably would be what? Deekshit, I hope you're fine, dear. And Tanish, PDF, you will be getting it post this particular live ends. The structural adaptation is formed by the plants to encourage cross-pollination and discourage self. Exactly. Okay, so Alaina is saying dichogamy. Okay, perfect. Non-synchronization, heterostyly, dichogamy. Okay, perfect. So you all have your own answer, unisexuality. Okay, listen to me. So the first one that I have over here is time difference. The easier language is what I have put up over here. Time difference means what? The pollen has released, but the stigma is not receptive. So who is going to take up this pollen? No one. So is here pollen release and the stigma receptivity is what? Not synchronized. That is what I mean by time difference. The second one is spatial separation. For example, over here you have the stigma. Baba, you have the stigma, but the pollen grain and everything is lying below. Do you think this guy can climb up there? Obviously, no, that is not possible. It is very difficult, right? So spatial separation is another thing that is being applied over here. The next thing that you have over here is what? Self-incompatibility. Now, this is a genetic mechanism by the plant. Plant itself knows that I am not going to allow any sort of thing happening up over here. Suppose the stigma is receptive. Okay, suppose the stigma is receptive, the pollen grains are released at the same time also. Still, when the pollen grains goes and lands over here, the stigma says like, no, that is not happening. I am going to prevent the pollen pistil interaction. I will not allow the formation of pollen tube. So this is a genetic mechanism that prevents pollen tube formation post what? The pollination process. Okay, now then what do you have? Then you have unisexual flowers. Now there are two conditions over here. Examples is what you need to remember better. Make sure you know the examples really nicely. First one is dioecious plant. Understand. For example, in human beings, you know we are dioecious. You have male and female. We are separate. Two different things, two different bodies. Similarly, there are some plants which will be having only the female flowers and only the male flowers. In that particular case, you just imagine one is having the female flowers out there and one is having the male flowers out there. The pollination has to happen like this only. That means cross-pollination has to happen, or what I call as xenogamy has to happen, right? This will anyway help in what? New genetic combination to form. This is one method. Where do you see this? This is more important. It's this case in the case of papaya. So they can ask you, the papaya shows what? Autogamy, geitonogamy or xenogamy? Understand this. They do not show autogamy or geitonogamy. Why? How can they show autogamy? Autogamy means flower is like this. From here to here it has to go. But now the flowers are in different plants. How do you think autogamy can happen? Geitonogamy also is prevented over here in the case of papaya. Only one thing can happen, and that is xenogamy. Now, on the other hand, unisexuality also works in the case of monoecious plant. Monoecious plants means what? They have both the sexes on the same plant. Okay, ma'am, if they have both the sexes on the same plant, how can they prevent everything? So listen to me carefully. If they have both the sexes on the same plant, it can be somewhere like this. One flower can be female, another flower can be male. This condition can happen, right? In this condition, my dear children, pollination has to occur like this. That means geitonogamy is going to happen. Okay, geitonogamy is going to happen. Now, again, genetically they will be similar only, but still, to some extent, you can prevent at least autogamy. So that is the only benefit of having monoecious plants. In monoecious, the sexes will be in the same plant itself. The example again is important. Chief Minister, please remember, C and M, castor and mace. Okay, castor and mace. Over here, geitonogamy can happen. Again, the good part of the story is that sometimes the pollen grain can land up there somewhere else doing xenogamy also. But anyway, autogamy would be prevented. Okay, I hope this is clear. So please remember the examples of this, and I hope this is clear. If yes, can you just give me a thumbs up, my dear children? And all of you who are giving the answers, I'm seeing you, okay? So Josna, hello, and Chandrasekhar, as well as why not you, Shobham, Chris, Mansi, Riddhi is baby girl, and Kesaya, and Kesia. And Rukmani, ma'am, I want the PDF, dear. You will be getting the PDF post the live. Okay, then only for example, any annotations are there, I have to write it now. So that's the reason I will be giving it after the live. I hope that works. Thank you, guys. Now listen to me carefully. Now, yes, we go on to the next chapter, but before I go ahead with that, I want your attention over here. Please make sure you go through the other concepts also. For example, artificial hybridization. Okay, artificial hybridization is a method that was being asked the previous year. With the help of that, you get hybrid varieties of plants. So the question can be coming like this. What is the method used by farmers to produce hybrid varieties of plants out there? It is artificial hybridization. In that you have certain processes, emasculation, where you remove the male part, and then you have bagging, and then you have the transfer of pollen grains, and then rebagging, and then tagging. So please know about this chutu things also. It will be really helpful for you guys. Okay, and anyone who is out there, can you tell me one very important thing? What do you call the wall layers of ovary? What is the wall layer of ovary known as? What is the wall layer of ovary known as? Perfect. Tagging, emasculation, bagging. Yes, Kushal, tell me. Ma'am, video is getting delayed. Ma'am, you're the best teacher. Thank you. With the video, I think, yes, it is getting delayed, but yes, Alena, please go ahead with the advantages of artificial hybridization only. Also, what do you get basically with the help of artificial hybridization? You get new varieties of plants, and these varieties of plants can sustain in different environment out there. Right? And you also have the right to choose the plants out there, which makes things more clear and easier. Okay? And Kushal, the explanation of what is needed, are you asking about this? There are time difference. Time difference basically, I'll just quickly wrap up, okay? Time difference means pollen release is there, but the stigma is not receptive yet. So that is one way of preventing pollination. Second thing, spatial separation. The stigma is somewhere over here, the pollen is somewhere over here. Do you think fertilization can happen easily? No. Third one is self-incompatibility, where the pollen grains lands on the stigma is receptive also, but still does not allow the formation of pollen tube. That is self-incompatibility, is a genetic mechanism. And the last one is the production of unisexual flowers, where a plant will produce probably one type of flower that is female, another plant might produce another type of flower that is male. So which is why cross-pollination is going to happen. Example is papaya, that is dioecious plant. Okay? Okay. Ma'am, is it compulsory for new students of Karnataka to write KCET? I have already registered. No, you should not be attempting it. You do not need to write the KCET examination. You just need to, register for it. Okay? Yes. Perfect. So, my dear children, apart from it, you see the questions over here. I have given you so many questions over here. Make sure you go ahead and practice all these questions. These are important. Okay? These are important. And Chris says exocarp, mesocarp and endocarp. Yes, that is the pollen grains layers out there, exocarp, mesocarp and endocarp. Perfect. Okay, now just give me a quick second, okay? Guys, I'll just check with the team if at all there is a way to fix the lag, because I also could see that there is a lag. Okay, just give me a second. Okay, guys, so some of you were saying that the video is lagging, which is exactly why I'm asking you. So if it is lagging, I might have to do one thing, that is switch off the whole setup out there and start up in the same live link only. It will be there. Is it okay, guys? Yeah, because I see there is a huge lag. If it's okay, just give me 5 minutes. Okay, if it's okay, give me 5 minutes. I'll just restart it and I'll come back. That works. If yes, give me a thumbs up, because I know it will be lagging. It does not, it is actually lagging. I could see that. Okay, so give me 5 minutes. So I'm just writing over here. Gia, we are solving the most important questions. Do not miss it out. Yes. Okay, children. Am I audible now? Is it clear now? Is it visible? Yes, perfect. Thank you, thank you, thank you, thank you, thank you. I think still the lag has not been fixed, but okay, anyways, so what we can do is what we can do. That's all, right? So fine. Okay. So listen to me carefully. Now what do we have over here is the next thing, that is what the next chapter, which is human reproduction. I hope every one of you will be doing all the questions which are there that I have mentioned already. Now when I start with this question, I want everyone to give me the answer like 5A, B, C, D. Identify the wrong pair with respect to the menstrual cycle. We have solved this question before also. Formation of corpus luteum happens in 15 to 28 day, secretion of progesterone 15 to 28 day, regeneration of endometrium 5 to 13 day, disintegration of endometrium 5 to 13 day. What do you think is the answer for this particular question? Any idea what is the answer for this particular question? 5C, Vinayak Poli says the answer for this question is C. Hassan says, hi Hassan. Kesaya says it is C. Ishant says C. Kushal also says C. Riddhi says C. C is what? Regeneration of endometrium happens from 5th to 13th day. Is it C? Guys, think about it. Listen, we need to solve this question. Come on, think about it carefully. Is the answer C or something else? Are these questions going to help in our exam? Definitely. Shravan, Okay, Chandrasekhar says D. Kavanashri also says D. Deepthi also says D. Yes, the answer for that is actually D. Listen to me carefully. This is how it goes. You have the first day and you have the 28th day. This is how the menstrual cycle goes on. Yes, half of it will be 14th day, right? Half of 28 is 14, and that is when ovulation is going to happen. Right? Yes. Now, from 1st to 5th day, my dear children, that's when the menstrual phase is there. That's when the endometrium is going to get disintegrated. You will be getting the bleeding out there. Now, from 5th to 13th day, which you see over here, this guy which has went down will be slowly forming back. You see regeneration happening up over here. Why? Because it is under the influence of estrogen. Estrogen, progesterone both went low over here. But now estrogen came into the picture, which is exactly why there is regeneration. Now, when the level of estrogen goes up really high, it also makes the LH go really high. When the level of LH goes really high, it leads to LH surge, leading to what? Ovulation. The follicles are going to burst open and is going to release what? The secondary oocyte. Now, that is known as ovulation, right? And whatever is left behind in the follicle, apart from the secondary oocyte, what do you call that thing? That is known as corpus luteum. It is known as corpus luteum. And what is the use of this corpus luteum? This guy, my dear children, is going to release progesterone as well as more amount of estrogen. Now, when progesterone is released, until now, regeneration was happening, but now it will get super thick over here. Why? Because progesterone is something that is responsible for thickening of your endometrium. This is super important. Now, listen to me. What is wrong pair with respect to menstrual cycle is being asked. Formation of corpus luteum from the 15th to 28th day. Yes, it happens. It happens. Corpus luteum is there. Secretion of progesterone 5th to 18th day. Yes, Baba, it also happened. Secretion of progesterone because corpus luteum is there. That is why secretion will be there. Correct. Regeneration of endometrium from 5th to 13th day. That is also happening over here. Disintegration of endometrium from 5th to 13th day. Wrong. It is happening in the 1st to 5th day. 1st to 5th day. So this is why it is wrong. Got it? Got it? Children, guys, please do not talk amongst themselves. Ask yourselves. It is really bad. Over here, I am yelling at the top of my voice and I am trying to put 100% of my energy and you guys are talking to each other. That's so sad, right? Yes. Okay. So, is it clear, this particular question? Super clear. If yes, give me a thumbs up. We'll go on with the next question. Okay, the answer is D. Do not get it wrong. We have only one day, ma'am. We want all the 2 and 3 and 5 mark important questions. In the channel, it has been given there. Can you go check it out? Okay, there's a video that has been released. Okay, hi, and thank you Sanjeevani and Hassan and Vinayak, Police, everyone out there who said yes. Now, here, the menstrual cycle is super important. If you mess around here, you will mess around. A lot of things are there. So, do not mess around the menstrual cycle. Okay? Fine. Now, coming to the month by month data, this is also something that is important, can give you 1 marker question out there, an MCQ question may come from here. So, please know about the month by month data. That is what happens during each month. First month, my dear children, when someone dies, what do? You check up the heart, right? Heartbeat is there or not. Similarly, when someone forms, the first thing that you check is whether heart has been formed. So, heart formation happens in the first month. By the end of the first month, what you will get? You will see heart formation being taken up. Now, the next thing that you have is second month, limbs and digits are going to be developing. In the second month, my dear children, there will be limbs and also there will be digits that are being developed over here. In the third month, my dear children, most of the organ systems and everything is going to be formed, and also the external genital organ. That is basically, in the case of males, the penis, in the case of females, the mons pubis and everything is going to be formed. So first month, heart. The second month, limbs and the digits. The third month, basically organ system and the external genital organs out there. Now fourth month, break. Fourth month, there is a break. It is not given in your NCERT. What happens in the fifth month? Fifth month, baby says that, God, what, Macha, you are doing something like this to me. I want to move around. No. So please make me move. So first fetal movements are observed in the fifth month, and not just that. Basically, there will be, and the baby also says one more thing. I want to show off in front of girls. Give me some hair also. So there will be hair on the head as well. So first forget. First fetal movement, and also what do we have? Hair on the head. Now God comes and says at the end of the 6th month that, hey, you wanted hair. No, have hair on the body also. So there will be fine hairs covering the body in the 6 months. The eyelids which are there, they are going to get separated. Eyelashes are also going to be formed over here in the end of these 6 months. Now listen to me very carefully. This is also super important. Month by month data, you know. Sometimes you do not understand when they ask you in terms of week. 1 month contains how many weeks? 1 month, my dear children, contains 4 weeks. It contains 4 weeks. 2 months contains what? 2 into 4, right? 2 into 4, that means 8 weeks. So if at all you get a question like what happens at the end of 8 weeks, same thing, limbs and digits and everything are going to be formed. What do you mean by end of first trimester? That means end of first 3 months. And 3 months basically means 3 into 4, 12 weeks. You get it? Now 5th month is done. End of second trimester, that means the 6 months are being done over here. And by the 6th month, my dear children, what do you get? 6 into 4, 24 weeks. This is super important. Do not mess around here. Okay? Ma'am, please send the MQP 1, 2 and 3. Dear, I have sent it in the WhatsApp itself. There is a playlist that is given to you in the previous messages. If at all you roll down, okay, you will be able to see the PDF link. Over there, you will be able to find it. Okay? Children, is this particular thing clear? Listen, one very important thing. If you are not cooperating with me, just imagine my plight. Okay, if at all you guys are not cooperating with me, not talking to me, rather you're talking to each other, do you think your teacher would be able to happily teach you guys? No. Whose exam is this? Is it my examination? No, it's your examination. Who is putting the efforts as of now? Me and you both, right? Do you want these efforts to go on waste? No, right? So please make sure you make the best use of it. Okay? Fine. Now go ahead with the next thing. In this chapter, in human reproduction, you are going to get a 3-marker question. There is no 2-marker questions from here. Now, anything related to menstrual cycle, spermatogenesis and placenta have more chances to come. That does not mean other questions cannot come. There are other questions also which might be coming in. And there are certain new questions that I've added up. Briefly explain the process of fertilization in humans. When and where do chorionic villi appear in humans? State their function. So, we'll be starting off with the first question related to spermatogenesis over here. And that goes like this. So this is a 3-marker question. It's a hot question. Make sure you listen. Okay. So study the following flow chart. Name the hormones involved at each stage. Explain their function. Hypothalamus, pituitary, testis and the sperm. So you have to tell how does this particular flow works and the hormones which are involved in each of the stages. So my dear children, I hope you guys know. If at all you do not know, listen to me out carefully. This is super important. Okay. That is, it goes like this. You have to listen to me. You have your hypothalamus. Hypothalamus is known as what? The pardada, the grandfather of all the hormonal glands out there. The hypothalamus tells everything to pituitary and the pituitary gland is going to obey that. What does the hypothalamus tell to the pituitary gland? Hey, pituitary, release the gonadotropins. Release what? The gonadotropins. Gonadotropins are nothing but the hormones that acts on the gonads. Okay, so it gives a signal by releasing gonadotropin releasing hormones. It says that I am releasing gonadotropin releasing hormones. This is a signal to you guys to basically release 2 hormones, that is LH, luteinizing hormone, and also FSH. So this guy releases this guy releases 2 of the hormones. Remember, pituitary gland has 2 lobes, anterior lobe and posterior lobe. Which one is affecting over here? This one. So this is the anterior lobe which is going to release LH and FSH. And I see a lot of you giving me the answers. Why not you and everyone? Perfect. Let's go ahead. Now, what does the LH do and what does the FSH do? Listen. Here you have the testis. In the testis, you have the testicular lobules. How many? 250 of them, right? If I zoom them in, basically this is a testicular lobule and it will be having seminiferous tubules here. Right? There are seminiferous tubules over here. Inside the seminiferous tubule, what is happening? The sperm formation happens up over here. Okay, what happens in between? There are Leydig cells which are going to release testosterone. Who gives the information to release testosterone? The LH. The LH acts on the Leydig cells. Remember always. And now the Leydig cells is going to release testosterone. Very, very, very important. Okay, now why do we need testosterone, ma'am? Listen, how do you think sperms are going to be formed? They need hormones. Where do they get the hormones from? From here. So from here the testosterone is going to travel into the seminiferous tubule. Okay, ma'am, but what is exactly happening in the seminiferous tubule? In the seminiferous tubule you have 2 cells. One is Sertoli cell, the other one is what? The other one is the spermatogonia, which is undergoing division. So listen to me, coming to spermatogonia, it basically underwent differentiation. You have got primary spermatocyte. This is the primary spermatocyte. It underwent meiosis. You got 2 cells. It is the secondary spermatocyte. It underwent Second round of meiosis to give 4 cells. What are these 4 cells known as? They are known as spermatids. They look like kids. They are immature. Spermatids, kids, immature. Remember like that. Basically, not you guys, okay? Small kids. You can think like that. Why do I say immature? Because they are globular in shape. Are sperms globular in shape? No, Baba, they are elongated. So what do you call it? Spermatids. They are what? Spermatids. And my dear children, the whole process is known as spermatogenesis. Why I am stressing on this? Listen. These spermatids have to transform into sperms, the actual sperms which looks elongated, and that is known as spermatozoa. Spermatozoa. And this process of transformation of spermatids into spermatozoa has a different name altogether. It is known as spermiogenesis. Spermiogenesis. So, ma'am, you are saying that within the seminiferous tubule, you have spermatogonia, and that is undergoing this. But what about the Sertoli cells? What is it doing there? Listen, the Sertoli cells over here, the FSH which is there, it acts on these Sertoli cells, and the Sertoli cells are going to release some factors over here. What does this factor do? This factor says that, hey, whenever the testosterone comes inside, I will make sure that the level of testosterone is maintained such that the whole process happens really, really well. So what is the role of the factors? To make sure the whole process happens really well. But majorly this particular process, that is spermiogenesis. So if you're asked this question, if FSH is not being released, which is the stage which is being affected? It is the formation of spermatozoa which is majorly being affected. The whole definitely, but majorly this particular process is being affected. Please know about this. This is super, super important. Okay, so LH, FSH. LH acts on the Leydig cell. It releases testosterone. FSH acts on the Sertoli cells. S over here, S over here. Sertoli cells release the factors, and factors maintains the level of testosterone, and over here it is mostly needed in the process of spermiogenesis. Okay, and ma'am, we need CS lectures. I definitely understand you. There will be CS lectures on this particular channel. Do not worry. And I believe that ma'am is coming tomorrow at 9 a.m. Sorry, 8 a.m. to do a live as well. Okay, I hope user not fine. I have got you cleared. And yes, Purvika. Okay, how long will the stream be? It will be ending at 11 p.m. is what I'm believing. Children, is this particular thing clear to you? If yes, give me a thumbs up. Everyone who feels that this particular thing is super clear to you, give me a thumbs up. This is important, okay? These are the hormones of spermatogenesis. Thank you, Sanjeevani, Hassan, and Jia, 11 p.m. exactly. Do not worry, I am not going to stretch it until the morning, 11 a.m. Okay, 11 p.m. I will make sure that everything that is super important for you, okay, which has the most chances of coming, I'll cover by 11 p.m., not beyond that. Tomorrow, we'll just do the passing package live. Apart from it, you get normal lectures. That's all. Okay, come on. And this is for you. You know what? I didn't sleep yesterday because of some reasons out there. There were some things happening up there. But I still came over here, thought of doing this live today itself, because tomorrow you will get your time to do your self-study. I do not want to hamper your tomorrow. So please, yes. And yeah. So over here, your answer goes like this. Make sure you write it like this. Hypothalamus hormone is GnRH. That is what is releasing. Function is what? Stimulates the anterior pituitary to release gonadotropins. Okay. What does the pituitary do now? Hormones are LH and FSH. FSH acts on Sertoli cells and stimulates spermatogenesis. LH acts on Leydig cells of testes to stimulate testosterone secretion and stimulates the release of factors. First of all, release of factors. You have to write about that. Okay. I have missed it over here, but write that. Now, testis, what does it do? Hormone is testosterone and function is promote spermatogenesis and development. And the sperms, what about them? The sperms, my dear children, that is not being repeated. Okay, so what are the hormones has been asked? I have mentioned about all the hormones. Okay? Yes, perfect. Alena, ma'am, can we get a break? That is too much to ask. From 6:20 to 7, that is a huge lot of time. I'll try definitely to give you a break in between, okay? But I'm not sure that long break or not. Yes. Okay, fine. Let's go ahead with the next question out there, and here you go. 3-marker question, again a hot type of question, please know. Try to solve it, everyone. Everyone, please try to solve it. Ma'am, how many meiosis and mitosis division takes place in male and female gametogenesis, both male plants? Oh my God. Satya, in the previous one, sexual reproduction flowering plants, I spoke about that. Here in the case of animals, I am telling you, basically, one round of meiosis happens. Okay, meiosis happens in 2 stages. I hope you guys know that. One stage where you get the secondary spermatocyte or secondary oocyte. The second stage where you finally get the ovum here, and here you get the spermatids at the end. So one meiosis is going to happen each of them. Okay. Come on guys, solve this. Given below is a flow chart showing ovarian changes during menstrual cycle. Fill in the spaces with the hormonal factors responsible for the events being seen. So here you have to tell the hormones which are being involved over here. Primary follicle dash Graafian follicle. Primary follicle dash Graafian follicle. So which is that hormone that will make the primary follicle turn into Graafian follicle? Everyone, come on. Can you tell me which is the one that is going to make the primary follicle turn into the Graafian follicle? Any idea? Up over here. Ma'am, what is placenta? We'll be discussing about that. Okay, there is a question. Okay, FSH. FSH is what you are saying over here for A. Yes, A is FSH. Okay, A is FSH, not LH. Voila. Why FSH? Yes, exactly, Sanjeevni as well as Hassan. The reason why FSH is it's follicle stimulating hormone. It stimulates the follicles, right? Primary follicle to Graafian follicle is being stimulated with the help of what? FSH. Okay, now done. Now over here, Graafian follicle, then you have ova. What do you mean by that? Basically, Graafian follicle is going to what? Rupture, and it is going to produce ova. How can it rupture? With the influence of what? Which hormone is playing over here? Which hormone? Yes, Nagaraj also FSH, Kirtana also. Okay, no FSH. Yes, Nagaraj says B is LH. What about others? About B. So A is FSH. What about B over here? B is LH. Rudra Muni Ayappa is also saying it is LH. Kesaya, it is also saying LH. Mansi also LH. Soul Mani LH. Everyone says LH. Yes. LH surge leads to what? Ovulation. That is how you get the ova. Now the ova is there. You form the corpus luteum also. So whatever is left behind is known as corpus luteum. What does the corpus luteum release? That is the question. Okay, C is basically what does the corpus luteum release? Any idea what does the corpus luteum release, Baba? Any idea about what is the corpus luteum going to release? Progesterone. Exactly, Hassan. And exactly. And Rupa, hello, hello, hello. Satvik, hello guys. Hello. Okay, Mansi says progesterone. Perfect. And Deekshit, I think you are solving the previous ones. Yeah. So over here, can I write the answer to be like this? The first one is going to be FSH because it's the one stimulates primary follicle into secondary, basically Graafian follicle. LH basically causes the surge for ovulation to happen. That is why you get the ova. Corpus luteum is the one that releases progesterone. Now there's one more question over here. Women experience two major events in their lifetime, one at menarche and the other one at menopause. Mention the characteristics of both of these events. What are the unique characteristics of both of these events? Both of these events, can you tell me? Of menarche and menopause. Menarche means basically what? The start of periods, right? The start of the reproductive cycle. Menopause means what? The stop, the ending phase of the reproductive cycle out there. Cool? So, menarche means first onset of menstruation at puberty, marks the beginning of the reproductive phase, and menopause is the permanent cessation of menstruation. And over here, yes, that's all. So this is the thing. Okay? Is this clear? Is this question clear? If yes, can you give me a thumbs up, guys? If yes, if this question is super clear to you, give a thumbs up. Sunanda, yes dear, I will be doing fast. Hi JJ ma'am, hello Vinay. Miss, these are the characters? Yes, these are the characters. Apart from it, you do not have anything else. Now, Vinayak Police Patil says, give me a thumbs up. Thank you so much. Okay. Now, okay. Listen to me carefully. Now, I'm going to the next phase. That is the 5-marker section. Before that, I have a question for you guys. Vote for this. How many of you need an understanding of the menstrual cycle as a whole? I can explain to you in 5 minutes. And menstrual cycle can give you questions, which is why I'm asking you again. Do you need an explanation of menstrual cycle? I can explain you in 5 minutes. If yes, type yes. If yes, type yes in the chat section right now. Give PDF, ma'am. PDF, you will be getting it post this particular live, dear, you will be getting the PDF with annotations, okay? Yes, ma'am. Okay, me. Okay, yes. Perfect. So let's listen. Listen to me clearly, and I'm pretty sure after this you will not do any mistakes in menstrual cycle, okay? So over here it goes like this. First day, and you have 28th day, right? Half of it is known as basically 14th day, and that's when you will have your ovulation, is what we believe. Now, what exactly happens is that two hormones, the level of two hormones, that is estrogen and progesterone, their level goes down. And these two hormones are the one that maintains the endometrium, which is why what is going to happen? The endometrium is going to shed down. That means you are going to get your periods. Now at the same time, my dear children, these two are what? Ovarian hormones. Your ovaries are going to release them. At the same time, there are some pituitary hormones which says that, oh my God, these guys have went down. Now it's time for us to go up. So can I say basically these two guys are enemies? Yes, somewhat they are like that. So this time when they go low, slowly they start increasing FSH and LH. And you might be knowing this particular phase where menstruation is happening, you call this phase as what? Menstrual phase. Slowly, since FSH is increasing on, the follicles which are there, they are also going to mature. Why? Because FSH is increasing. But one thing happens under the nose of FSH, and the FSH would not know it. That is, when the follicles are maturing, you know, granulosa cells are being formed over here. These granulosa cells are going to release estrogen. The dushman, the enemy, is being released over here. So slowly what is happening, the level of estrogen is increasing. Since the level of estrogen is increasing, you know this guy was important for the maintenance of endometrium. The endometrium slowly starts regenerating, which is why I'll also call this phase as what? I'll call this as proliferative phase. Why? Because my endometrium is slowly regenerating. That is why I'll call it proliferative phase. And the same time, I'll also call this follicular phase. Why? Because the follicles are also generating up over here because of FSH. Now what happens? Listen to me. Estrogen is being released. When estrogen is being released, it says that FSH, you go down. It goes down also. It goes down also a bit. Same thing happens with LH also. It goes down. But one fine day, one thing that is so uncommon happens. And what is that thing? Estrogen level goes up really high because the follicles are generating, right? Estrogen level is going to go up such high, such that it becomes so happy. And when we are happy, sometimes we let the enemies also be happy, right? Similarly over here, FSH and LH also become so happy such that there is a huge spike in LH, and that is known as LH surge. And that's exactly when ovulation is going to happen. It happened because of what? Estrogen rise first. Then because of LH, ovulation happened. Majorly because of LH, but who was responsible for this? This guy. Now ovulation happened. What happened after ovulation? Your graphene follicle which is there, it is going to rupture and it is going to release what? The secondary oocyte. Whatever is left behind over here, my dear children, what do you call this? This thing is known as corpus luteum. It is known as corpus luteum. Who was responsible for corpus luteum, Baba? It was the LH who was responsible for formation of corpus luteum, because it is the one that caused the surge, right? So I'll say Loki, still LH is the one that is responsible for corpus luteum formation only, and that is when we say that we are entering the luteal phase. We have entered the luteal phase. The corpus luteum which is there, it is now going to release the most important hormone, that is progesterone, and it also releases estrogen, but majorly this guy. So the endometrium which was just regenerating, it is not, oh my God. Will I get it back? Okay, so anyways, where were we? Okay, I'm not going to make it again. It will waste a lot of your time. We're over here. You remember what did we have over here? We had corpus luteum. Yes, we had corpus luteum. This is 28th day. This is 14th day. And over here, LH spike happened. And LH was the one maintaining corpus luteum, right? The corpus luteum over here is going to release progesterone and also estrogen. The progesterone which is there, it is going to make sure endometrium becomes very, very thick. And not just thick, it starts releasing certain very important factors. Why? because you want, okay, why? Because you want these, when the sperm and the egg meets to form the zygote and the zygote comes here to the endometrium, you want the zygote, or basically not the zygote, the embryo to get nourishment. That is why it makes the endometrium release certain things like glycogen and things like that, which is why I'll not just call this phase as luteal phase, I will also call this phase as what? One more phase, which is known as secretory phase, because your endometrium is secreting. So always remember, the hormone which is involved in regeneration is estrogen. The hormone which is involved in implantation means embryo to come and attach over here and do all these things, that is progesterone. Now listen, there is a twist over here. I told you progesterone as well as estrogen are enemies with LH and FSH. When the progesterone is being released over here, it says that I do not want the LH to be released. Make sure the LH is not there, otherwise I will not be released. But sometimes we have to be grounded in our life. Same thing happens over here. If LH is not there, corpus luteum is not there. If corpus luteum is not there, progesterone is not there. If progesterone is not there, endometrium will also not be there, which is why again the menstrual phase starts up. On the other hand, if at all fertilization would have taken place, even though progesterone will say LH to go down, there will be someone else to maintain the corpus luteum out there, and that is known as human chorionic gonadotropin hormone, HCG, you know, placental hormone. That guy maintains the corpus luteum. I hope this is clear. And now whatever is left behind over here is known as corpus albicans. Yes. Children, is it clear? I know in between the slide only went up ahead, but yes. Hi ma'am, hi. Hello, hi. Ma'am, speak loudly. Is it? I should speak loudly. Okay. Fine. So this is all about that, and yes, that's all. Cool. Let's go ahead then. I hope now you will be able to solve questions from menstrual cycle. You should be okay. You should be able to do that. And let's go ahead. Now we enter the 5-marker zone of human reproduction. Listen to me very carefully. This is super important. You can grab easy 5 marks in this session by practicing these diagrams, because a short diagrammatic question is going to come from this particular chapter, that is human reproduction. Do not miss out on this diagram. It can be female reproductive system, can be male, can be mammary gland, can be schematic representation of spermatogenesis, or basically oogenesis also. Pooja, hello, have taken your name. Okay, now over here, you can also get questions like this in diagram. This is something that is uncommon, has not come. So draw a sectional view. So I think I have put as a separate question itself. Yes. Draw a sectional view of seminiferous tubules of human and label the following cells in the seminiferous tubules. So you have to draw and you have to label them as well. This type of question can also come in drawing part. Yeah. So please know that. What is that? Cells that divide by mitosis to increase their number. Cells that divide by mitosis to increase the number. So over here, I hope you know spermatogenesis, how it works. It works very simple. You have spermatogonia. Okay, you have a diploid cell that is known as spermatogonia. This spermatogonia is going to undergo mitosis. What does it undergo? Mitosis. And it forms 2 cells. One cell will later form, again divide to form lots of spermatogonia. The one cell which is being formed over here, it differentiates. Okay, it differentiates into what? The primary spermatocyte. It is the one who says that I want to form the sperms later on. So we say that it got differentiated. It is mature now. Right? Now what happens to this mature primary spermatocyte which is diploid in nature? It undergoes meiosis. Meiosis 1, when it gets 2 cells. These 2 cells are going to be haploid. There's a reductional division happening up over here. So always remember, if at all you're talking about haploid number of chromosomes up, right? So it is going to be what? It is secondary word. The term would be secondary. So this is the secondary spermatocyte which is being formed over here. Now what happens to the secondary spermatocyte? Again it is going to divide, Baba. And now you tell me, what does it form? The second meiosis that happens at the end of it, what do you get? Ma'am, CS. Sujan, dear, you will be getting CS lectures also. Do not be worried. Okay? What do you get over here at the end of things? Any idea? Spermatids, exactly, Hassan. The answer for that is spermatids. You are going to get spermatids at the end. So there will be spermatids, all of them are going to be haploid in nature. Later, after spermatids are being formed, they transform into what? Remember, spermatozoa. Spermatozoa, that is the elongated form of sperms. What do you call this process? Spermiogenesis. Do not mess it around this, please, I'm telling you. And finally, the sperms are going to go. That is known as aviation, that is spermiation. Spermiation, aviation, remember like that. Okay, exactly, everyone out there, Shravan Joshi and Sanjeevni and Rudra and Munaza, Mansi, Riddhi, Khesaya, Rizwan. I see some people have been there from the starting, Alayna, Vinayak and Varsha. I'm super proud of you. To see you still there, okay, because I know multiple channels are doing live at this particular point of time and there's a natural tendency to go to your regional language, but I'm still glad you're here. Okay, anyways, the cells that divide by mitosis to increase their number is what? The cells that divide by mitosis to increase their number is what? Your answer for this particular question becomes spermatogonia. Spermatogonia. Okay, second one, cells that undergoes meiosis 1. The one that undergoes meiosis 1 is going to be known as what? Primary spermatocyte, Baba. It is going to be the primary spermatocyte. Okay, fine. The cells that undergo meiosis 2. Now this you answer. The cells that undergo meiosis 2 will be known as what? First polar body, that is happening in oogenesis, not in spermatogenesis. Okay, the cells that undergo meiosis 2 is what? It's okay. We all get things wrong sometimes, okay? Secondary cell law, secondary spermatocyte, okay? It is known as secondary spermatocyte. Exactly. Okay, now what do you have? Cells that will help in the process of spermiogenesis. Now you tell me. This we have studied earlier. I'm slow in typing. It's okay, dear. Okay, cells that help in process of spermiogenesis. That helps in the process of spermiogenesis. You have to think about it. We studied it in FSH, LH and stuff like that. Cells that helps in the process of spermiogenesis, as in conversion of this to this. And I'm waiting for your answer, then only I'll go ahead. D. Sertoli cells. Awesome. Yes, it is Sertoli cells. I told you Sertoli cells release the factors and the factors are going to help especially in spermiogenesis. So over here, your answer is going to be Sertoli cells. Okay, now perfect. Rudra and Sanjeevni, A, D, V, K. Okay, Alisha, hello. So many new children are there. Okay, Tarun, you get full and full. But listen to me carefully. Cells produced due to spermiogenesis. What are the cells that are produced due to spermiogenesis? At the end, what do you get after spermiogenesis? After spermiogenesis, you get something. What do you call those cells as? Mumtaz is also here. Mumtaz Begum, hello. Spermatozoa, exactly. You call them as spermatozoa. Done. So you can get this type of question, and when you get this type of question, that means that you have to draw it and show. So how do you draw, Baba? You just have to draw like this. So my dear children, the lowermost cells which are there, they are going to be spermatogonium. Okay, they are spermatogonium which are there, which are finally dividing. So you just need to understand that after spermatogonia, you make a next cell out there and you name it primary spermatocyte. At the top of it, my dear children, make more cells and write it secondary spermatocyte. Okay, at the top of that, you basically made spermatid. So here spermatid is been mentioned wrongly, actually should not be spermatid, but anyway, this is how your NCERT processes, so you can also go ahead with that. Okay, so this is spermatid and then you have spermatozoa. So the only thing that you have to keep in your mind is whenever you're making this, the lower one should be spermatogonia, then primary spermatocyte, then secondary spermatocyte, then the spermatids, and then the spermatozoa. Label like that. And in between that, if at all you see Sertoli cells. Okay, the Sertoli cells are also here and there, so please mention that also. This is what you have to keep in your mind. Okay, fine. Yes, section-wise analysis have been done there, and that's how I made this PPT. Cool. Shall we go ahead then with the next thing that is out there? So these type of questions you can expect, and apart from that, the diagrams which are there, this one, tomorrow morning 10 a.m. video is there where I have what do you call I have made it and I have shown you how to make it. That will definitely help you guys. Apart from it, there is a diagram like this also. Spermatogenesis, oogenesis and menstrual cycle I have told you, and then this particular diagram is there where please know that morula, which is there, a stage of embryo, it's formed here in the fallopian tube, okay, and then the blastocyst, which is a stage that is rare for implantation, that is here in the uterus. Please know, blastocyst is a stage at which implantation is going to happen. Okay? Cool? Ma'am, mammary band is really tough. It is going to be. So, watch the video that is there tomorrow, that is going to come tomorrow morning. It will definitely help you out. If you think it is tough, it is definitely going to help you out. Listen, I'm not Picasso, but yes, this video could help you. And listen, my dear children, there is a picture, okay, which I have missed over here somehow. You should know that picture. That picture is there in oogenesis. If at all you have seen the picture of oogenesis, right? The what do you call the follicles which are generating and the follicles which are degenerating. All these things are being given over here. So please make sure you go through that particular picture. Write it somewhere. So homework for you: oogenesis picture. Oogenesis picture. Okay, cool. Now listen to me carefully. Over here, I will be just explaining you something about oogenesis also. It goes the similar way only. Know the basic thing. This is oogonia. Oogonia also undergoes mitosis. Okay, one cell which will be there, it is going to differentiate into what? The primary oocyte. Primary oocyte. It is also going to be diploid in nature. Now it undergoes meiosis, but this meiosis is what? Unequal division. Meiosis 1. So you get one cell big and one cell chutku cell only. They are haploid in nature. This is going to be the first polar body. Someone was asking me that, Pooja. I hope now it will be cleared. And here you get the secondary oocyte. Always remember, from the secondary phase onwards, it becomes reduced. Haploid number of chromosomes are gonna come. Okay. Now, ma'am, louder. But here the mic is working really well. More louder. Is it so? And now, again, when you divide it, you get unequal division. At the end, you get the second polar body, and here you get the ovum. Here you get the ovum. This is meiosis 2. Listen, meiosis 2 happens when fertilization is gonna happen, not after fertilization. You only think. You have to listen to me very carefully, okay? Some people ask that when does meiosis 2 happen? After fertilization, after fertilization, how can it happen? You need ovum for fertilization. No, ovum will be done, ovum will be formed only after meiosis 2, right? So meiosis 2 happens just before fertilization, as in when the sperm enters. That's when meiosis 2 is going to happen. That's when you get the ovum and the secondary oocyte. Okay, and always remember, during ovulation, it's not ovum which is being released, it is secondary oocyte which is being released. Fine, fine. So this is the thing that is there. That is what you need to know. And let's go ahead. And the ratio over here, if at all you see, in spermatogenesis, from one spermatogonia you get 4 sperms. In the case of oogenesis, from one oogonia you get only one ovum. Only one ovum. So the ratio is kind of different. Always remember, this small details could be helping you really, really well. Fine, let's go ahead with the next chapter. Guys, give yourself a big tap on your back, because you guys are super awesome. And we are entering the next chapter. Kudos to you. Please solve all the questions. That is my vinti from you guys. Obviously, if I go and talk about all the questions out there, this video will never end. And you will not get time to do self-study. That is why I am explaining the things that could be coming. In 2 marker, more chances for infertility and art-related questions. And in 3 marker, more chances for STIs, that is sexually transmitted infections, and MTP, that is medical termination of pregnancy related question. Okay. So, what do we have? Okay. This questions, I think we have solved it before, but I want you guys to solve. Suggest any 2 arts, or basically art technique, assisted reproductive technology, that can help the couple to have a child, but the problem is with the female partner. The female partner is having problem, okay? Which is the assisted reproductive technique that is going to be suggested in this particular case? Any idea? And also, guys, please make sure you share this video with all of your friends out there. Okay. Any idea what is the answer for this particular question, guys? Zift and gift. Alina says zift and gift. Okay. What about others? Any other answer? Riddhi also says gift and zift. Okay. I'm waiting for others also. Zift and gift. Gia also. Gia is there. Hello, hello, hello. Ma'am, is this video enough for getting out of out? It will definitely help you reach at least up there. Yes. Sanjeevani and everyone. Okay. Zift and gift. Why zift and gift? Gift definitely. Why? Because the female, in this particular case, the female has a rudimentary ovary. Okay, her ovaries are not fine, which is why gametes are not being formed. In this case, what do? You take another donor's gamete and you put it in the female's fallopian tube. You will put it in the fallopian tube, not in the uterus. Why? Because you want fertilization to happen normally there. Okay, that is gift. Second thing, as you said, is zift. Zift basically means probably because of some reason in her body fertilization is not happening. So what are you doing? Basically, with the help of IVF, you do fertilization outside the body. You either transfer the zygote or you transfer the embryo up to 8 blastomere stage. Where do you transfer it? Inside the fallopian tube. Why, ma'am? Why inside the fallopian tube? Because if it is a zygote or the embryo up to 8 blastomere stage, it has to be there only. It will keep on dividing and it will slowly go to the uterus by itself. So zift and gift is the answer for this question. Please write that. Do not come and write IVF and come. Okay. Do not just go write IVF and come. It will be wrong. You have to write zift or gift. If at all you're writing IVF, explain zift and explain IUT also. Okay, got it? Yes. Now, 10th question. A healthy couple came to know that both of them are unable to produce functional gametes. Both of them, male, female, everyone. Okay. Kusum and Kusuma are, okay, wait. Should look for an art technique. Name the art that can be suggested to help them bear a child and explain the procedure involved in it. Listen, over here the problem is with both these people. Now what do you do when they are not able to produce functional gametes? What is a technique that can be used, applied over here when they are not able to produce functional gametes? Any idea? For those of you who said IVF, I hope you will also write gift and basically IUT and come. Okay, do not just write IVF and come. For the previous question. For this question, yes, you have to do IVF. You can do IVF. In IVF, you have to make sure that basically, what are you going to do? You are going to take the gametes from donors. You can take the gametes from donors. That is one technique that is there. Yeah. Fine. And followed by, you always write zift and gift, zift and IUT. This is what you will do after this. Apart from that, is there any other thing that you can do? You can separately mention that, no, don't. Okay. So this is something that you can do. And also, if you want, this is something that can be done commonly. Now, separate things if you'd want for both female and male. For example, female is the one that is having gametic problem. So basically, gift can be performed. In the same female, for fertilization to happen, artificial insemination has to be done from a donor sperm. From a donor sperm. Okay, not from the actual sperm, actual husband out there. From the sperm donor, you can take the sperm and do IUI. That is intrauterine insemination. But in that particular case, this would be enough for you. I think that will be adding up more problem out there. Ma'am, will IVF come in previous question? In the previous question, if you're writing IVF, you have to write zift and IUT also. Okay, zift and IUT as well, if at all you're writing that. And you have to mention it is taken from a donor. If you're doing IVF, you're taking the egg from a donor, you're doing it, and then you're putting it inside the female body. Mention that also. That is what I said. That is this. Artificial insemination. Okay, listen. If you just write artificial insemination, over here, the answer will not be enough. Why? Because it is saying both of the female and the male are having problems. So you have to look at the both direction. So IVF is something where both donors can be giving this sperm as well as the egg. Now, if at all you want to deal with them separately, so you will write it. The female basically over here. Okay. The female over here will have to undergo gift, and the male who is there will be doing artificial insemination from the donor sperms. From the donor sperm. Both this technique has to be done in a couple. Otherwise, you can just do IVF where you take the donor sperm and egg and basically go like that. Cool? Yes? Now, Explain the technique that can help a healthy married woman who is unable to produce viable ova but can provide a suitable environment for fertilization. Again, this is going to be gift. Okay, this is going to be gift. I got to learn a new Malayali word. Yes. Ma'am, important question. These are the important questions, champ. Okay. There are questions, I have not taken PYQs from the previous year. Mostly there are newer questions that are being solving over here. Yes, Shivanand, I will be giving you the PDF after the class ends, right? Yes. Now, over here you are going to do gift only. Why? Because you are not able to produce viable ova. That means gift has to be done. What you have to mention is, in this technique, ova is taken from the healthy donor and transferred into the fallopian tube of a woman who cannot produce viable ova but can provide suitable environment. Where is the fertilization happening? Always understand, it is happening in vivo, not in vitro. In vitro means in the lab, but here it is happening inside the female body. This is something super important. In vivo, in vivo, in vivo. Write this and you'll get your 2 marks out there. There's a 3-mark question that is there. Remember over here, as I said, you can get so many questions related to infertility as well as art in the 2-marker category. There can be reasons asked for infertility. Can you guys tell me the reasons for infertility? Anyone out there? Can you tell me the reasons for infertility? Hi ma'am, good evening. Hello Manishri, good evening. Reasons for infertility. I'm waiting because there's a lag. That is why probably I cannot keep quiet and stand. That's why I'm repeating it again. I hope you're not getting irritated. If you are, I'm sorry. Reasons for infertility. Disorder, psychological disorders, okay. What about other things? Reasons for infertility is important. It can be because of psychological reasons. It can be because of rudimentary ovary, okay. That means, okay, physical reasons are there. Drugs, yes, exactly, Rizwana and Kirtana. And Alayna says physical, congenital, immunological. Good. Physical, as in basically, as I said, the organs are not working well. Congenital, because it can be genetic problems that is there. Okay, immunological, because your own cells are killing your own cells. That is like that. Okay, and yes. Congenital, that is a physical, physiological, and stress, drugs, disorders, everything. Perfect. So please know about all of them. They can be asking you. Do not get it wrong and write it and come. Yes. And also there was one more thing. Because of STIs, sexually transmitted infections also can be a reason. Okay. Physical, congenital, physiological, and disorders which are there, immunological reasons which are there, and STIs which are there. There are all such routines due to which basically what is going to happen, infertility issues can rise up. Okay, Asha, hello dear. Okay, now we go to the next thing. And my dear children, over here I have few more questions out there. You have to go ahead with that. 2 events that are inhibited by intake of oral contraceptives. If oral contraceptives are being taken up, what are the 2 events which are being disrupted? Ovulation as well as implantation. What are emergency contraceptives? Emergency contraceptives, I hope you guys know that. Anything related to hormones. Okay, it can be oral pills. It can be hormonal IUD. Everything that is there, it is basically going to inhibit fertilization or basically implantation. So we call them as emergency contraceptives. They have to be taken within 72 hours, okay, of the coitus. Super important. Ma'am, important question PDF. This is the question PDF, dear. And yes, in the video, okay, I'll be giving you that. Kishan, I'll be giving you that. Soon after this particular thing ends, I'll be sharing with that with you guys. Yes. Perfect. Now, I'm strictly going ahead with the belief that you are going to look at these questions and are going to solve it, okay? Please do that. If you're not doing that, probably you might be missing on things, and I really do not want you guys to do that. Okay, next thing that we have over here is basically IUD is an emergency contraceptive. How is that possible? Okay, listen. Hormonal IUD is an emergency contraceptive. Hormonal. Not every type of IUD, hormonal one only. And apart from that, as I said, oral pills are the ones that are something that acts as emergency contraceptives. Okay. Saheli, better, Sanjeevni, not write Saheli, okay? Better, it will be better if at all you do not write Saheli. Rather, just write oral pills out there, okay? Because Saheli, again, it can help. It is a contraceptive, definitely. But it is once in a week pill, okay? And it has its own, it's a non-steroidal pill, so obviously. Yes. Okay, guys, before can you explain ICSI? I can explain ICSI. Listen to me. So before I go do the entry into the 3 marker, I will just brief you about the IUDs and other contraceptive methods which are there. Can you please listen to me carefully? Okay, everyone, I just need your attention, nothing else. So first method of contraceptive is natural method, and in natural you have 3 of them. Okay, I'm just saying, I'm not writing it. In 3 of them you have, one is periodic abstinence. What do you mean by periodic abstinence? During a certain period, that is, you call this as a fertile period, day 10 to day 17 of your menstrual cycle. 14th the day ovulation happens, but it can happen before, it can happen after also, which is why we say day 10 to day 17 no one is going to have sexual intercourse. Why? Because there are high chances for ovulation to happen. Egg will be there, egg might be there. So that is periodic abstinence. Okay, that is first method. Second method is withdrawal method, where what is happening is that the penis is being removed before insemination is being done, such that the sperms are not being released inside the female body. It is also known as coitus interruptus, because why? You are interrupting the sexual intercourse out there in between. Now the third one is lactational amenorrhea. Lactational amenorrhea means a mother who is lactating, okay, who is breastfeeding. In her body, the level of a hormone, prolactin, is too high. Now this prolactin will not allow ovulation to happen. Automatically, this prolactin which is there, it will not allow the FSH and LH hormones, which is why ovulation is not going to happen, is what we believe. It is there for 6 months, but sometimes this can fail, which is why all the natural methods which are there, we say that they are highly unreliable methods. Okay, there is high risk factor, but there is no side effects. Why? Because they are all natural. There are 3 methods. Okay, done. Now the barrier method, we come into barrier method. We have condoms, we have valves, we have diaphragms. In condom, my dear children, it is the best one to prevent conception. Apart from that, it is also the best one to prevent STI, sexually transmitted infection. Why? Because condom is covering the penis, and also basically no fluid is going to be being shared between 2 human beings out there. That is why we say that it is the best method of contraception as well. Okay, and apart from that, you have valves as well as diaphragm. They are effective contraceptives. They are not going to be helping in prevention of STIs, because valves are now inserted in the cervical region. They are being inserted in, suppose this is the cervix, so they are inserted over here. Still the penis is over here. The sexual fluids, there are chances that it can come. This is the vagina, right? So sexual fluids can come in contact. That is why we say it is not a reliable method. Again, conception will not happen for sure, but STIs can happen. Now, so only thing that we have is condom. Now you have IUD. In IUD, it is super important for you to remember each and everything. Mostly you're going to get an MCQ question based on that. Okay. In IUD, what do you have, Baba? In IUD, you have 3 types. First one is non-medicated. This I will write because I think that, I do not know, I get a feeling that this is important. Non-medicated IUD. And what do we have? We have copper releasing IUD. Okay. And then what do we have? We have one more thing that is known as hormone releasing IUD. Hormone releasing IUD. Okay, in non-medicated IUD, suppose this is the uterus. Over here, something like this is being inserted. What is this? This is known as lipase loop. Lipase loop. Do not mug it up. Remember, it looks like a loop. And why is this being put? Because of one very important reason. Why? You want your body to be active. Listen. What exactly happens is that you are inserting something foreign inside your body. Your immune system becomes super active. Your immunological cells become super active at this particular point of time. Now you imagine any sperm which is entering, those are going to be captured by these immunological cells. So we say basically with the help of lipase loop or non-medicated IUD, they are not releasing anything. They are just making our immune system active such that there will be phagocytosis of sperms. The sperms are going to be eaten up. Phagocytosis of sperms. Now copper releasing IUD, my dear children, again imagine the uterus. Over here you can have copper T, which looks like a T-shaped thing. You can have copper 7, which looks like a 7-shaped thing. You can have multi-load. Okay, multi-load 375, that is all super important. All these things, my dear children, copper T, Cu7, multidrop 375, they are important because they release copper. What does the copper do over here? Copper does one very important thing. It holds the tail of the sperm and says, Macha, you cannot move inside the body. So basically, the motility of the sperm is the one that is being ruptured over here. And apart from motility, there is also one more thing, that is the fertilizing capacity. Okay, the sperms will not be able to do the fertilization properly. So motility and the fertilizing capacity is being ruined up because of copper releasing IUD. Now we have one more thing, hormone releasing IUD, and the example for that is progestasert and levonorgestrel. Levonorgestrel, LNG20 is what you write. Okay, and also progestasert, you know the name and you get an understanding that they are synthetic form of hormones. They are not your natural hormones, they are synthetic form of hormones. Listen, in our body you have seen the function of progesterone, estrogen. They are very much important in ovulation to happen, for implantation to happen, for everything to happen. But now because you're giving hormones from outside, what is going to happen? There is an imbalance that is going to be caused. And everywhere where there is hormonal thing mentioned, for example, in oral pills there is hormones. In implants, okay, implants are things that you put under the skin, subcutaneous things. They release hormones. All these things, my dear children, which are hormonal, they prevent ovulation. They prevent implantation. How implantation is prevented? Basically, the endometrium is very thin such that the embryo will not come and attach over here. So it is not suitable for implantation. Apart from that, the cervix which is there will be a mucus plug being formed over here such that the sperm cannot transfer. So that is being seen in this particular case. So there is a mucus plug being formed in the cervix. That is not found in CUT. That is basically found in the case of hormone releasing IUDs or basically oral pills. That is cervix hostile to sperm. Exactly, Alana. So I hope you have got it. These are three things. Know their examples, super important. Apart from that, you have oral pills. I told you they have to be taken every single day except the time for menstruation. Apart from 1 to 7 days, rest of the days you have to take it. And apart from it, if at all you do not want to take oral pills, there is Saheli. It's once in a week pill. It is non-steroidal. It does not release any hormones. It works ulta. Basically, the estrogen which is there, the receptors of the estrogen which are there, they mess around with the receptors. They are not hormone releasing pills out there. So Saheli was made by CDRI, Central Drug Research Institute of India, and basically that is a non-steroidal drug. It is really useful, and yes. And apart from that, you have implants, and finally you have surgical method, vasectomy and tubectomy. Vasectomy means basically you're cutting the vas deferens. Tubectomy means you're cutting the fallopian tube out there. Anyway, the sperm and the other gamete is not allowed to transfer. Okay, this is one of the high reliable method out there, surgical method, but it has poor reversibility. You cannot reverse it because anyway you have made a cut and stuff like that. You get it? So this is something that you have to remember. And in the case of vasectomy, even though you cut the vas deferens, do not think that the spermatogenesis is not happening. Spermatogenesis happens. The sperms reach up the vas deferens also, but they are not able to move ahead. Why? Because you have made a cut over here. So they get degenerated there. Similarly, in the case of tubectomy, there is formation of ova there, but menstruation is still going to happen as well. Okay, because hormones are being released, everything is very normal. But only thing is that the gametes are there which are not going to get transferred. That's the only thing. And always remember, all the contraceptives have side effects. It can also lead to breast cancer. This is some important information. Okay, this will be some important information. It can lead to breast cancer as well. Oh, okay. Hi Jasmine ma'am. Hello Daivik. And will you be taking live session on Monday morning? I am thinking of doing a recorded session rather than a live session, dear. That is what I believe I will be doing. Will that work? Okay. Ma'am, what is the difference between in vivo and in vitro? In vivo means inside the body fertilization is happening normally. Okay, the sperms are being released inside. It is happening inside the body. In vitro means in the lab it is happening. That is about, yeah. Ma'am, need ecology explanation. We'll be doing that, dear. And after tubectomy, periods occur? Yes, it does. Because ovum is being released anyhow. All the hormones which are there, they are also there. That is why exactly there will be periods. Okay? Yes. Perfect. So I hope that is super clear, and now we'll go ahead. Contraceptives, all the contraceptives, majorly the pills. So it is according to your NCERT, which says that the contraceptive methods which are there, they have side effects. It can lead to mood swings, it can lead to cancer, it can lead to having nausea, cramps and all of it. So please remember that. Sometimes we think only the good part of the contraceptives, but there are also some bad parts which you have to remember, because it might come in MCQ and you might mess around. Yeah? Hello Chandrakant. Hello, hello, hello. Okay. So children, let's go ahead with medical termination of pregnancy then. Yes, we have spent a lot of time over here. Let's go ahead. What is MTP, and mention any 2 medical grounds on which pregnancy can be terminated. By the way, one more question. That is, if at all a person undergoes vasectomy, do you think semen is going to be released? The answer is yes, okay, because I do not have the patience to wait for the answer as of now, because it is lagging. So yes, it is going to be released. Semen is going to be there, but without sperms. Sperms are not going to be there in the semen. Yes, okay. What is medical termination of pregnancy? It is basically voluntary, okay, or intentional termination of pregnancy. You want to terminate, that is why you are terminating by medical or surgical methods out there. And mention it in 2 medical grounds on which pregnancy can be terminated. So on 2 medical grounds, what are 2 medical grounds, Baba? One thing, that is basically over here, when it is risk to mother's life, okay, or probably it is risk to the child's life. The child might have some congenital disorders, that is genetic disorders and things like that. In that case, it is approved, okay. And over here, please remember these 2 informations out there. First thing, when was MTP legalized? In the year 1971. When was MTP Amendment Act? So MTP was legalized in 1971, but people, you know what they did? We human beings are too cruel. When MTP was legalized, people started doing amniocentesis, got to understand the sex of the child, and used to kill the girl childs out there. The female feticide rate was slowly increasing way too much. And not just that, there were so many people who were not using contraceptives and who were underage. They did sexual intercourse and probably might be bearing children. And that's when they went to doctors who were not enough qualified, and that led to so many deaths, okay? Which is why Medical Termination of Pregnancy Amendment Act was being put down. This is also very, very important. So when was that being put down? You do +1 over here, it becomes 20. You do ulta of 71, it becomes 17. 2017 Medical Termination of Pregnancy Act, okay, Amendment Act was being put, according to which if at all a person is doing medical MTP up to 12 weeks, that means you're talking about 3 months, then it is considered to be safe, but you need a signature and you need an approval from at least 1 registered medical practitioner. Now if at all you are going ahead and doing MTP in 24 weeks, then you need approval from what? 2 medical practitioners, and it is not considered to be safe. So always remember, MTP can be performed up to 24 weeks, but it is not safe. It has to be performed within 12 weeks itself. Got it? Okay, sweetie, thank you so much, dear. Ma'am, what is the safe period for MTP? It is going to be 12 weeks. If at all it is done before 12 weeks, it is going to be safe. Otherwise, it is not considered to be super safe. I hope that is clear. Yes. 12 weeks means 3 months. 12 weeks means basically 3 months. Up to 6 months, it can be performed. Always have an idea of that, but it is not considered to be safe. Cool. I hope that's clear. Afzal, you will get the PDF post the live is done, okay? And my dear children, I sincerely request all of you to share this video link to all of your friends. I literally am not really happy seeing the number of children who are watching this video. Why? Because you have your examination later on, and probably this video, if you are missing right now, I might not be able to take up your doubts later on. And yes, it's a building time that you have as of now today. Okay, utilize it, share it with your friends, ask them to join. We have just covered 3 chapters, we have so many left. Yes. Okay, perfect. So I think this is clear, and now we go to the next thing. There's a question, 3-marker question. The picture shows the contraceptive device used by women. Identify the contraceptive device shown in the picture. Mention how it prevents fertilization and conception. This is a question that came. So I was trying to hide the answer, but you know what? I saw the answer is clearly visible. This is copper T. Always see it looks like a T, right? It looks like a T. This is copper T. How does it prevent fertilization? I told you it suppresses the motility of the sperms. Yeah, motility of the sperms and also the fertilizing capacity of the sperm. Okay, I'm sure most of them are resting because it's maths exam today. Yes, I get it. Yes. Okay, so yes. Hello, Muzammil Patel. Okay, so this question is also sorted. We are going to the next chapter. We had a question on venereal, yes, this question we missed. So, define venereal disease, mention the modes of transmission of HIV and hepatitis B, and what are the symptoms of venereal disease. So, what is venereal disease? First of all, venereal means sexually transmitted infections or reproductive tract infections out there, okay? And how are they transmitted? Usually through sexual contact, okay? Usually through sexual contact. Now, listen to me one thing very carefully. So, there are some of them, okay? Jennifer, hello. There are some of them which are transmitted through other modes also. Most of them, obviously every one of them or everything of them are going to be transferred through sexual contact, but some of them can be transmitted through other modes also. So that is basically HIV and hepatitis B virus. You have been clearly asked, mention the modes of transmission of HIV and hepatitis B virus. So they can be transmitted through what? Unprotected sexual intercourse, definitely, because it's an STI. Apart from that, transfusion of infected blood or blood products. Okay, so blood can also be a medium for transfer. Apart from that, sharing contaminated needles and syringes. You might have seen drug addicts, okay, sharing the needles among themselves from infected mothers to the child. That is also how it transfers. I'm telling you again, STI is usually transmitted through the genital fluids, but some of them gets transmitted to the other body fluids such as blood also. Example for that is HIV and hepatitis B. Now, I have a question for you guys and you have to give me the answer, okay? Which are the three diseases which cannot be curable? Which are the three STIs which cannot be curable? I have mentioned so many times before. Man, if we done the question bank MCQ, can we get 15 on 15? No. There can be any question that can be coming from somewhere else also, dear. We cannot guarantee that it can give 15 on 15, but yes, if at all you religiously practice all the questions, basically you understand the concepts, probably in that case you might be able to do more questions like that. So yes, in that case I'll say there are chances, but you need to understand the concepts really well. Okay? Ma'am, are you from Karnataka? No dear, I'm not from Karnataka, and I got my answer once. First one, Sanjeevani says hepatitis B and genital herpes. Perfect. Yes. Yes, one more, come on. Hepatitis B, genital herpes, these two are incurable. Apart from that, there is also one more thing. And what is that one more thing that we have? Hepatitis B, genital herpes, Deeksha, Jia. One more, come on guys, I gave you a trick to remember it. It's simple. HIV. Acha, Sanjeevani already gave the answer, HIV in before. Okay, so listen to me. There are 3 H which are incurable. One is HIV, one is genital herpes, okay, and the third one is hepatitis B. 3 H, they are hepatitis B. They are what? Incurable STIs are there. If you have it throughout your life. Apart from that, you have other STIs, for example, gonorrhea, syphilis, and all those things. They are curable. Okay, they are curable. Now, the symptoms which are there, you need to remember that also. Excess ragu, syphilis is not something that is not curable. Okay, dear? It's curable. It's curable. These 3 are the ones which are non-curable. So early symptoms is itching. There will be discharge. There will be pain. There will be swelling in the genital region. Now females may be asymptomatic. Okay, they might have less symptoms, which is why they probably might not know they have it. Okay. Now over here, you have some very important things that can be happening in venereal diseases. One is pelvic inflammatory disease, as in over here in the pelvic region, my dear children, there will be inflammation in the organs out there. That is what you say, pelvic inflammatory disease. Second one, abortions can also happen. Third one, stillbirth. Stillbirth means baby is being born, but it is a dead baby which is being born over there. The next one, ectopic pregnancy. Ectopic pregnancy means instead of baby being born in the uterus, it is being formed in the fallopian tube. Now that can lead to the death of the mother as well as the child. That is known as ectopic pregnancy. Now what do we have? We have infertility. Infertility can also take place. Yeah, we saw it's a reason. And reproductive tract cancer can also happen. So these are a few other things that you might try to write. Yes, what is going to be different? Genital pain is also a symptom. Yes, dear, it's a pain. So pain, itching, discharge, swelling in genital region, all of them comes up symptoms. Make sure you know this small information are there. And how do you prevent STIs? First thing, avoid multiple partners. Second thing, use condoms. Third thing, talk to the counselor out there. That is how you avoid this. Okay? Lakshmi, I am in Karnataka, but I'm not from Karnataka. Okay, so I'm in Bangalore. I'm taking the class from Bangalore itself. So I am first, second PUC. Do not be worried. Okay, so I'm not teaching for any other state boards out there. Yes, yes. Okay. Till what time the live will be there, Jennifer? At 11 p.m. sharp, it will be ending. Okay. Pedigree, it will be coming, Gia. One by one, we'll go. Okay. So come on, guys. So this has been done. Yes. And now the last thing that I want to tell about the chapters, that is reproductive health, is your duty is to understand what are the reasons of population explosions. Okay, there are multiple reasons given over here: infant mortality rate, maternal mortality rate, and things like that. I'll explain you one very important thing, that is amniocentesis. Suppose this is the baby that is being formed over here. Around the baby, my dear children, it is filled. It is basically inside a sac. What do you call the sac as? Amniotic sac. Amniotic sac. Inside that, what fluid do you have? Amniotic fluid. Amniotic fluid. Basically, in the fluid, there will be certain cells that are shed from the baby. Okay, I take this cell, I examine this cell, I get to know the genetics. With the help of this genetics, I am basically able to understand what are the genetical disorders that this baby probably can have. Okay, genetical disorders only. Ma'am, can you find jaundice with the help of this? No. Is jaundice a genetic disorder? No, Baba, it's not at all a genetic disorder. It's not at all a genetic disorder. Okay, so please understand, only genetic disorders, for example, sickle cell anemia, Down syndrome, things like that can be found out. And since you're anyway dealing with genetic material, you can also find the sex of the child. But for this reason, that people started using amniocentesis for performing female feticide, that is exactly why there was a ban on amniocentesis in our country. Okay, why there was a ban? Because people started using for sex determination. By what was the actual use of it? Actual use of it was to find genetic disorders. Along with that, obviously, yes, you can do sex determination, but you should not be doing female feticide, right? Yes. So that is the only thing. So can you do sex determination with the help of this? Obviously, yes, you can. Should you do what do you call female feticides? Obviously, no. Okay. Ma'am, I'll be disappearing till 7, ma'am, and now you will be doing my favorite chapters. Oh my God. Okay. I'm so sorry, Alina. If at all you can, just watch it like up there. And yes. So it's Rosa. Okay, I'm getting it. I'm so sorry, dear. And yes, please go ahead. Please go ahead and you can watch it. Okay, the live anyway will be recorded. Perfect. So let's go ahead now with the next chapter that is there, Principles of Inheritance and Variation. I hope, my dear children, everything is sorted. Okay. So we started taking the live at 4 o'clock. Can we take a break now for 10 minutes? For 10 minutes, we shall take a break. If it works, everyone. If it works, give me a thumbs up and then we'll go ahead with this chapter. Listen, I had explained art. Can you just go back and... Yeah, but that won't take more than 10 minutes, right? Yeah, okay. Yes, perfect. So we'll take a 10-minute break because my back is paining. And over here, 5:55 is the time now. So basically, we'll be back by 6:05. Okay, I'll just write it like this. Kit Kat break. Be right back by 6:05. Okay. hello, 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 hello. Hello, Selvi, Anand, Deekshit, Sanjeevani. Correct at right time. Yes, I should definitely take some vitamin D, right? Yeah, even I think so. Okay, guys, so the Kit Kat break is over. How many of you actually had Kit Kat during this break? I had one apple, and I also hope everyone who are keeping rosa, you are having something. I think it's the time to break the fast, right? Yes. Yeah, I ate Kit Kat and back now. Perfect. So, ma'am, these questions for Karnataka board also come in. Yes, dear. Hello, Varsha. Hello, Pushpa. Hello, Selvi Anand. Thank you. You're also cute. Nal Aron. Hello, dear. Okay, so we are back with some more tasks out there. We have to complete the chapters, right? So, principles of inheritance and variation, my dear children. From here, I see linkage in 2-marker section. Linkage and recombination is somewhere where you can get more questions from. Pedigree, icon related questions have more chances, but that does not mean that other questions do not have any chances. So, make sure you go through all of them properly. I'll be explaining few of them over here as well. So, let's go ahead with one of the MCQs first. In the F2 generation of a Mendelian dihybrid cross, the number of phenotypes and genotypes are. You're talking about a dihybrid cross, okay? Mendel's dihybrid cross over here, and you have to talk about what is the number of phenotypes and genotypes that you observe. Hello, Kuvalesh. Okay, ma'am, I have not taken anything. Please have something. I hope you should have some energy. Okay, Kannadigana, hello, dear. Okay, and children, please share this video link for all of your friends, and let's rock biology. Okay, how many phenotypes and genotypes are there? A, B, C, D. What do you think is the answer for this? Ambika Swami, yes. Sanjeevani says 5D. 5D is what? Phenotypes 4 and genotypes 9. What about others? Do you have any other answers? Only one person have the answer. What about the others? Hello, Kandigana. Hello. There is a lag. That's why probably it will be coming slowly up there. Sampath Kumar. Okay, I'll try to remember that name. Children, the answer for this question, Gia says the answer is D. Okay, so 2 votes for D. And PSK also says it is D. Yes, the answer for this question is D. Remember the ratio 9:3:3:1 in a dihybrid cross. You're talking about a dihybrid cross, right? Yes. 9 of them are going to be round and yellow. They are parental character. You know, you crossed round and yellow parents with what? Wrinkled and green parents. So 9 of them, majority of them is going to be round and yellow. One of them over here is going to be wrinkled and green. Is going to be wrinkled and green. What about the 3 and 3 over here? They are the non-parental character that you never saw until now. So round was with yellow until now, right? But this time round is going to go with yeah, round is going to go with, I thought it went green. Okay, and here wrinkled was with green until now. Here it will be wrinkled with yellow. So how many phenotypes, physical appearances can you see? 4 phenotypes are there. So 4 phenotypes done. Genotypes, how many of them are there? So if at all you have seen the cross, you see 16 combinations are possible. 9 plus 3 plus 3 plus 1 is equals to what? 16 combinations are possible. Now does that mean 16 genotypes as well? Wrong. So many genotypes are being repeated, Baba. So you cannot count them again and again. Unique genotypes, if at all you are asking, there are only 9 of them. And how do I remember it? The first number over here is 9, and that is how I remember it. So genotypes is 9, okay, and the phenotypes which are there, they are 4 in number. So your answer for this question is going to be D. So everyone who got it correct. Okay, Pushpa, why ma'am? Why as in basically, listen, I told you 16 genotypes are there, but so many of them are repeated again and again. No, capital R, small r, capital Y, small y, they are repeated so many times. I will not count it every time again and again. I'll only count it once. Got it? Sorted? So your answer for this question is D. Everyone who got an understanding of it, just give me a thumbs up. We'll go ahead with the next one. That cry. Yeah. Yes, that happens with me. And by the way, everyone who is watching the video, I hope you are also clicking on the like button. Why do I see that 508 votes are there? That you have already done the like and someone is doing right now also. But the likes are only up to 160. Come on, guys. When you like the video, what is going to happen? It reaches to many other children as well. Okay, clear, perfect. Let's go ahead with the next question out there. In Drosophila, genes for yellow body and white eye show lesser recombination, and white eye and miniature wing show very much high recombination in a cross. Why? Oh my God, what is this question? I do not even understand this question. Listen, this question is super simple. They are asking you, you need to have an understanding over here. Okay, the understanding goes like this. The gene for white eye, okay, or basically, I'll just write over here. This is yellow body gene and this is white eye gene. You know, they are very close to each other. They are very close to each other. Suppose these two parts get swapped. What is going to happen? These two genes are going to go that side also. Whatever you do, these two genes are going to go together only. So can I call them tightly linked genes? Yes, I'll say that they are tightly linked genes. And what's the problem or what's the good thing with tightly linked genes? They will show less recombination. That means recombination means newer combinations. Do you think they will make newer combinations? No, Baba, they're always moving together. What new combination do you expect out there? So lesser recombination. Tightly linked genes means lesser recombination. You and your best friend, you're so close. Do you think any other friend can come over here in your friendship? Obviously, no, you will not allow that, right? Usually, that's the case. So, they are tightly linked genes, no new recombinations. Now, over here, when we talk about one more character, that is miniature wing. Miniature wing. This miniature wing gene is very far from these genes out there. So suppose I'm crisscrossing this part, this will be going there, this will be going here. Obviously, you will get a new part altogether. You will not have miniature wing with you always, right? So what did you get to understand from this? That is basically when you talk about these two genes, they are loosely linked. They are going to be loosely linked. And when they are loosely linked, there are high chances for what? New combinations to form. There are high chances for newer combinations to form. So I say that they have more recombination rate. They have more recombination rate. Now, anyone who have read your NCERT really well, can you tell me the percentage of recombination that is exhibited by them and that is exhibited by them? So 1% percentage of recombination, only 1% percentage of recombination is exhibited by tightly linked genes. Whereas when you talk about the loosely linked genes, that is miniature wing and white eye, or miniature wing or yellow body, they have higher percentage of recombination. That is something else. Can you tell me that what is the exact number out there? Ma'am, can you send the PDF? PDF I will be sending you post this particular live is being done. Okay, post this live is done, I will be giving you the PDF. Ma'am, can we see the same pattern of repeating of questions in biology paper as well from model question paper? So it's not like exact questions will be repeated, the concepts might be. So I'm trying to make you help with the concepts which are there. Okay? Okay, 37.2, okay, Deeksha said 1.3, 1.3, 1.3, Varsha said 1.3 and Sanjini said 37.2. Okay, so listen to me, 1.3 is for these guys, 1.3% recombination because they are tightly linked. 37.2% recombination which is there, it is shown by this guys. Remember, 50% is the highest recombination and in that you have this and that is a big number. Ma'am, Drosophila advantage, they can be grown easily on a simple media. They basically their progeny, they can produce multiple progenies all at once. Their life cycle is only up to 2 weeks and their characters can be easily distinguished, the male and the female character under a normal microscope. All of these are the characters out there. Okay, ma'am, I also said 1.3. Exactly, yes. So you guys gave me the answer for 1.3 and 37.2, which is what I meant. Okay, so everyone, awesome. Okay, so this is your answer. How do you frame the answer? I hope you have got to understand that. How do you frame the answer? Yellow body and white eye genes. Remember, yellow and white. Okay, yellow and white. Lesser recombination. These genes are closely located on the same chromosome. When genes are close, crossing over occurs rarely between them. Therefore, low recombination frequency is being observed. This condition is called strong linkage or tightly linked genes. Okay. On the other hand, white eye and miniature wing. Higher recombination is there. These genes are far apart from on the chromosome. Crossing over more frequently happens over here, and there is high recombination frequency that is clearly being seen. Always understand Y and W in the alphabetic chain, they are really close. So they are here close also. Here also they are very close. You can remember like that. Okay? Fine? Yes. If this question is super clear, can you just give me a quick thumbs up? Everyone, if this question is super clear, just give me a quick thumbs up. We will move on to the next question. Ma'am, how to find the PDF you're asking? Dear, after the session ends, I go ahead into the WhatsApp channel, I type the message, and there I'll be giving you the PDF. Okay? You have to join the WhatsApp channel though, that's all. Same here. I'm not saying exactly, listen, prepare for the worst, always. I always tell that, prepare for the worst. Yes, repetition can be happening, repetition may not be happening. Why not know extra things about things such that you will be on the safer zone, yeah? Thank you guys for giving me a thumbs up. We'll go ahead. Listen, pedigree analysis related question can come. So all this, all the things that you see over here. Okay, when you have this, that means male. When you have this, that means female. When you have this, that means affected male. When you have this, affected female. When you have this and this, that means male and female is mating. When you have this and this, that means two lines, they are very close. This is consanguineous mating, that is mating between relatives. Okay, when you have, okay. So when you have something like this, always make the male over here first, then the female. And if you have something like this, that means both the parents are mating and you have a female daughter out there. All these things should be there. Understood? When you have something like this being written, that means the sex is not being specified, whether male or female. If at all you are coloring it up, that means the person has having the disease. If at all you are writing 5, that means there are 5 children whose sex is not being determined. Okay, there are small things, so make sure you go through that. That's why we came here, preparing for worst. Exactly. We are preparing for worst. Yes? Yes. Ma'am, WhatsApp channel link is there in the description section of this particular live video. Okay? Fine. Now, there is a, pedigree that is mentioned in your NCERT. I hope you have seen this pedigree. Okay, this is the only disease that is mentioned as autosomal dominant, a dominant disease. Everything else in your NCERT is recessive, only this one is dominant. Okay, only this one is dominant, and that is what? Myotonic dystrophy. In this case, when I say it's a dominant disorder, how do you are you easily able to find out a dominant disorder? Very simple. In every single generation, one person is going to be affected. So here the female is affected, here the son is affected, here the female is affected, here again the daughter is being affected. So every single generation, don't you see one person is affected? Yes, that means it is what? A dominant disorder. It is a dominant disorder. Now over here you see males and females are somehow equally being affected. Here actually the females are the ones that are mostly being affected. So we say it's an autosomal disorder. Okay, it's an autosomal disorder. The sex-linked disorder and pedigree chart has not been given to you in your NCERT, but please understand this. Okay, if at all males are being affected more, then it might be sex-linked also. Now on the same area, if at all you come over here, this is for sickle cell anemia. Sickle cell anemia is an autosomal recessive disorder, right? So you will see skipping of generation. Here there is no person who is being affected, but the next generation there is a person. There are 2 people who are being affected. Why? Because she might be a carrier, my dear children, or he might be a carrier, or they might be a carrier. We never know. Okay, they might be carriers, and carriers do not show the disease, which is why they are not colored. So you see skipping of generation. Here also the son has, but the parents do not have it. Why? Because again they can be carriers. And my dear children, here out of 3 of them, 2 of them are males, but do not go ahead with sex-linked disorders and things like that. It is just an autosomal disorder out there. It is just an autosomal disorder out there. Okay? Okay. Fine. Now listen to me. I have a question for you guys, and I think just a second. Yeah. Since we are doing pedigree, I will go ahead and solve a 5-marker question if at all you guys are okay. Okay, ma'am, your eyes are beautiful. Thank you, Macha. Okay, listen to me. A cross between a normal couple resulted in a son who was hemophilic and a normal daughter also. So here, whenever a question like this comes, you need to read the full question first. Okay, you did a cross between what? Normal couple, and the son was hemophilic and the mother and the daughter was normal. Okay, the daughter was married to a normal man. To their surprise, the grandson was also hemophilic. Okay, represent the cross in the form of pedigree chart. Give the genotypes of the daughter and her husband. Okay, write the conclusion you draw of inheritance pattern of this particular disease. Remember, it's a 5-marker question that came in question bank. It's a HOTS question. Everyone needs to know this. Okay, are you excited about this question where I am? I am really excited. Okay, you should know these type of questions. It works. Okay, so let's solve it. First of all, we need to make the parents. How do we make them? We make like this, right? Male and then female. Okay, we are saying that they are normal couple. That means we should not be shading them up. Now, basically a son and a daughter is being formed. So again, we make the son and we make the daughter. The son would be like this and the daughter would be like this. Okay, now what? Here you are saying that the daughter was married. Okay, daughter is normal. So daughter is normal, but the son is hemophilic. Hemophilic, that means he has the disease. I will shade it up. I will shade it up, right? And over here the daughter is normal. Now what? The daughter was married to a normal man. A normal man, so you do what? This thing, okay? And what do you get? To their surprise, the grandson was hemophilic. That means their grandson, not their grandson. Their grandson are hemophilic. So what do you, their grandson are hemophilic, as in basically you make a son over here and you color it up. And you color him up. Now, the question is not done over here. The question is not done over here. We need to do the genotype also. We need to understand the genotypes of daughter and her husband. This and this. Okay. Before that, we need to come over here and discuss what is happening up here. Listen to me carefully. Here, if I say hemophilia is there, and when I say that a son is being affected, first of all, have this in your head. Son is being affected, right? So son, when son is being affected, he should be having an X chromosome and a Y chromosome. Y chromosome, he'll get it from his father. Okay. X chromosome, he'll get it from his mother. And this X chromosome has to be affected, then only the person will be having hemophilia. So that means mother will have one chromosome which is being affected, but mother is not having the disease. That means she also have one another chromosome that is not being affected, right? So she is what? XXH. And what about the father over here? The father over here, my dear children, the father over here can be a normal guy. He's a normal guy. He's a normal guy. Sir, there is nothing like carrier male over here. So if he's normal, he's just XY. There is no other option that you have over here. Done. Done. Now, you know the, what do you call, genotype of this guy. Genotype of this guy is what? Since he's hemophilic, he will be having something like this. Now, we have to find the daughter's thing. So daughter's thing, how do you find out? Very simple. The same thing you have over here. You just cross it up. Listen to me. XY and XSH. Then this and this comes together, you get XX. When this and this comes together, you get XHH. Which do you think will be the female? The female again will be this, because she is the one who would carry the disease and give it to the son. You get it? So she will again be a carrier. That means you have to choose this. You have to choose this, because you are saying the husband is a normal guy. Your husband is a normal guy. You're saying that, right? So husband anyway will be XY. So if at all the child is getting a gene, obviously it will be from the female parent out there. You get it? Now you make the cross. Again, over here the person will be having an affected X chromosome and a normal Y chromosome. This is how you have to make it. Ma'am, please, once again, guys, everyone, once again, listen to me carefully. Cross between normal couple is there. Normal couple, okay. What do you do? Son is hemophilic, you color it up. And normal daughter, you make it. First, do not go and write the genotype. First, you make it. And then you write the genotype, okay? And then you are saying the normal daughter is married to a normal man. But their grandson was hemophilic, as in their grandson was hemophilic. So daughter's son is basically hemophilic. So you color it up. Now, basically, what do? You try to find out the genotype. And always, when you try to find out the genotype, start with the people who are affected. Then go back. Do not go from them to here. Go back like this, okay? If a man has to be affected, one X chromosome should be affected, right? Yes. So I have made one X chromosome that is affected and one Y chromosome should be normal. When I say a normal man in hemophilia, that means he should be just XY. The female is the only one that can be a carrier. So you make H. Why do not make HH both the places? Because in that case, she should also be affected. But that is not the case. She is normal. So that is why we are not coloring up. Got it? Got it up? Okay, same thing happens over here also. We know that this guy is hemophilic and the parents are not affected. That means it is from the mother, and basically you write it up like this. Okay, done. Ma'am, how to find if it's X-linked recessive? If it is X-linked recessive disorder, it will again skip the generation, and majorly the males will be the one that is mostly affected, and you will not see transmission from male to male. You will not see transmission from male to male, as in if the father is being affected, only the father is being affected. It will not transfer to the son out there. Usually they do not make it so complicated. Do not worry. Usually they will not make it too complicated. Yes. Guys, done, right? Let's go ahead then now. I hope this question is done. Okay, write the conclusion you draw of the inheritance pattern of the disease. What is the inheritance pattern? You know hemophilia is what? Sex-linked recessive disorder. So you basically write about that hemophilia shows X-linked recessive inheritance. The disease is transmitted through the carrier females. It occurs mostly in males, while the females are usually just the carriers. And trait can skip a generation and reappear in the next. Why? Because it is what? It is a recessive disorder. Jia, thank you. You're most welcome, dear. Mallikarjun also, okay, you're most welcome. Perfect. Okay. There is a thing that, what is there? Usually, hemophilic females are not being formed. Do you know the reason for that? Hemophilic females are not being formed. This is a very important question. Any idea why? Why hemophilic females are not being formed? Why only hemophilic males are being formed? Why not females? If at all you have any idea about it, come on, tell me first. For a person, for a female to be hemophilic, she should be having both the X chromosome be affected, right? Right. Now listen to me. If this guy is having his X chromosome affected, he will be having the disease. Here she can be just a carrier. Okay, she can just be a carrier, but here he will have the disease. You have to understand that this is not color blindness. People can live with color blindness, but here people cannot live with hemophilia. They might die. So that is the reason the male might not be viable until might not be there alive until the reproductive age, because they might die because of hemophilia. That is why there are rare chances for a female to be hemophilic. If at all the females are there, sorry, if at all the males survive, then yes, there can be chances. Okay, because she should have both affected X chromosome, and that is something very rare. Okay, in color blindness and all, that is possible. Both the X chromosomes can be, because the person can live with it, but here the males cannot. Okay, that is a reason. You also always need to know that. And yeah, did you understand what I said? It is a bit complicated. If you understood it, just give me a thumbs up. If not, then please let me know. I'll explain it to you again. I do not mind it. Okay. Clear. Because X-linked recessive needs both X chromosome, and also because the fathers who are affected will not be alive until that particular reproducible age. That is the reason. If they were alive, then obviously would have worked. Okay. Cool then. Let's go ahead with the questions that we were solving. Okay. So I told you from here you get linkage and recombination, pedigree, icon related questions. That is usually for 2 marker, but I just explained a question that came into my mind because I saw that. Okay, anyways. Now you have one more question over here in 2 marker section. Try solving this. This is a hot question. A pea plant with purple flowers was crossed with a plant having white flowers. Purple flowers and white flowers, they were crossed. Okay. The progeny produced. Kannadigana, is there anything that I can help you with there? Okay. Anyways, the progeny produced only purple flowers. When you purple and white, when you crossed, you got only purple flowers. On selfing, these plants produce 482 plants with purple flowers and 162 plants with white flowers. Explain the mechanism that accounts for these results. So listen, when you are saying that initially you had purple flowers and basically you crossed with the ones that have white flowers, you got only the purple flowers. Okay, and then you are saying basically you crossed again, and then what you saw? You saw 482 plants being purple and you saw 162 plants being white in color. What does it show? Doesn't it show 3:1 ratio? Doesn't it show 3:1 ratio? Yes. Okay, it is going to be yes. And Jia, ma'am, 1-day strategy I have given to you, dear. Please go through it. Explain again, ma'am. This question I can explain to you. Listen to me carefully, okay? Everyone. Purple flowers and white flowers were being crossed. All the plants which were obtained, they were purple in color. This shows what? Law of dominance. You remember tall plants, white plants, when they were crossed, all the first generation were tall, right? You get 1 ratio. Now, when the purple flowers, now when you need to do second generation, okay, when the purple flowers were crossed with the purple flowers itself, what do you get? In the next generation, you found 482 plants to be purple. And you also saw 162 plants which were white in color. You did not see that in the first generation. Why? Because it was a recessive trait. You saw in the next generation because it follows 3:1 ratio. Why is this happening? Because of law of segregation and things like that. right? So you just have to use your brain regarding your tall and one gene inheritance. So according to Mendel's law of dominance, the purple allele expresses itself in the presence of the white allele. Hence the cross between this and this produce all purple color flowers. Okay, so basically when I cross PP with small p, all of them, whatever you do, cross will be like this only. This one is dominant, which is why all of them were purple. It does not mean the small allele was not there. It was there, but it was recessive. Then next thing that you explain is, according to Mendel's law of segregation, the two alleles of a gene separate during gamete formation. So these two alleles which are there, capital P, small p, which is there, when the gametes are being formed, they are going to separate. Okay, one is going to go to one is going to have capital P, another one will have small p. Right? And now these combinations are going to fuse up together, giving rise back to what? The white color flowers as well. So you get purple and the white flowers back. So you say that, and then that's all. And you have got the answer. Okay. Uday, why to fear when Jasmine ma'am is here? Okay, it's like close to dear. It's not exactly 4:82. That means 3:1. Is it close to 3:1 ratio? You can think like that. Uday Buka, I'll tell you one thing. What's the use of just fearing about biology? Just thinking and being feared about biology. Do you think it will be of any good to you? So it's okay to be, be worried about a subject. If it's okay to have the feeling of being feared and things like that, but that should not overtake your, ability, your actual ability. Always understand that. You have time, make the best use of it, you'll get good score. Okay? Ma'am, it should be 486, not 482. Yeah, dear, I told you, it's somewhat 3:1 ratio, that suffices, okay? It's somewhat 3:1 ratio, so it suffices. You do not need to be exactly like that. I get your point. Okay, all the maths champions out there, I got scared. Okay, anyways, I hope this particular question is clear. I hope you know how to frame the answers. First, you explain about this law, then you explain about this law, make a cross, and then show over here. I have not shown that. Okay, done? Yes. If it's clear, everyone, can you give me a thumbs up? Can I move ahead? If it's clear, give me a thumbs up. We'll go ahead. Ouch. Motivation, ma'am. I have motivated you. Why do you think your teacher is standing up over here and taking live if she thinks that if every one of you were just being scared, I would not have taken live. I am taking live because I believe that my children can come out of that zone and will be trying for themselves, right? And you are one of them who are there in the live. So yes. Okay. Next thing, 5-marker questions, schematic representation and genetic disorder related questions have more chances to come. And listen to me carefully. Actually, you can get 1 5-mark very easily because schematic representation of 1 gene inheritance, 2 gene inheritance or incomplete dominance has high chance to come. You have to practice from your end. I don't think I'll be showing it to you over here. Do you need monohybrid, dihybrid or incomplete dominance? If at all you need, please let me know. If at all anyone needs an explanation of 1 gene, 2 gene or incomplete dominance inheritance pattern, you let me know. Otherwise, I'll go ahead with the other type of questions which can be asked from this chapter. Because I think we have solved a lot of them previously, right? So I thought, so this and important. You will get 1 5-marker question from here for sure, is what I believe, okay? Easy, ma'am. So I will not go ahead because I know it wastes a lot of your time, right? So this is a homework for you guys. Please do it. It's a 5-marker fixed question. Okay, 5-marker fixed question. Please do that. Perfect. Nandini, no. Everyone is saying no. Okay, fine. Okay. Or you can get questions like this on dihybrid cross and things like that. Okay, on dihybrid cross also. So observe the representation of a dihybrid cross and answer the question that follows. So this is a dihybrid cross that is given to you. Capital R, capital Y is fused with small r, small y, small y. Like a normal thing, right? Now, write the phenotypic ratio. What do you think is the phenotypic ratio? Come on, guys. Answer A. Answer A. What is the phenotypic ratio of dihybrid cross? What is the phenotypic ratio of dihybrid cross? So one gene inheritance, Baba, you first explain about the monohybrid clause, definition of it, a cross between parents who differ in just one particular character. You mentioned about that. Then go ahead with the first generation, write the parents, then the gametes, then everything. Okay, make the Punnett square, explain the ratios if there is something. Then go ahead with the next set of parents, and that's how you go. 9:3:3:1. Hello, Kumar Jangwada. Okay, so many. 9:3:3:1 is the answer for A. Now identify the homozygous dominant genotype in the progeny. Come on, guys. Homozygous dominant genotype in the progeny. You have to find the homozygous dominant genotype in the progeny. Any idea? Homozygous dominant. From here. Homozygous dominant. Homozygous means both the things are going to be same. Capital A, capital A, capital Y, capital Y. Maya Gowda. Hello, Maya. Okay. Vinayak also capital R, capital R, capital R. Gia also capital R, capital R, capital Y, capital Y. Uday, dear, I will be doing a toppers answer sheet tomorrow. You can watch that video. Apart from that, yes, I have done several times. Yes. Yes, saw it. Okay, Pushpa, K, Lavanya, everyone says yes. So it is capital R, capital R, capital Y, capital Y. When you say homozygous, homozygous means over here the alleles are going to be similar. Okay, so capital R, capital R, capital Y, capital Y. This is what you mean by homozygous, and both of them are equally dominant as well. Okay, identify recessive genotype in the progeny. What is a recessive genotype, my dear children? Next question. Kirtana, Vinayak, Kumar, and Kumaranjan. Kumaranjan is, I do not know how to pronounce it. Venkatesh and everyone. Riddhi, Tanish. What do you think is the answer for recessive genotype in the progeny? Come on. Smaller, small y. Exactly. Maya Gowda, Kanadigana, as well as Hassan, Sanjeevani is back. Okay, Kirtana, everyone is giving the answers. By the way, if at all I'm missing out any names out there, I'm so sorry. So it will be smaller, small y. When you say recessive, it will be like this. Okay, number of parental type progeny. Any idea? Number of parental type progeny, how many of them will be like the parents? Will be like the parents. Any idea? If at all you know the ratio, 9:3:3:1, you will be able to find the answer. 9 of them are going to be round and yellow. This is parental. 1 of them is going to be wrinkled and green. This is also parental. Now over here you have 3 and 3. 3 is what? 3 is going to be 3 is going to be what? 3 is going to be round. Was with yellow, this time round will be with green. Here wrinkled was with green, here wrinkled will be with yellow. Now you tell me the answer, which one is parental, which one is non-parental. 9 of them. Rupa HR is also there. Hello. Okay, 9 of them. Okay, Shreya says 10 of them. Vinayak says 10 of them. So those of you giving me correct answer, I'm just reading out their name. Okay, push. Okay, 9:1 now. No, you will not say like that. Kirtana says 10, Vinayak says 10. Listen, whenever you're asked number of parental type of progeny, you just have to say 9 plus 1 is equals to 10. Okay, it will be just 10. Now, next question. Chalo, if at all you are able to understand this, you will be able to solve that also. Number of recombinant type progeny. Number of recombinant means new type of progeny which is there. Number of Numan Inamdar. Listen, 9:3:3:1 is the ratio. 9 of them are round and yellow, the parental character. 1 of them wrinkled and green, that is also parental. These 2 guys are the new characters. Round was with yellow until now, you make the new character round with green. Wrinkled was with green until now, wrinkled is with yellow now. These are the non-parental character. 6 of them, exactly. 6 of them would be the answer for the next thing, that is how many recombinant type of progenies are there. They are going to be 6 of them. Cool. Now, I'll ask you a question. This is, yes, perfect. Everyone, Deekshit, Kirtana, Jennifer, Riddhi, oh my God, Jia, Rupa, and Pushpa. And if at all I'm missing someone's name, I'm so sorry. Yes. So listen to me carefully. This type of questions are important. Please know about it. And also one more thing. I have asked this question so many times. I'll ask this once more. If at all in a question paper it is given that 64 progenies are being produced. Okay, 64 progenies are being produced. How many of them are going to show non-parental character in a dihybrid cross? If 64 progenies are being produced, how many of them are going to show non-parental character? How will you get to the answer for that? You're very close. Please try for it. Okay, SMN, Hegde, Kanadigana, everyone correct. Manya. Ma'am, important diagrams tomorrow morning at 10 a.m. The video will be released. Hassan, 24. Exactly. How did I get 24? Listen to me carefully. When I say 64 progenies are there already, okay, and I have to find the non-parental one. When we had 16 progenies, 9 plus 3 plus 3 plus 1 is equals to 16. Out of the 16, 3 plus 3, 6 of them were non-parental, right? So out of 16, 6 of them were non-parental. Now when you are asked about 64 of them, what do? You multiply with 64. What do you get? 16 into 4 is equals to 64. 6 into 4 is equals to 24. So out of 64, 24 of them are going to show non-parental character. And what about the rest? They are going to show the parental character. Is it okay? Did you understand it? If you understood it, give me a thumbs up. Exactly, Jia and everyone out there. Okay, Manya Shri and KV Lavanya, Vinayak, Rupa. Ammu, hello dear. SMN, Riddhi. Ma'am, MCQs 1. Dear, I had already solved the MCQs in the previous day. Please, if at all you can watch a recorded video of it, it will save a lot of your time. Okay, so watch the all one marker video I've recorded. It's there in the playlist. Okay? Clear? Thank you guys. Perfect. Okay. Ayyo. Now, what do we have? We have okay. We have the next question in 5 marker. Given below is a representation of amino acid composition of the relevant translated portion of B chain of hemoglobin. Okay. So, just a second. I'll show you something. So they are saying that this is how the translation or translated portion of the B chain is happening. I'll zoom this and show. You have to focus on this image really well. Everyone had the focus on the image. What is happening over here? It is GAG. Basically, in the mRNA, it is GAG. Okay. In the mRNA, it is GAG itself. And here you have glutamic acid being formed at the 6th position. Glutamic acid being formed at the 6th position. Now the question is, yeah, sickle cell anemia, no? Sickle cell anemia. Listen, this is where we do most of the mistakes. In every single place, no, usually what they have done is that they have changed GUG and they have mentioned valin. So our brain automatically thinks that this is a wrong thing that is given to us. No, this is correct one only, no. GAG is for glutamic acid and glutamic acid is there at the 6th position, right? Isn't it there? So do you think it's there for sickle cell anemia? It's normal person. The person is normal, macha. The person is actually normal. Listen to me. So is this representation indicating a normal human or a sufferer from certain related genetic disease? Give reason and support of your answer. This is a normal human being and we got fooled. Okay, this is a normal human being. Why normal human being? Because, listen to me. Okay. In normal people, GAG is there and GAG codes for what? Codes for glutamic acid. In people who have sickle cell anemia, you have, instead of A, you have U, GUG. And that codes for what, Baba? That codes for valin. Always remember, A comes before U. So it was A first in normal, U came later on. Similarly, G comes before V. It was glutamic acid first, then came valin. So over here in this particular picture, everything is correct. GAG is there, glutamic acid is being formed in 6th position. That means this person is totally fine. It's not been changed. So it's a normal person. Okay, so you have to support your answer like that. How do you write the answer for the first one? This representative indicates a normal human, not a sufferer. Reason, in the beta chain of normal hemoglobin, the 6th amino acid is still glutamic acid. That is the reason, right? Now the second question which is being asked, what difference would be noticed in the phenotype of the normal and the sufferer related to this particular gene? If at all, okay, if at all, I am saying in general, a person who is having sickle cell anemia and person who is normal, what is the difference that they will be having over here? In the phenotype, in the physical appearance. So obviously they do not look different. Do the people look different of the people who have sickle cell anemia and the people who are normal? No. Their cells are going to look different. Which cells, Baba? The red blood cells which are there. So normal person, RBCs are bioconcave disc-shaped structures are there. Whereas when you talk about a person who is suffering from sickle cell anemia, RBCs become sickle-shaped, okay, or crescent shape. Please remember that. That is a difference which is there. Exactly, Meena, the shape of the RBC. Okay, yes. And coming over here, who are likely to suffer more from the defect related to the gene represented? The males, the females, or both males and females equally? What about that? Come on guys, tell me the answer for that. When you say sickle cell anemia, what do you think? Who is more affected, males or females, or both of them equally? Biconcave to elongated. So biconcave to, yes, spindle shaped, yes. Mention spindle shaped, okay? Come on guys, I have a question for you guys. Both of them, exactly, Hassan. It's an autosomal recessive disorder. That means autosomal means anyone can have it, male, female, anyone. Okay, so yes, maybe equally, no, not maybe equally, it will be equally. Why? Because when I say a person is having, suppose it is like this, okay? HBA is the normal one, HBS is basically the gene which is affected. Similarly, another person HBA, HBS. Just suppose. Now when you cross it, this and this comes together, you get HBA, HBA, normal person. This and this comes together, you get HBA, HBS, a carrier person. This and this comes together, I get HBA, HBS, again a carrier person, because they have one being affected. Now finally, when this and this comes together, you get HBS, HBS. That person is going to have the disease. So out of 4, 1 is going to be affected. But can you tell it's a male or a female? No, because these things, my dear children, anyone can have it. This can be a male, this can be a male, this can be female, this can be female. I do not know that. You get it? So both equally, we never know. Both of them have equal chances of having the disease. Both males and females are equally affected. Why? The gene for beta chain of hemoglobin is located on an autosome, not a sex chromosome, right? Therefore, the defect is inherited equally in both the sexes. Got it? Got it? Understood? Fine. Yes, if it's perfect, you have understood it, just let me know. Thank you, Kanadi. Okay, where are my other friends gone? Okay, anyways. Yes, ma'am. So are you liking these type of questions out there? Remember, as I said, I am preparing you for the worst. So make sure you practice these questions. Make sure you know about everything. Dhanishri, Hassan, and Rupa, everyone. Thank you, guys. Okay. I think with that, we have completed this particular chapter as well. Do you have any confusions, doubts that you want to ask me? One more very important thing that is there from this chapter is sex determination. Okay, sex determination, I'll just brief you about that and then I'll go ahead with molecular base of inheritance. Okay, got to go, miss. We'll soon join. Yes, sure, Hassan. Okay, so listen to me carefully about the, sex determination as well. There's only one case where female heterogamity is being seen. Okay, female heterogamity is being seen. Female heterogamity means females are going to produce 2 types of gametes. That is being seen in 2 conditions only. That is seen only in 1 condition, that is in the case of birds. In the case of birds, there is ZZZW. Here, ZW is the female. The female is going to produce Z type of chromosomes and W type of chromosomes, which is why we call them female heterogamity. In the other cases, for example, when you talk about us human beings, we have XX, XY. Here XY chromosomes, X and Y separately, hetero type of gametes, 2 different type of gametes are produced from what? The human being, the males actually, the males, right? So this is something that is seen in what? Human beings as well as Drosophila. Apart from it, what do you have? You have one more example, and that is seen in insects. You have XX and XO. XO basically means you just have 1 X chromosome, other chromosome is not there. So basically, when they form the gametes, they can either carry on the X chromosome or they will not pass anything to the gametes out there. In that condition, it will be O. So here also, you see heterogamity. This is found in the case of insects. For example, you have grasshopper and things like that. And finally, what do you have? You have one more thing, and that is haplodiploid sex determination. Where do you see that? Ma'am, explain the honeybee one. Okay, I'll explain that. Okay, I'm going to explain that only. Ma'am, are you an Indian, ma'am? That's a new question, dear. Why did you ask me that? Okay, so this has been done, and now you have one more thing, that is honeybee. Honeybee shows haplodiploid type of sex determination. Okay. Why haplodiploid, and what do you mean by haplodiploid? Here, the haploid ones are going to be the males, and the diploid ones which are there, they are going to be the females. Always remember, haploid ones males and the diploid ones are going to be the females. Okay. Now, when I say that, suppose you're talking about a female honeybee. it will be having 32 number of chromosomes. Why? Because this is a diploid number. When you talk about a male honeybee, it will have 16 chromosomes. Why? Because it will be having haploid number of chromosomes. But how did we reach up to this particular level? First of all, how did we reach up to this particular level? Because of the sole reason is that when female honeybees, they form the gametes, they have to undergo meiosis. They have to undergo meiosis, and you know meiosis is a reductional division. The number of chromosomes becomes less. So in the gametes which are formed, they will have only half the number of chromosomes. That is 16 and 16. But males already have 16. What meiosis they will do, Baba? They will not do any meiosis, right? They will not do any meiosis. So basically, they will do mitosis and they'll form gametes. They are doing mitosis. They'll form gametes which is having just 16 number of chromosomes, just like how you had before also. Okay. Now, this 16 and this 16 comes together. Suppose this gamete and this gamete comes together. You have 32 number of chromosomes. You will say this is a female honeybee that is being born. On the other hand, how is male honeybee being formed? Okay, because of only one thing, that is here the gamete which is there, it is not going to undergo any fertilization. No fertilization is undergone over here, which is why it will soon transform into a male drawn. This is known as Parthenogenesis. This is known as Parthenogenesis. Very important thing, do not get confused between Parthenocarpy and come over here. Okay, so can I say that the drawns which are produced, the males which are produced, they do not have a father? Yes, they do not have a father. But do they have a grandfather? Yes, because the females are produced with the help of male and the female ones. So they have grandfathers, they do not have fathers. These are some things that are just given in your NCERT, that's why. Shivanand asked that because of your English is good. Thank you so much. Okay. Okay. I hope this particular thing is clear for everyone. If yes, can you give me a thumbs up, children? Explain bird chromosome, dear. I explained. They show female heterogamety, right? Guys, we are running out on time. We have to go ahead with the next chapter. Can we go ahead? Okay. Lac-operon, Rupa, I will be explaining soon about that because in the next chapter it is there. Okay? Guys, my dear children, I hope none of you are fighting with each other, right? Yes, perfect. So let's go ahead. Before that, everyone who is preparing for KCET, one very important update for you. The price have dropped. Okay, now KC30 is available for ₹2499. If at all you're interested, take it up. And also, there is an offline crash course. There is a flat 3,000 off going on. So if you're looking for an offline thing, we can go for that. Finally, there is ABHYAS KCET test series available just for ₹999. So anyone who is interested can go for it. Otherwise, let's go ahead. We enter the world of molecular basis of inheritance. Joint, ma'am. Is it, dear? Okay, that's nice. Love you, ma'am. Love you too, dear. Okay, that's so sweet of you. Okay, molecular basis of inheritance, 2 markers. Structure of nucleosome has a high chances to come. Apart from that, HGP related questions. HGP means what? Human genome related questions have more chances to come. Here I have given you so many questions, n list of questions. Do not be worried about them, seeing them, because the questions are 2 questions, okay? And they are for helping you to understand more concepts so that you can be safe. Now, when I say diagram based question, nucleosome definitely have a chance to come. Apart from that, transcription unit also has a chance to come. I threw my pen, just a second. So please make sure you go through it, okay? Just a second. Yes. Okay. KKR, hello dear. Fine. So, first question, starting with an MCQ. Average rate of polymerization during DNA replication is what? A, B, C, D. You have options. You have to choose from those options. The average rate of polymerization during DNA replication is what? Two. Okay. Come on, guys. I'm waiting for your answer. Kirtana says the answer for this question is C. What about others? Only one student is watching my video. Oh, Rishi Raj is there. Hello, Rishi Raj. Says the answer for this question is C. And, ma'am, it is Karko. Acha, Karko, I'm so sorry, KRKO. Okay, I do not know how. Okay, Kirtana, Sanjana, Shivanand, Jia. Okay, some people have different answers. Kannadigana, Jia, Vinayak, Police, Supershri. Okay, fine. So, what's the answer for this particular question? So, those of you who are saying A, 200 base pairs per second. What do you think? Is that correct? Wrong. Because the answer is 2000 base pairs per second. 2000 base pairs per second is the average rate of polymerization during DNA replication. This is given in NCERT. Should not be missed. Make sure you go to the details. It is 2000 base pairs per second, not minute. Okay, it is per second. So it is very, 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 very fast. Maya Gowda, can you explain again which question, dear? Okay, I hope this is super clear to you. It's not 200, it is 2000. It is 2000, okay? Ma'am, explain again. This question you're saying average rate of polymerization, as in, for example, you have DNA polymerase, okay? How many nucleotides does it add per second? That is the question. So average rate is 2000 base pairs are being added up, okay? That is the case. Cool. Alaina Sayada, you are back. You just have missed one chapter. The next chapter is right up there, molecular basis of inheritance. Do not worry. Okay, ma'am, PDF. So I think, Maya, now it's okay. You understood it? Yes, and we go ahead with the next one. How many base pairs of DNA are present in a typical nucleosome? Mention the types of amino acids present in a nucleosome. So these type of questions can also come when you talk about nucleosome structure, right? So please understand, first know the nucleosome structure. What is it? There are how many histone octamers are there? How many histone proteins are there? 8 of them, which is why you call it as histone octamer. Okay, 4 in the front, 4 in the back. Now, what about the DNA, Baba? DNA over here is going to be covered by something. DNA, sorry. DNA which is there, it is going to wrap around your histone proteins out there. Right? How many base pairs of DNA are there which is being wrapping around? It is 200 base pairs of DNA. Is this why you were confused between the polymerization rate? It is just 200 base pairs of DNA which is up there, right? And now going ahead, talking about the next thing, the histone protein which is there, one more histone protein is there. You should not be ignoring this guy. This is known as H1 histone. It is known as H1 histone protein. Together they are going to form a structure that is known as nucleosome. It is known as nucleosome. Your question is, how many base pairs of DNA are present? 200 base pairs of DNA are present. Proteins which are present, not proteins, basically amino acids which are present in the nucleosome is what? Do you know that DNA is negatively charged? I hope you guys know that. And what about the histone proteins? They are positive, negative, they attract, right? And what makes the histone proteins positive in nature? The amino acids. There are 2 amino acids. Los Angeles. That is how I remember it. Okay? Los Angeles. As in lysine, and you have arginine. Lysine and arginine are the things that you need to remember. Done. Now going ahead. Histone octamer, I told you about that. Linker histone is also known as linker histone. That's all. Okay, so explain that and your answer is being done. Jai Jasmine ma'am, thank you. And exactly, content is thank you. Maybe less people are watching, but the content you are providing is awesome. Thank you so much, Sampreet Gaming. That means a lot. Yes. Ma'am, computer science you will do, dear. If I do computer science, probably you will not come to this channel again, which is why another ma'am is there for doing computer science, okay? Ma'am, I like your voice. Thank you. I like you guys. Okay. Now, talking about one very important thing you should be knowing about that is, listen, these nucleosomes which are there, why do you have nucleosomes? Because you want to fit your DNA inside the nucleus. Nucleus is chunu and your DNA is very big, right? So how do you do that? You basically wrap around the thing and basically you put it inside the nucleus. Now when you wrap around, suppose this is a nucleosome, this is another nucleosome, another nucleosome, another nucleosome. The chain of nucleosome, it looks like bead on a string, right? You call this structure as chromatin. What do you call this? Chromatin. The chromatin which is there, when it condenses a bit more, you will now call this as chromosome. Okay, know about that. There are 2 types of chromatin, heterochromatin and euchromatin. The loose packing, if at all it is there, suppose this nucleosome is here, another nucleosome is there is loose packing in between, there is space in between. That means that RNA polymerase can come over here, do the transcription, do a lot of things over here. It is transcriptionally active type of chromatin. You call this as euchromatin, and it will be taking light stain. Why? Because there is so much amount of space. Now suppose nucleosomes are very close to each other. Nucleosomes, suppose they are very close to each other like this, no space at all. Okay, like toxic boyfriends and girlfriends. No space is provided over here. In that particular case, it is going to stay in dark, and transcriptionally, by dear children, it is not going to be active. There is no RNA polymerase that can come and attach over here. You get it? Okay. So please know about all the basic things out there. Apart from it, they also have non-histone proteins in the chromatin. Yes. Ma'am, how to score 45 plus? You solve these questions with me and you will not just score 45 plus, you will score 70 plus. I know the paper is out of 70. I was just being sarcastic. You will score really good. Ma'am, PDF you will get after the live, and yes. Okay. Ma'am, I'm still single. Be single. Do not be ready to mingle. Okay? Okay, let's go ahead. The next question that we have is, both the strands of DNA molecule do not act as template during transcription. Justify 2 reasons. Guys, can you give me the answer for this particular question? Okay, both the strands of DNA molecule do not act as template during transcription. Justify with 2 reasons. What are the 2 reasons that you have? Any idea? Ma'am, hi. Hello, Sudarshan. Any idea why 2 strands do not act as template during transcription? You might have seen that basically if this is one strand, this is another strand. One strand runs in 3' to 5' direction, other strand runs in 5' to 3' direction, right? You call this strand as the template strand. By the way, I am drawing a transcription unit, so everyone pay attention over here, okay? And this strand is known as a coding strand. This strand is known as a coding strand, right? Yes, perfect. Now listen to me. Over here you have the promoter region, and over here you have the terminator region. Why do you think transcription happens only in one particular, what do you call, strand out there? Because maybe, okay. Ma'am, guys, I need the answer for this particular question. Come on. What are you guys doing? Machas. Fast, give me the answer. Why both the DNA molecules are not acting like template? Why only one is acting like template? Coding strand. Ma'am, can I skip this chapter? Don't do that. Don't skip this chapter. Okay. So if both the strands act as templates, they would produce 2 different RNA molecules, which would code for 2 different proteins, causing confusion in genetic information transfer. This is going to code for something else. This will also code for something else. Okay, that is the reason basically you should not be doing that. Otherwise, there might be a huge lot of confusion around in the nucleus. Next thing, the 2 RNA molecules formed would be complementary also. Whatever is formed over here, for example, I have A, G, C, T strand over here, and I have opposite of it, that is A goes with T, C, and over here I have G, and I have A over here. When I make a strand opposite to this, they also will be complementary to each other. And do you think they will be doing anything else later on? No, Baba. That is also a reason. So 2 reasons are there. If both strands are there, basically they will produce 2 different type of RNA molecules giving 2 different type of proteins, and that could be difficult. The next one that we have over here is 2 RNA molecules formed will be complementary to each other, and basically that can form a double strand. Okay, ma'am, how many marks from the model question paper? We cannot deduce that, dear. Okay, we cannot deduce that in biology, because in biology I have never seen question to be repeated as such. Concepts are repeated. So apart from diagram related question, nothing else. So please, concepts can be repeated. So I cannot give you a guarantee on any of this thing. I'll say do not leave your model question paper. Go through the concepts which is there. I'm doing the same thing over here also. I'm going beyond that, okay? I'm solving difficult questions of the same concepts as well. So please have that. You can watch this video later also. Yeah? Yes. Asra, ma'am, I came home after exam, slept, I was fasting. I will start now. So how do I exactly start from where? I need out, ma'am. So dear, from the starting of this lecture, you can go and start watching. Okay, you complete it. It will be really good for you. Yes. Okay. Fine. Now, going ahead with the next question. Children, is this clear? If it's clear, can you give me a thumbs up, everyone? So I see a lot of scatter over here. I do not want that. Everyone, can you give me a thumbs up if it is clear until this particular point? Can we go fresh with the next question that is out there? Yeah? Is that okay? Ma'am, you work so hard for us. Thank you. Thank you so much, Manya. So Manya comes in between and tells some sweet compliments, and Yes, thank you so much. Ma'am, can you explain this once more? Yes, I can do so. Listen, so over here you have two strands, right? There is a promoter sequence and terminator sequence. The question is why both of these strands are not acting like a template for transcription. Because, listen, this strand will make an RNA, this strand will also make an RNA. Both of them are going to form different type of proteins. That is going to create a confusion. Listen, this is a normal cell. Here there are no robots involved in our nucleus. So it's a very systematic process that has to happen. So if at all we do that, it will create a lot of confusion within the cell. That is one point. Second point, when you make a complementary strand to this and when you make a complementary strand to this, don't you think that two new strands which are being formed, they will also be complementary to each other? Yes. So in that case, do you think they will be able to do transcription, translation, anything later on? No, Baba. They will not be able to do that. That is also a reason why we do not have both the strands acting like a template. Yes. H.T. Raja, I am really good. Thank you. Yes. Fine. Now we go to the next concept. I told you human genome project related questions you can get. Now I did not understand which question could be important. So I thought I'll explain all of it together. Listen to me carefully. Anything related to it could be coming in. Okay. First thing, what was the aim of human genome project? First thing, okay. First one was identify all approximately 20,000 to 25,000 genes in the human DNA. First thing. Second thing, determine the sequence of the 3 billion. Okay. First, you get to understand the genes, and after understanding the genes, you need to know the base sequences that is there in each gene. So determine the sequence of the 3 billion chemical base pairs that is there for the whole gene out there. Next thing, store the information in the databases. So whatever information that we got, we have to store it. After storing it, what do? We just store it and keep it up? No, Baba. We improve the tools for data analysis. Otherwise, what is the use of storing this data? We need to improve the tools on data analysis. Then what do? We transfer the related technologies to other sectors also, so that they can also go ahead and do their own activities out there. Fine, or basically find good things for us, yeah? And finally, to address the ethical, legal and the social issues. These are going to be what? These are going to be the aim out there. Okay, these are basically the aim out there. Next thing that we have is, have any other one more thing. The Human Genome Project was a 13-year-old project coordinated by the US Department of Energy and the National Institute of Health. Please remember, you can get an MCQ question because in preparatory, I remember this thing popped up. All of a sudden, this thing popped up and I was literally surprised. So I can expect anything from this chapter, okay, happening anywhere. So please know, Human Genome Project is a 13-year-old project coordinated by what? US Department of Energy and also the National Institute of Health. Know that. Fine. Now one more thing is there. One more thing is that many non-human model organisms. So usually we think that only human beings are being decoded and things like that. No, there are other organisms also which have been known. Okay, for them also the DNA has been sequenced. What are these organisms? Examples is C. elegans. Example is bacteria. Example is yeast. Example is Drosophila, that is fruit fly. Okay, apart from that you have plants. In plants you have rice and Arabidopsis. They also have been sequenced. So listen to me carefully. Think once more. What are the organisms? Bacteria, yeast, C. elegans. C. elegans is basically a nematode. It's a sort of round one sort of thing. It comes in that particular category itself, not exactly round worm. Next thing that we have is Drosophila, that is fruit fly, and in plants we have rice and Arabidopsis. Cool. These are the new organisms that were also being sequenced. Now, approaches for DNA sequencing. This is a question that is there in your MQP. What are the two ways in which you can do DNA sequencing? So one is expressed sequence tags. Now you might feel that I am speaking Spanish, but it is not. Okay, so this is there in your NCERT. Please listen to me carefully. Expressed sequence tag means this is your DNA. DNA. And DNA is very long. Do you think every part of the DNA is being transcribed? No, Baba. Only sub part is going to be transcribed. So whatever is transcribed, basically we are going to just sequence those particular parts. That is known as expressed sequence tags. So basically the ones where the DNA is going to get transcribed, those regions are being sequenced. That is known as express sequence tag. That is one way. Otherwise, we will sequence everything. That is known as whole genome sequencing. That is the next thing. Whole genome sequencing basically means each and every part of the DNA, whether it's coding, non-coding, everything is being sequenced with the help of sequence annotation method. Who gave sequence annotation method? It was Frederick Sanger. Okay, Frederick Sanger. S here and S over here means the same thing. Got it? Is this particular thing clear to you? Give me a thumbs up. LC basically means ethical, legal and social issues. Okay. Ma'am, I'll start watching this video all from the start. Does this video have all MQPs as well as extra questions? Extra questions are there. MQP related concepts are there. Questions from MQP I have not kept it over here, dear. Okay. So if at all, Aksa, in that particular case, if you go to the playlist, okay, if you're someone who want to solve MQPs and the other questions that is there, and not very, very new questions, if it's just a starter, there is a video that I have done. Complete biology. There's a recorded video which was stimulated live. It is 10 hours long, but okay, it's 1 marker all is there, 2 marker is there, 3 marker is there, 5 marker is there, everything is there chapter wise. You can watch that. You have to go to the playlist in our channel. For that you have, and under that you have to go to biology. You will see complete biology thing. Okay, crystal clear. Thank you. Okay. Kids zone, ma'am, nothing is going in my mind. I have studied, but I am really scared. I have been there. Okay, I have been there. Listen, stop this video. You go, listen to some great music, wash your face, and think that whatever happened, I am going to restart. Okay, or probably you can take a page and write out what the issue is there. Find the solution, you'll understand it better. Come back and then watch the video. Okay, ma'am, perfect. Let's go. Now, there are a few more things about Human Genome Project which is there. That is, human genome contains how many things? These are basically the findings of HGP, which also you should be knowing, right? So, ma'am, 3 hours successfully completed. Hey, full God, thank you so much, Lavanya. Okay, 3,164.7 million base pairs are there in our human genome. I hope you know that. 3,164.7 million. No billion, it is million. Okay. Next thing that we have is what? The average gene contains 300 base pairs. When I say average gene, a gene which is there, it contains what? 3,000 base pairs. But then there is this guy that is dystrophin. Dystrophin is a protein. It contains 2.4 million bases. One gene contains 2.4 million bases. Do you think that's crazy, right? Yes. So that is also there. Now the total number of genes which is there, it is 30,000. Until now we have studied 3 things. Whole genome is having 3,164.7 million base pairs. Average gene size is 3,000 base pairs, but dystrophin has 2.4 million base pairs. And then we studied that basically, if you estimate the total number of genes out there, there are 30,000 genes out there. How much? 30,000 genes. Going ahead. Next thing. The functions are known for over 50% of the discovered genes. Rest 50% we do not even know their function. Only 50% of the genes which are, listen, these are the genes which are discovered. Out of that, 50% of the genes we know their function. 50% we do not even know. Now, going ahead, less than 2% of the genome actually codes for proteins. If at all you talk about the whole genome, only less than 2% of it is actually going to code for proteins. Rest of them are chumma sitting there, okay? And what do you think the rest of things are? It is basically repeated sequences. They do not have any direct function as such, but basically they give us an idea about how the chromosomic evolution and everything has taken place. Okay, that is very much important. Now, one important thing that can come in your MCQ is this. Which is the chromosome that was being sequenced first? It is chromosome number 1. And chromosome number 1, my dear children, has the maximum number of genes. How many is the maximum number of genes? It is 2968. So, 2968. You know how do I remember it? Papa 7 guy is out of the game, okay? 7 is not there, rest every number is there. 9, 6 and 8 is there. 7 is not there and 2 though, you know, right? So 2968 is the highest number of genes which are there on a chromosome, and that is found in chromosome 1. And Y, chromosome of Y, that is in the males, my dear children, they have the least number of genes, and that is only 231. That is only 231. Apart from that, scientists have also found that there are 1.4 million places where I and you differ only in single basis. So I might have AGCT, you might have ACCT, and someone else might have ATCT. So single nucleotide morphism might be there. My meaning for Jumma settings here. Oh my God. Okay. Guys, I hope this is super clear to you. Please know the numbers. That is why I have detailed them as well. If it's clear, can you just give me a thumbs up? So I told you that you can get questions based on nucleosome related thing and also HGP related thing. Apart from that, I have explained everything which is there, which is important. Make sure you go through it. And I'll go ahead with the next thing. 5 marker questions. Hi ma'am. Hi Yashwant. Children, understood? Clear? Understood and clear? If yes, give me a thumbs up. Thank you, Sanjana. Thank you, Srina. Hello. I'm seeing new names out there. I feel so happy. Okay. Guys, please drink some water as well. Understood. Thank you, Jia Krishnamurthy. Okay. Now from here, lac operon, transcription and RNA polymerases, search for DNA, genetic code related questions have more chances to come. So this is a very broad segment. Okay. Different type of questions are coming over here. Ma'am, I take a break. If you want, you're tired. Maybe you have back pain as well. Still doing it. Thank you so much. It means a lot. Thank you, guys. So today I have back pain, but day after tomorrow you have examination. So we do not have a choice either way, right? So yes. Drink water, stay hydrated. Okay, passing package tomorrow. I'll be coming live for that. You are the only person who made molecular base of inheritance easy. Thank you. Yes, okay. So let's go ahead with the first concept that is there. Okay, there's a question like this, okay? There is a question like this and okay. Guys, give me the answer for this particular question. Name the process involved in the following. Okay, you have to try. One parental DNA strand is conserved by the daughter cell. What is this particular process? Name the process which is there. Only one parental strand is being conserved. Only one parental strand is conserved by the daughter cell. Which is this particular process? What do you call this? Thank you for your support. Thank you for your support as well, Kanadigana. Ma'am, how much can we expect? I cannot give you the numbers, dear, because even I'm not sure about that, but I'll request you to go beyond your MQPs as well. Okay, do not just stick to your MQPs. Read it, obviously. Go ahead with that. Passing package, tomorrow I'll be definitely coming live for that, dear. Okay, I still have to make the PPT for that. Guys, I'm asking you a question. One parental DNA strand is conserved by the daughter cell. What process is this? Remember, normally what happens, the DNA strand is there in one particular cell. Now, when you make two daughter cells, what is going to happen? The DNA strand separates and you get a new strand altogether. Now, they're asking you a question, what do you call this particular process when one parental strand is conserved by the daughter cell? What do you think is the answer? Call me Sampu. Okay, Sampu. Semi-conservative replication. Exactly. Okay, and Kesaya and I think Manya gave me the correct answer. Krishnamurthy also. It is known as semi-conservative. So I'll just rub off the answer. It is semi-conservative replication. Next question. Separation of DNA fragments. What is the process with the help of which you do separation of DNA fragments? Separation of DNA fragments. Kirtana also gave me the correct answer. Jia also gave me the correct answer. Ma'am, at what time? At 3 o'clock I have kept the passing package live, dear. Ma'am, make last main HOTS question live, please. I'll check, okay? Gel electrophoresis. Yes, Jennifer says the correct answer. It is what? Gel. Oh, you saw the next answer also. It is gel electrophoresis, the separation of DNA fragments, right? Next thing is removal of introns in hnRNA. Removal of introns in hnRNA, it is known as what? It is known as splicing. Remember, from the DNA, you get the RNA. So whatever thing is there in the DNA, introns, exons, everything is there in the hnRNA also. But this hnRNA has to form the mRNA, the mature form of RNA. And it says that I do not want introns, please remove it. And that process of removing the introns is known as what? Splicing. It is known as splicing. Okay? Yes. Now, attachment of Rho factor to the DNA polymerase during transcription. What do you think is the answer for this? Attachment of Rho factor to RNA polymerase during transcription. Come on, try the answer for this. Yes, everyone gave me the correct answer, splicing. I am seeing the Abhi, hello dear, K. Lavanya, Vinayak and Krishnamurthy, Yashwant, Kidzone. I mean, the answers are coming a bit here and there, but yes, you are attempting it and I'm really happy for that. Initiation. Alaina says initiation. You're talking about Rho. You're talking about Rho factor. Think about it. Think and say. Okay, no, nothing happened. Rho factor, come on. This is important. It's important. Termination, row dependent termination, exactly. So for those of you who do not understand this question, listen to me carefully. 3' strand, 5', 5', 3', promoter, terminator, right? RNA polymerase, if at all it wants to transcribe the template strand, it needs to come and attach over here, but it is super shy. It is what? Super shy. So shy. S is for sigma factor. So with the help of sigma factor, it will come and attach over here. Okay, it attached over here, and basically it was making the new RNA strand over here. This is the new RNA strand which was being made. Now the time came when these two guys had to break up. DNA and RNA was breaking up. So it is crying, and crying in Hindi you call this as Rona. Okay, what do you call it? Rona. So Rona basically crying. R stands for what? Rho factor. It is crying because it has to break up. Okay, so towards termination you need rho factor, and you call this as rho dependent termination. Fine, you call this as rho dependent termination. So the answer for this particular question would be this. I hope you will not forget it now. Yeah. And polymerization of amino acids to form a polypeptide on mRNA. Polymerization of amino acid, that means the grouping of amino acids together to form a new polypeptide chain is known as what? Basically translation. That happens in translation. You get too many amino acid sequence, right? That is translation. Do not get confused. Sometimes they can give you the question in a way that makes you like, oh my God, I will not be able to do it. So yes. Now explain the experiment to prove the DNA replicates semi-concentrically. That is what do you call important experiment, measles and stall experiment. Make sure you go through that also. Anyways, so I hope these type of questions are clear to you. Everyone who feels that until E everything is clear, just give me a quick thumbs up. We'll go ahead with the next one. Yes? Thumbs up, thumbs up. Ma'am, please sing the song. Song? Macha. If I sing song, everyone will sleep, no? Okay. 5 marker. In the context to lab copperon, when Jacob and Monod cultured E. coli cells in medium devoid of lactose, E. coli cells formed colonies, and when lactose was added, the E. coli cell metabolized lactose by synthesizing enzyme. Explain how lab copperon switches off and on. So basically, it is just a way of basically, what do you do for wasting a lot of your time? You just need to know how does lab copperon work. Disha KB, ma'am Malayali ano? Ade, na Malayali ano. Okay, anyways, let's go ahead. Let's see how lac operon works, okay? And it's a simple working base out there. So there is a promoter gene, and this promoter gene is for a, so promoter sequence is for a regulatory gene. Listen to me, if at all you listen to me carefully, you will be able to solve any question related to lac operon, okay? So over here, promoter region for what? For a regulatory gene. I'll call this gene as I gene. What is this gene? This is going to regulate everything. This is a regulatory gene. How does it regulate? It actually over here, it regulates negatively. Okay, ma'am, what does it produce? It produces repressor protein, Baba. It produces what protein? A repressor protein. Okay, so this is for that. Agreed. Now, ma'am, what is the space over here? The space is for something more useful. Listen to me. So there is another promoter region. Okay, another operator region for 3 genes out there. Z, Y and A. You know Zyada, so Zia, you can remember like that. That is how the flows goes. Okay, Z over here stands for what, Baba? Z over here stands for an enzyme that is known as beta-galactosidase. It's an enzyme that will catch up the lactose, break it into 2 things, glucose and galactose. So it is what? Beta-galactosidase. Okay, what does it do? It breaks down lactose into glucose and galactosidase. Now, Y gene which is there, Y gene codes for what? Y gene codes for permease. Why? Because you want to increase the permeability of the cell. What permeability, Baba? What is happening over here? Listen to me carefully. Where is the lac operon? Please, Shobha, do not spam. I'll give you the answer. Okay. Lactoperon is where? Lactoperon is inside the E. coli, no. And you want lactose to enter, that means you want to increase the permeability of the cell. So permease increases the permeability of the cell. Okay, what about A? A is coding for trans-acetylase. Now you believe that trans-acetylase do not have any big function, but listen to me carefully. Trans-acetylase is going to deal with everything that is there. You know, so many byproducts are going to be formed. There are so many toxic things that are going to be formed. Trans-acetylase is something that is going to deal with it. Now, you use your brain and tell me, when I am not growing E. coli in lactose medium, when I am not growing E. coli in a lactose medium, lactose is nowhere there. Do you think there is a need for all these enzymes? Chumma, you're wasting energy and your time, right, for doing transcription of these enzymes. So that's exactly when this guy comes into the picture and says that we do not need these enzymes. Why are we making it? Let's stop it. Okay, let's stop it. The repressor protein will come and bind over here, and I will not allow the RNA polymerase to do transcription. So RNA polymerase goes like this and comes back like this because it is not allowed to do transcription. Why? Because lactose is not there. This is the condition when lactose is not there in the media, and in this condition we say it is switched off. We say it is switched off. Okay, now listen to me carefully. Now listen to me carefully. In the E. coli medium, I am adding some lactose. Okay, now this lactose is going to go what? Lactose is going to come inside. When lactose comes, repressor protein is like, hey, Macha, you're back, Macha. Okay, I'll come for you. So it goes from here. The repressor protein and the lactose, they have been friends, BFFs from childhood. So these guys comes together. It is now free. It is now free. Since it is now free and it is going to undergo transcription, we say that basically transcription is on. This is switched on condition. But who was the one who is responsible for this? It is basically lactose, which is why I'll call this what? I'll call this as an inducer. It is helping in the process of transcription. It is not just lactose, it can be also allolactose. It can also be allolactose, which can act like an inducer, and that's when we say it is switched on. Now basically then this lactose is going to be broken down. Now there might be some children who might be confused, as in, ma'am, how did lactose come inside if at all these guys were not being made? So listen, transcription still happens, very less transcription happens, but it happens. Permease is still there, that is why lactose got entry at the first place. Okay, ma'am, will they make computer science video? Yes, they will. Tomorrow at 8 AM you have a live also, is what I believe. Ma'am, can you explain ZYA again, please? Definitely I can. Z codes for beta-galactosidase. It's an enzyme that will break lactose into two, glucose and galactose. Why? Because this glucose is a source of energy. Okay, permease increases the permeability of the cell membrane such that anything can enter the cell. Trans-acetylase basically is going to deal with everything in between. There are so many toxic substances which are going to be formed. There should be someone to deal with that, and that's the role of trans-acetylase. Got it? Okay. Spurti here, ma'am, you're so sweet. Thank you. Ma'am, how many lines we should write for 2 marker, 3 marker and 5 marker? 2 marker. Listen, 2 marker you can write 3 lines. Depends on the question, Macha. It's not like we can clearly say that like this is the number of lines you should be writing. In 2 marker, 3 lines. In 3 marker, depending on what the question is, there might be a small question. So write whatever is being asked, complete it and write it. Okay, 5 lines probably. In 5 marker, go ahead with that. Okay, so you guys know it's not 10th grade, okay, 8th grade where we have line system. Over here, obviously, whatever the question is, whatever is there, you have to answer it accordingly and be precise. That's all. Okay, ma'am, thank you for saying about CS. You're most welcome, dear. Okay. Killing expression by ma'am. Thank you so much. Okay, guys, is this clear to you guys? Okay, so there's a negative regulation that is being seen over here. Does lactoperon also have a positive approach? Yes, it has, but it is beyond what NCERT is, what is written in your NCERT as of now. Okay, cool. So I think I have the points for lactose operon. So make sure you write this properly. So when is it switched on? Role of lactose in regulation of lactoperon. In the absence of lactose, the repressor protein binds to the operator. This blocks RNA polymerase, so structural genes are not being transcribed. In the presence of lactose or allolactose, acts an inducer. It binds the repressor, changing its shape. The repressor cannot bind to the operator. RNA polymerase transcribes ZYA. And the soothing. Okay, this is what you need to write. Okay, fine, works. Can we go ahead with the next question, champions? Lacoperon is officially done from my end. If it's officially done from your end, give me a thumbs up and we'll go. For Human Genome Project, how many points should we write? Depending on the question which is being asked. If they're asking you just the organism, just name the organisms out there. If they're asking you, they will not ask you, listen, if at all they're asking you the findings, they will not ask you for a 3-marker. They will ask you for a 5-marker question. You get it? So you'll write the entire thing. Okay, they might ask you the aim for a 3-marker. So you know this question itself is small. So they know that. Yeah? Yes. Math 80 on 80. Yes. Oh my God. Perfect. Super proud of all of you. Okay. Thank you guys. Let's go ahead with the next question out there. Given below are sequences of nucleotides in a particular mRNA and amino acids coded by it. 5 AUG, UUU, UUC, GAG and go. Write the properties of genetic code that can be correlated from the above given data. This is from the model question paper of the previous year. Okay, this is important, please. Ma'am, I got 65 above in my preparatory, but I lost 2 and 3 mark in MCQs. How to score 20 on 20 in part A? Be very sure about your concepts. Probably concept knowledge might not be super strong, even though the marks are super great. I'm not saying it's not strong, but I think that we should be, you know, the concept is good, or probably when different type of questions comes from that, you are not able to answer it. That again means that there is some void here and there. So we need to solve more questions. That is the only way. If you have time, please solve questions from the model question. Sorry, question papers. QB, that is question banks. Yes. Ma'am, why lactose is called inducer? Because lactose is going to help in the process of transcription to happen. It is inducing something. It is helping something. That's why inducer. Okay. It's not definitely enough for MCQs, but yes, you should be doing it. Guys, come on. Ma'am, points I showed you, dear Gia. Please check it out once. Okay, now when you have something like this, what are the properties of genetic code that you write? Over here, we can clearly see that AUG, which is there, it is triplet codon. So this is one particular thing. All the codons which are there, they are in triplet nature, right? The second thing that we have is UUU, UUC, GAG, UAA, and things like that. Over here, if at all I see, all of them are read in a contiguous fashion. There is no full stop, there is no comma, there is nothing over here. So it is in contiguous fashion. Okay, rest thing, the next thing that we have over here is that each codon, for example, AUG is coding for methionine, UU is for phenylalanine, UUC is also for phenylalanine. So basically 2 codons are there for phenylalanine itself. What is this feature? This feature is known as degenerate feature, where multiple codons are going to code for the same amino acid. What do you call this as? Degenerate feature. Okay, next thing, as I said, one codon is coding for only one amino acid. Do you see that UU is coding for something else or things like that? No, each codon which is there, it is coding for one amino acid only. So I will say that feature is known as unambiguous nature. A codon does not code for multiple amino acid, only one amino acid. Now, the next thing, UAA is not coding for anything. So probably there are some stop codons which are not going to code for anything. How many stop codons do you know? Any idea? UAA. Apart from UAA, other stop codons which are not going to code for anything. Any idea about that? Alina says AUG has dual function. Yes, that is also a point. We'll come to that. Yeah? I'll write it. So AUG, which is there, it has dual function. What is the dual function? It codes for methionine and also it is a start codon. It is coding for methionine and also it is a start codon. These are dual functions out there. Guys, one more question is there. Please try. Stop codon, UAA. Apart from UAA, what are the other stop codons? UAG. Exactly, Gia. What about others? What about others? Come on. We need to try. We need to cover it up. UA. Okay. Alina said UAG and UGA. Exactly. Listen. UA, UGA, and UAG. Okay. UA, UGA and UAG. These 3 things are there. These 3 things are super important. Yes, Yashwant, yes, Muttaraj and Dia, Lakshya, nice name. Bushra, everyone. Nice. Nagu, RK, Yashwant, Vinay. Perfect, Karnataka. Okay, so these are the few features that you need to write. Please know that 64 codons are there. Out of that, only 61 of them will code for things. The 3 codons which are there, they are stop codons. Now there is one more feature which is there, which is not been shown up there, and that is codons are nearly universal. That means basically codons which are there, in the case of bacteria, a codon will be coding for the same amino acid. In the case of human beings also, the codon will be coding for the same amino acid. This is known as what? The universal nature of a codon. So this can be an MCQ question. So basically, if this codon is coding for this in bacteria, and this codon is coding for the same amino acid in a human being, what does it show? It shows universal nature. But again, there are exceptions. What are the exceptions? Exception is for the mitochondrial codons and for some protozoans. So mitochondrial codons and some protozoans, they are the only exceptions that you have. Rest everything is same. Done? Now I had a question. Yeah. So you know methionine is the codon that is coded by only one codon, right? You know that, I suppose. Rest everyone have multiple codons. Similarly, one more guy is there who is coded by only one codon. Any idea? One more guy is there who is coded by only one codon. Any idea what is that particular thing? I'm waiting for you. Hi Vishal, ma'am again. Okay, listen. So I am asking you that over here, methionine is something that is coding for AUG. Say, AUG is something that is coding for methionine, only one. Okay, apart from that, there is also one more codon. There is also one more amino acid that is coded by only one codon. What is that? Tryptophan. Someone said tryptophan. Mansi was the person who said tryptophan. So yes, that is the answer. Tryptophan. TRP stands for tryptophan, not trypsin. Okay, tryptophan. That is the one that is coded by UGG alone, no one else. So please know about all these things also. Apart from that, I'll tell you one thing. Can I give you one small homework? The people behind this homework is for you guys. Write it somewhere. You need to do it. The people behind the finding of genetic codon. Please do this. Important. Okay? Important. Do that. You might miss your marks. Okay, fine. Let's solve the next question out there. Answer the following. Is this question clear? Everyone who feels that the question is clear, I can go ahead easily, peacefully. Give me a thumbs up. We'll go ahead with the next question. We'll go ahead with the next question. Construct a complete transcription unit. Okay, with promoter and terminator on the basis of hypothetical template strand given below. So this is a hypothetical template strand which is given below. And what you have to do, you basically have to create a complete transcription unit altogether. And write the RNA strand transcribed from it. So you make the transcription unit, then you make an RNA strand transcribed from it, and then you do what? Write different types of RNA polymerase in eukaryotes. Okay, so listen to me, you have 3 different tasks and we have to do it. Thank you guys, Vinayak and Sanjana, Alayna, Rishraj and Yashwant, Kanadigana, and everyone. Yes, perfect. So over here, let's solve it. So this is a strand that is given to us. Always understand the arrow. Whenever you have an arrow sign over here, it means that it is a 3' end. Okay, and this will be oppositely, obviously, the 5' end, right? So let's make it. What is a strand that is given to you? 3' and 5'. Over here also you have to make another strand which will be in opposite direction, that is 5' to 3'. The first thing that you need to do is make the promoter thing. So this is the promoter and this is going to be the terminator. Then what do? You label it. What do you label? That this strand is what? The template strand. This is undergoing transcription. Template strand. Okay, and this is going to be what? The coding strand. Coding strand. Now what do? Whatever is written over here, you have to write it over here. Okay, right? So A, T, G, C, A, T, G, C and A, T, A, C. You write it like this. Now you make opposite strand of it. This is a DNA, right? This is a DNA. So you have to make it exactly like this itself. So A goes with T, T base pairs with A, G base pairs with C, C with G, A with T, T with A, G with C, C with G, A with T, T with A with T, and finally C with G. You made it, you make it properly. Done. Now what do we have to do, Baba? We have to do one more very important thing. Write the RNA strand transcribed from the above transcription unit. This was the DNA. We have to make a new RNA strand which is there. Hi Zainab. Okay, how do we make that? How do we make the new RNA strand? So listen to me. Do not put too much of your efforts. New RNA strand means it will be in the direction 5' to 3'. Why? Because here the RNA polymerase is going to attach, and basically it is going RNA polymerases, DNA polymerase, everything makes a new strand in which direction? 5' to 3' direction, right? Yes. So over here, what is going to happen? Now, will you write the entire thing again and again? Do not do this mistake. Listen. Wherever you had T, you have to replace with U, because whatever the new strand will be, it will be complementary to this only. It will be complementary to this strand, and you have already made a complementary strand. You just need to replace the T with U. So you write UACG, UACG, UAUC. That's all. Wherever T was there, you replace it by U, and that's how you got a new strand right there. Got it? Simple, macha. Fine. Did you understand everything until here? Everyone, happy birthday, mama. Happy birthday, mama. It's my birthday. Thank you, guys. Happy birthday, dear Sanjana. Okay, so I think people, since they are literally terrified for biology, they are finding reasons to be happy. Since that is the only reason, probably it might feel like it's everyone's birthday. Happy birthday to everyone. Please be happy. Okay, let's study. Come on. Okay. So, yes, guys, did you understand it? Did you understand it? Everyone who understood until this particular point, give me a thumbs up. Very, very important question. Thank you. Okay, Elena Saida and Vinayak and Rupa, everyone. Thank you. Yes, ma'am. Zainab also. Now, write different type of RNA polymerase in the case of eukaryotes. This is important. Okay. So, what are the different type of RNA polymerases? In the case of prokaryotes, no, there is only one RNA polymerase. Please know the difference between prokaryotes and eukaryotic transcription and everything. Super, super important. Very, very important. Okay. Now, over here, how do you write the RNA polymerases which are there? So there are, I know I have written the answer over here, but I need you to put your attention to what I am saying. Probably you will not forget anything about it then. RNA Pol 1, RNA Pol 2 and RNA Pol 3 is there. RNA Pol 1 is responsible for the transcription of what? First priority is given to something that letter starts with R only. Okay, you can remember like that. That is rRNA, ribosomal RNA. And there are different types of ribosomal RNA. And everything ends with 8. 28S, okay, 18S, and 5.8S. All these types are basically transcribed with the help of RNA Pol 1 itself. Now the next thing that we have is RNA Pol 2. What does it do? Second priority is given to transcription of DNA to hnRNA. That is our actual function, no? So formation of hnRNA, heterogeneous nuclear RNA is what you call it, which is basically the precursor of mRNA. That is being done by RNA polymerase 2. Now RNA polymerase 3 is there. 3T, okay, 3T. That is why over here it is going to transcribe tRNA. tRNA, and not just tRNA, there are some other RNA that is known as small nuclear RNA, snRNA. Apart from that, so small nuclear you have to write in small letters only, okay? Apart from that, from this guys, okay, one type of rRNA is not being transcribed up there, it is transcribed up over here. What do you call this as? You call this as 5S rRNA. So you know everything was ending with 8, which is why that guy was excluded over here. It is being put over here, 5S rRNA. Please remember this. Is this understood? Very important question. Happy Ugadi, advance happy Ugadi, dear. Once more, please explain, ma'am. Which one, dear? The A and B part is what you're asking, Riddhi? Are you asking about the A and B part? Ma'am, I can't understand this lesson. If it's too difficult for you, there is a masterstroke video that I have done. It will be helpful for you. Watch it. You're most welcome. Okay, A part. A part is something that you're not able to understand. Construct a complete transcription unit. This is one. So listen to me. Over here, they have given you a strand. They have given the arrow over here. That means this is 3' end. This is a 5' end, right? That means this is a strand that is going to be transcribed, because you know the template strand will always have 3' to 5' direction. The other strand will be having, the new strand which is being formed is going to be formed in 5' to 3' direction as well. So we make the template strand, right? We put promoter region over here. We put terminator region over here. We make a complementary strand out there. Done. This is our template strand. This is our coding strand. Which one is going to be transcribed? This one is going to be transcribed. So when you do the transcription, what do? You make an RNA from the DNA, right? Transcription means RNA from the DNA, right? So over here, A base pairs with U in RNA. Yes. So I've got U. T base pairs with A. Yes. G base pairs with C. That is how I go. If at all you see, there is a slight resemblance between both of them, because this is also complementary strand of this. This is also complementary strand of this. The difference is that this is a DNA strand. This is an RNA strand. You have to make a new strand in 5' to 3' direction only, and that's all about the answer. I hope, Riddhi, you have got it now. And also, someone else was also asking me that, Sanjana, I believe. Marshall Dierenberg as well as Matai are the founder of genetic codons. Exactly. There is one more guy whose name is Gamau. Okay. Looking good, ma'am. Thank you, dear. You're looking great too. Okay, fine. Let's go ahead then. If this is clear, guys, just give me a thumbs up. We need to go ahead with the next chapter. We cannot stay around over here for long. Ma'am, come to Bangalore. I have been to Bangalore. I've been to the mangrove forest also. Okay. Vishal, I'm fully heartbroken now, ma'am. Not able to perform good at anything. I'm not worth of anything. Realizing it's just an examination. Think, guys, it's just an examination. Always understand this. The thing that you're writing over here is just an examination. It does not determine how capable you are. Okay? But yes, if at all, it's like a challenge for you in life. You know, life is just built of challenges. Now you pass your school, you will have college. In college, you have deadlines, assignments, this and that. Then you come to job life where you have again assignments, this and that. So many things will be there. Every single time when you have a challenge where you feel that you are not able to do it, if you give up, will you be able to see the next thing in life? So along with challenges comes lots of beautiful things. You know, the college is the best period that you will ever have. And college also has so many bad things around it. The job life, my dear children, it might literally feel so bad at times, but it gives you basically happiness for some who follow their passion. It gives money who basically help in going around seeing the world or doing whatever makes them happy. So if every bad thing has something good, always understand that this is a phase, it will pass on, you go ahead. Okay? Yes. Yes, you will rock Biozanab. Yes, that's the spirit. Come on. Okay, now next question that we have over here, and thank you for giving me that thumbs up, and explain the experiment which was final proof for the genetic material. What is the next thing that is out there? Final proof for the genetic material is what you have, this particular thing, Hershey and Chase experiment. So in your MQP, okay, in your MQP, you had Griffith's experiment being asked. So you need to practice Griffith's experiment, but apart from that, do not ignore Hershey and Chase experiment, which is why exactly I've brought it up. So this is going to be the final proof for the genetic material, okay? And this is how Machalog it is going to go. So listen to me everyone carefully. Come on, everyone listening? Yes? So there were two bacteriophage, and these bacteriophage were going to eat up the E. coli, or basically they're going to attack the E. coli. These two guys which are there, they are viruses. What are they? They are viruses. Now, what do? I have a confusion whether DNA is genetic material or the protein is the genetic material. But I literally know one very important thing, that the outer thing which is made up over here is made up of protein, and the inner thing which is there, probably it could be made up of DNA. I am not sure. Okay. What do is that I use two very important things, radioactive things. One is sulfur. I call it S35. Listen, the 5 looks like S only, so you can remember like that. S35. And then you have radioactive P32 labeled DNA over here. Okay, what are you going to do? You are basically going to put it over here, you're going to put it over here. Now, you are going to infect the E. coli with this. When you infect the E. coli with this particular thing, what is going to happen? The general thing is that the bacteriophage is going to pass down its genetic material into the E. coli. That is how it usually works, okay? That is how it usually works. So it passed down. Suppose it passed down both the condition. Now you do blending. Why blending? Because all the bacteriophage which was there, you want to move it away. That is why you do blending. Now after blending, what do you do, Baba? After blending, you do centrifugation. Centrifugation is basically where everyone is going to make do like this. So when everyone goes around like this, what exactly happens is that whatever is what you call has high weight will be settling down. Whatever is like loosey things, they will be going up there. Okay, so what do you have? You have pellet and you have supernatant. You have pellet that settled down and you have supernatant. This is a known fact that obviously E. coli is heavier, which is why it will settle down, right? E. coli will be settling down. Whatever is left behind will be there in the supernatant. Okay. So if E. coli has settled down and if E. coli is radioactive, we get to know that basically it is carrying phosphorus or basically sulfur. We get an understanding of that. Let's see that. So basically, if you reached until here, you will be able to understand this also. Listen. So when we did this, we got an understanding that radioactivity was dictated in the supernatant, not inside the E. coli, but in the supernatant, in the fluid. Okay. And I tagged the bacteriophage here with sulfur, and sulfur is a part of protein. I know that sulfur is a part of protein. That means protein was definitely not passed down to the E. coli. Otherwise, probably the pellet could have been radioactive. But here, radioactivity is not been seen. Okay, now what do you have over here? In this particular case, pellet, again, supernatant is there. In the pellet, you have the E. coli, and the E. coli was radioactive as well. And this particular bacteriophage was being labeled with P32. And phosphor is a part of your DNA. So DNA was passed to the E. coli. That is why it was showing radioactivity. So that was the final experiment that was being done. And that's how we proved that what? DNA is the genetic material. Okay, class duration, dear, I told you it will be ending at 11 p.m. Okay, how much marks can I expect if I solve all model paper? Dear, you might be, I think, the 100th person who is coming and asking me this, and I am telling you 100 times also, no one ever can predict that in biology. The concepts could be repeated, the questions, I'm literally not sure the questions could be repeated as such. Because in a subject like bio, it will be very easy for you to score the question exactly is being repeated, right? So please do the MQPs, but do not just rely on the MQPs. You have to go ahead and practice questions from the question bank as well. That is exactly why I have taken a live up over here. Yeah? So children, is this particular concept clear to you? There is one more experiment, Griffith experiment. I think that you will understand that. Do you need me to make you, do you need me to explain Griffith experiment as well, or is it okay? Make sure you go through this experiment, super important. Also, semi-conservative replication of DNA experiment is also important. This is how you write the answer, write it properly, and yes. Okay? Ma'am, chota break after this session, is it? Do you want chota break? I'll give you chota break. Okay? Explain the points also. Okay, Baba, I'll explain the points. Listen. So, aim is to determine whether DNA or protein is a genetic material using bacteriophage infecting the E. coli. Infection. 2 types of bacteriophage were used. Okay. Left side, protein coated labeled with radioactive sulfur S35. Protein contains sulfur, but DNA does not. Here, right side, what do you do? DNA labeled with radioactive phosphorus. DNA contains phosphorus, but protein does not. These viruses infect the bacteria, both these viruses. Second process is what? Blending. After infection, the mixture was blended to remove all the phage protein coats attached outside the bacterial cells. Centrifugation. The mixture was centrifuged to separate pellet as well as supernatant. Okay, in the pellet you will find the bacterial cells, in the supernatant you will find the virus coats out there. Observation, protein labeled phages. Okay, protein labeled phages, no radioactivity in bacterial cells. Radioactive was found in the supernatant. Okay, in the radio labeled phage, radioactivity was found inside what? The bacterial cells. No radioactivity in the supernatant. This is something that is different, right? And what is the conclusion, Baba? Only the DNA entered the bacterial cells and that is the formation of new viruses, which is why DNA is the genetic material. Okay. Ma'am, I'm sure at 11 it will not be ending because you gave motivation to some, but they are not from bio and just came to enjoy and doing what is not needed. I'll end it. I'll end it at 11. That's my word. Okay. You know, that's a thing as a teacher. When someone comes and asks you something, you feel to answer, right? So yes, we cannot ignore that. Guys, so I hope you will be doing the semi-conservative replication of DNA experiment, measles and stall. This is something that you have to do from your end. Do not leave it. And Griffith's experiment. These two experiments are also super important. Please do that. Okay. Lac apron, dear Malika, I have already covered. Just go through it once, Baba. Please. That will be really, really helpful. Okay. Okay. Perfect. So guys, shall we take a break of 5 minutes? Shall we take a break of 5 minutes? And then we'll go ahead with the chapter evolution. Yes, I get it. Okay. You will get the PDF post the live, dear. Yes, ma'am. Okay, fine. Let's take a break then. See you at be right back at 8 o'clock. Ma'am, I'm proud of you, ma'am. Oh, thank you. I'm proud of you too. Hello, hello, hello. Am I audible? Children. Guys, let's go ahead with the next chapter out there. Yes, ma'am. Elena Infectious. Okay. Thank God you're back. Yes. Guys. Okay. Let's go ahead with the first question that is there from the chapter Evolution. And it goes like this. Pouch mammals of Australia survived because of lack of competition. Statement number 2. Continental drift has both positive and negative effect on population. What do you think is the answer for this particular question? Hello, ma'am. Good evening. Good evening, champ. Okay. Children, question for you. Come on. A, B, C, D. What do you think is the answer? Both statement 1 and 2 are correct. This is directly from your NCERT. From the question bank actually. And yes, Shravan Kumar and my dear children, let's not talk over here, guys. We have so many media in life, right? Social medias are there, and you feel Jasmine ma'am's live is the perfect place to talk. Okay, Sanjana, okay. Both are correct. Okay. Masi says A, Sanjana. Okay. Rishirat says C, Rupa says A, Jia says A, Alaina says both are correct. Okay. A, Sanjeevni A. Okay. Both statement A and, 1 and 2 are correct. This is the actual answer. And we solved it in yesterday's life. It's one of the MCQs, right? So listen to me. For those of you who do not know, please look and listen. Okay. Pouched mammals of Australia survived because of lack of competition. Yes, they did. Before that, you need to have a basic idea that from reptiles you got mammals. Right. Now you had mammals, and there were some mammals, that is South American mammals. South American mammals. Okay. Now what exactly happened is that there was a continental drift part away, which is why South America joined up with what? North America. South with North America. In North, there were some big organisms out there which basically outcompeted the Papa South, South American mammals out there. Which is why what happened? Basically, the North ones, they succeeded, and the South ones, they didn't. Now, the next thing that we come across is that because of the same continental drift, there were some pouched mammals of Australia. Okay, so pouched mammals of Australia, they did not have anyone else to compete with. Why? Because every one of their competitors went away because of the continental drift. So can I say continental drift basically has a good and a bad impact, positive and negative? Obviously yes. And pouched mammals of Australia survived because of lack of competition? Obviously yes. So your answer for this question becomes A. Done. Now we go ahead with the next question that is out there. Okay, so we go ahead with the list of things out there. 2 marker and 3 marker things. In 2 marker, analogous and homologous organs related questions and evolution of plants have more chances to come. Apart from that, there are other questions also being mentioned over here. We will be seeing few of them right up over here as well. Okay, so let's go talk about that. And in 3 marker, diagram of Stanley Miller's experiment and also adaptive radiation of the mammals has more chances to come, but there's no guarantee of it. So be ready with all the other questions as well. Yeah, Stanley Miller's experiment, anyway you have to practice the diagram. Hi, hello Preeti. But apart from that, you have to look for the other things also. Are you hearing me, everyone out there? Yes, so let's go. First thing, in the 2 marker, they asked you a question, that is, mention the 2 key concepts of Darwinian theory of evolution. Come on, my dear children. The 2 key concepts of Darwinian theory of evolution. What were the concepts? Tell me. Ayo, what were the concepts of Darwinian evolution? Just see, Summa said that evolution is happening, let it happen. Or did he have an actual thing to say about it? Yes, once the board end, we will do KCET. Guys, I'm asking you a question. What were the 2 theories of Darwin's theory of evolution? 2 very important key concepts out there. Come on. We need to complete the live. Ma'am, how many marks from this session, if at all? I wished I'll say 70 on 70, but then obviously I'm trying for it. Adaptive radiation now. ACNHT, no. Chemical theory of evolution, no. Guys, this is where we lose our marks. This is where we lose our marks. We might not know the key concepts. So key concepts are two of them. One is branching descent and the other one is natural selection. Perfect, Rakshita. Okay, branching descent and natural selection. Branching descent means basically we come from our ancestors. We have ancestors, basically they divide, they reproduce, and that's how we come. Natural selection means the one which are fittest in nature. The nature will be selecting them, and those will be the ones which will be reproducing and keep on going. So these are the two key concepts. Branching descent, all organisms have evolved from a common ancestor. There was a common ancestor. Okay, through gradual, slow changes. Always remember, Darwin always said there are slow changes. Natural selection, individuals with favorable variation survive and reproduce more. So these are the two key concepts of Darwin's theory of evolution. If at all it is clear, just give me a thumbs up. If at all it is clear, everyone give me a thumbs up. Yes? Perfect? Okay, it's okay. That's how we learn, right? That's how we learn. Next question is, genetic drift is a chance while gene flow is constant. Substantiate. Genetic drift is a chance but gene flow is constant. How can you substantiate that? How can you substantiate that? Any idea? Thank you. Chudu. Okay. Is there anything that you want as a doubt? If you have anything, I can tell you. So anyways, genetic drift is chance. Listen to me carefully. Abba, listen. Over here, you have, suppose, green moth, and you also have yellow colored moth. You have green moth and you have yellow moth. The green moth is saying, I am the one which is dominant. I am going to dominate over anything. You go. You should not be right over here. Then one fine day, one big elephant came and stamped on all the green ones, but the yellow ones survived. So it occurred by chance. This is known as genetic drift. This is known as genetic drift. Gene flow is constant. Why it is constant? For example, I am over here. We Indians are over here. Do you think that Americans cannot come to India? They do come to India. There are so many tourists from different places who come to India, right? So isn't the gene flowing out there? Yes. So suppose when I am saying, for example, in an area you just have tall and dwarf people. In an area, you just have tall and dwarf people. From somewhere else, people can come out. Medium-heighted people can also come over here, right? Don't you think the allelic frequency is going to change over here? Yes, Baba. It is constant. It can constantly happen, right? So how do you frame your answer? Genetic drift is chance while gene flow is constant. Genetic drift, you just tell the definition of it. It's a random change in the allele frequency due to chance events, especially in small population, right? Whereas gene flow is a transfer of genes between populations through migration, and it is something that is happening continuously and causing continuous exchange of alleles. Okay? Can't it be called artificial selection? Is there a term like that? I'm not sure. Okay? So please make sure you understand this. I hope it is understood. Okay? So basic thing is just there. So when I say the green ones or yellow ones, they have genes, they have alleles. So green ones probably capital G, the yellow ones probably small g. If I say the capital G is there in more amount, the frequency of capital G is in more amount, I cannot always stick to that. Why? Probably tomorrow elephant can come and cramp over them, and probably I might have this allele in more amount. The frequency of the other allele can increase. But this occurs by a chance, it occurs in a small population. That is genetic drift. Gene flow is basically gene migration and stuff like that. Is this clear to you? Everyone who feels that this particular part is clear, give me a thumbs up. Ma'am, please reply. Model paper in the Eshtu Barte. So dear, even I cannot tell that how much would be coming from the model question paper. But please make sure you go through it, go through the concepts of those questions. Okay, please do that. Okay, coming back. Is this clear? Everyone who feels that it's clear. Ma'am, my brain stopped braining. I get it. It happens. It's because of this chapter. Don't worry, your brain will come back to the same position. Okay? Yes. Perfect. Next thing. So these two questions are something different that I added up such that you have a practice of these type of questions as well. So know that. Okay? And apart from it, let's solve. What is convergent evolution? Give an example of convergent evolution in plants and animals. Riddhi, your brain will be coming back and braining now. Okay, so what is convergent evolution, Baba? Convergent evolution means probably there were different ancestors, but now they're staying in the same habitat, so they're converging to a point, might be having some similar functions as well. For example, you have a butterfly and bird. Both of them are in the sky, same habitat. Have they come from the common ancestor? No, Baba. But have they the common function? Yes. They are flying, they have wings. Okay, structurally are they similar? No. Butterfly wings you can do like this. Bird's wings, can you do like that? Don't do like that. Okay. Anyways, so these are the few things that you need to keep in mind. Okay, got it? So over here, it is independent evolution of similar functional traits in unrelated organisms due to adaptations to similar environmental conditions resulting in analogous organs. That is too complicated a definition. So you can just write that basically convergent evolution means the organisms would not be having a common ancestor, but rather, since they're staying in the same habitat, they will develop similar functions and they will have, yeah, that's all, the similar functions. But structurally, they are not going to be similar. And these type of organs which are like this, which have similar functions but are not structurally similar, are known as analogous organs. Then you have to give the example of evolution of plants, convergent evolution in plants and animals. Listen to me. One example that we have over here is for plants is the root and the stem tumor of this thing. What do you call that? Sweet potato and potato, right? What happens over here? They do not have a common ancestor. So sweet potato, Baba, they have the stem. Here also potato has the stem, right? Suppose this is a soil, this is a soil. Now what happens? Here the root transforms into storage units. In this particular case, in the case of sweet potato, they transform into storage units. What about over here? In this particular case, my dear children, the stem itself transforms into a storage unit. But aren't both of them storing? Yes. Is it from the common structure? No. So that means they are storing. Why? Because they are staying in the similar habitat. They are staying in the similar habitat. That is why. So can I say it's convergent evolution because they were not related but now they have common function? So I'll say it is convergent evolution, and these are what? These are analogous organs. Here this is stem tumor, here this is what you call root tumor, and this is what? This is potato and that is sweet potato. This is an example for plants. In the case of animals, you have wings of butterfly and the wings of bird. Apart from that, what do you have? You have so many other examples. For example, eye of octopus and eye of human beings. Eye is for seeing only, but basically is it made up of the same structure? No, we are not related. They are invertebrates, we are vertebrates. Now next thing that we have, flippers of penguins and flippers of dolphin. Okay, flippers of penguin and dolphin. Penguin is a bird. Okay, still it uses flipper to swim. Similarly, dolphin is a fish. Sorry, no. Dolphin is a mammal. Forgive me. Sorry, Shaktiman. Okay, dolphin is a mammal. Still it has the fins, and basically it uses flippers and it uses for basically moving around. So go through it, the examples. It will be really good if at all you know that. Okay, similarly, the examples for analogous organs are also super important. Okay, super important. Ma'am, can you solve all the MCQs of chapters from the question bank? It will take too much amount of your time, and that time would not be worth it as of now. Okay, listen to me. Yes, ma'am, what is your name? My name is Jasmine. Myself, Sania, ma'am, because of you, I scored out in biology practicals. Thank you. Oh, thank you. Sweet of you. Okay. Anyways, children, if at all you have got it, just give me a thumbs up. Everyone who have got convergent evolution, give me a thumbs up. Need a thumbs up. Guys, I'm asking you a question. Yes, thank you. Again, ma'am, this one. Ma'am, what is the probability? Probability could be 70%. Okay. Perfect. Let's go fast. Okay, go fast. My God. Name the evolution of any two plant forms through geological time periods from the Tachiophyte ancestor. This is your MQP question. I put it here for a reason that it can show up and you might be tensed. Okay, so listen to me very carefully. In the case of plants, you need to know the basic thing out there. Okay, the basic thing is What do you have? You have algae. From the algae you got bryophytes. From the bryophytes you got pteridophytes. From the pteridophytes you got gymnosperms. From the gymnosperms you got angiosperms. Okay, so always remember this thing. Algae is there, then you have bryophytes, then pteridophytes, then you have gymnosperms, and finally you have angiosperms, right? So in this particular picture, you are being asked to give the evolution of a plant form from the tracheophyte ancestors. So listen to me. First thing is chlorophyte ancestor. Chlorophyte means the ones that have chlorophyll, and that is found in the algae. Initial organism that came up is what? Algae. So over here you have chlorophyte ancestors, that means algae. From algae, Baba, you got bryophytes. So bryophytes, if at all you see there's a line going up there, here in the case of Cenozoic era, you got the bryophytes. Okay, now we have tracheophytes. Tracheophytes are nothing but pteridophytes itself, the lower forms of pteridophytes. Listen, pteridophytes are the first vascular plants. They started having xylem and phloem, and a part of xylem is trachees. That is why you call them tracheophyte ancestors. So they are the ancestors of what? Pteridophytes. So from tracheophyte ancestors you got rhinier type plants. From rhinier type plants you got xylophyton. You have to remember this. Okay, what do you have? You have tracheophyte. From tracheophyte you got rhinier type plants. From rhinier type plants you got xylophyton. Now from xylophyton, my dear children, you are going to make pteridophytes and you are going to make gymnosperms also. Listen, so over here anyway they are ancestors of pteridophytes only, right? So if you go at this particular level, you see Spinopsid, horsetails, these are pteridophytes. If at all you go once more, it is ferns, these are pteridophytes. In which era is this happening? This is important. Mesozoic era. In Mesozoic era, at what particular area? Jurassic and Cretaceous. Jurassic and Cretaceous. So you have ferns and you have Spinopsids. Ferns and Phenopsids. Basically, this is what? This is the evolution of pteridophytes until now. From here, tracheophyte, rhino type plants, xylophyton, and then you write ferns and also horsetails. Where is it happening? Mesozoic era, Jurassic and Cretaceous period. Done. Now we talk about gymnosperms, the next group of plants which are there. Yes. So over here, tracheophytes, then rhinotype plants, then xylophyton. From xylophyton, Baba, you get ginkgos, conifers, nettles, and cycads. Okay, or basically from here you get what? Ginkgos, conifers, and also nettles. These three things are there. These three things, Baba, are gymnosperms. Actually, cycads are also gymnosperms itself. So basically four things, but from xylophyton you're not getting cycads. Okay, so you write ginkgos, nettles, conifers, and cycads. C square and G square. C comes first and then comes G. Please know the flow also. That can also be asked. There's a MCQ question that I saw. Okay, got it? Very, very important thing. So conifers, cycads, ginkgos, and nettles. Done. Now if at all you want to make angiosperms, how do you do that? After xylophyton you have to put progymnosperms, then you have to mention seed ferns, and then you can mention dicot came first and then came monocots. Okay, dicot came in Mesozoic era, and in Cenozoic era you got the monocots. Got it? Yes, guys, what is happening up there? I missed a lot of things, is it? Children, please focus on what is being taught. It will be really, really good. Yes? Okay, is this clear? Ma'am, you look pretty. Thank you so much. Guys, is it clear? Is this concept clear? Questions can come. Please do not miss out on it. Yes, Alana. Yes, Rocky. What is happening over here? Ma'am, please explain again. Okay, Rocky Krishnamurthy says explain again. So yes, I have to explain. Wait. Just a second, guys. Yes, if artificial selection is there, it's not given too much importance over there. Don't worry, okay, guys, and come over here. Now listen to me. I'm telling again, everyone who did not understand, listen to me very carefully. Okay, I will not be repeating it. It might take a lot of your time. From algae, you got bryophytes, pteridophytes, gymnosperms. From them, you got the angiosperms. That is the flowering plants, right? So first thing is chlorophyte ancestors. By that we mean basically ancestors of algae, right? Now over here you have got bryophytes at the end. You have got bryophytes. After algae you have got bryophytes. Then you should be getting pteridophytes, but it's not a one-step process. In pteridophyte, Baba, you have certain ancestors' name that you have to remember. Tracheophyte, Rhinia type plants, Xylophyton. Tracheophyte, Rhinia type plants and Xylophyton. Done? Done. Now from Xylophyton, you actually make the pteridophytes. One pteridophyte is spinopsid, which you call as hostales. Another one is ferns. So basically you write this, and directly this, or this, and directly this. Both of them came in Mesozoic era. Okay, Jurassic and Cretaceous period of Mesozoic era. Done? Okay, now we talk about gymnosperms. Where did gymnosperms come through the picture? So over here, what do you have? Tracheophyte, Rhinia type plants, Xylophyton. From Xylophyton, what do you get? You get ginkgos, they are gymnosperms. Conifers, they are gymnosperms. Knee-tails, they are also gymnosperms. And cycad is also gymnosperm, but you get it from elsewhere. Okay, so C square comes first below and then comes G square. So always remember this is how it goes. So if you are asked or put them in correct order. So conifers, knee-tails, dicots, monocots. That is how it is going to go. So that is why I am saying C and this. Okay, done. Now the next thing that we have is basically angiosperms. So from Xylophyton we got progymnosperms, from progymnosperms we got seed ferns, from seed ferns we got dicots. In angiosperms you have dicots and monocots. So dicots first and then you have monocots. Dicots in which era, in which zone? Mesozoic, and monocots in Cenozoic era. That's all. Done, guys. Is it clear to everyone who is watching this video? Okay. Ma'am, morning class is there. Morning recorded session will be there. Live will not be there. Day after tomorrow. Okay. Ma'am, you look much pretty. Thank you, Irfan. Shaz. Okay. Okay, guys. Let's go ahead with the next question out there. Children, 3A. 3-marker question. Okay. Let's go ahead. Australian marsupials and Australian placental mammals explain convergent evolution and adaptive radiation. Justify the statement. Guys, is it clear? The previous question was clear. I am going ahead with the next thing. Please, is it okay? Biology is a beautiful subject with learning together. One with the best teacher. Thank you so much, dear. Okay, perfect. Thank you guys for giving me the thumbs up. Listen to me. My energy level is going down. Okay, so yes. So Australian marsupials, Australian placental mammals, you have to say how do they show convergent evolution. So you have not just studied about convergent evolution, you just said they show adaptive radiation. You never studied about convergent evolution, right? So today we are going to do so, and for that I need your help. Please listen to me very carefully. Suppose this is an island, and in this particular island you have the Australian marsupials right up over here. Okay, from this few of these guys went up this side. Okay, wait. Okay, few of them went this side, went this side, went this side, went this side, went this side. What does this show? This shows divergent evolution. Why? They had a common ancestor. They moved in different direction. That is why we say that they are showing what? Divergent evolution. They are divergent, right? Right. This is also an example of what? Adaptive radiation. Is it? Yes. Obviously, they are radiating and they are getting adapted to these particular locations out there. Perfect, perfect. Now, at the same location you think, one more set of guys came into the picture, that is placental mammals. They also moved this, it went again. Why God, why God, why God? Not today. God, God, God. Okay, anyway, this picture was easy, so it works. So Australian mammals. Placental mammals. Okay, so some went this side, some went this side, some went this side, some went this side, some went this side. When you talk about placental mammals alone, they are also showing one very important thing, that is what? Adaptive radiation, divergent evolution, because they also had common ancestors, they also moved away. But now, when you talk about both of them together, you have to look at this particular point, right? They were having different ancestor. They did not have in common ancestor. They were having different ancestor, but they are converging to a point. Aren't they? Yes, my dear children. They are basically what? They are converging over here. And this is super, super, super important example of convergent evolution. Okay? Okay? Got it? And yes. Fine? This is super important. Now, I hope you have got it, why it is showing convergent evolution. So, listen, suppose a A mouse came from one side, okay, from placental mammal side, a mouse came. From Australian mammal side, Australian marsupial side, there will be marsupial mouse. You get it? So they are staying in the same habitat, so they will be obviously having some common features as well. Got it? Yes. Now, if we read the answer, we have to write it like this. In Australia, many marsupials evolved from a common ancestor, agreed, and diversified into different forms adapted to different habitats in a geographical area. This is an example of what? Adaptive radiation. Now, in the same area, placental mammals in Australia evolved independently. Did they look at them and evolve? No, Baba. They evolved independently, but developed structures and habits similar to those of marsupials, right? Yes, and because of this same thing that repeated, my dear children, we see that they had convergent evolution. Both of them did not have any common ancestors. You can write it in your own words. And basically, they came up together. Cool. Now, over here, this question is super important in this one more particular case, and that is this. Please remember, placental mammal side, you have this. Australian marsupial side, we have this. You can get MCQ questions based on this, as in you have to pair them up. So always remember, the smaller names are in the placental mammals, the bigger names are in Australian marsupial side. Mole, marsupial mole. Anteater, numbat. This name is different, so please know, numbat comes over here, okay? Mouse, marsupial mouse. Lemur, spotted cuscus. This is totally different, you need to remember. Smaller name came over here, bigger name came over there, okay? Flying squirrel, flying phalanger, okay? Bobcat, Tasmanian tiger cat. Wolf, Tasmanian wolf. So the names that are totally not similar is flying squirrel, flying phalanger, but flying phalanger is a bigger name, right? And lemur, spotted cuscus. This is totally different, so you have to remember it. Anteater and numbat. This is also different. Cool. Understood? If understood, give me a thumbs up, everyone out there. Ma'am, very good teaching. Thank you so much, dear. Ma'am, come on, you're doing the best. We believe in you. Thank you so much. Even I needed some motivation out there. Okay, one day strategy has been released in the what do you call, YouTube. Go watch it. The Lights of Dean. Ma'am, your class is so long. It's for you. Yes. Short questions, dear. We are solving that over here. Okay, let's go ahead. Okay. Perfect. I think that's clear. And now we have state Hardy-Weinberg principle. Explain any 4 factors affecting the Hardy-Weinberg principle. This is super important. How do we do that? What is the advantage of marsupial mammals over the placental ones? As such, according to your NCERT, no such advantages that is there. Basically, 2 different life forms could sustain in an area. That itself is a big thing, right? Yes. Okay, guys, let's go ahead. Ma'am, by solving, by this session, how much do you solve? I'll say 70 and 70, but yes, you have to go ahead. Explain BT concept, we will be doing that. Okay, guys, we'll go ahead, I see. And please do not make me to a level so that I have to put you on timeout. You know, I can do that. I'm not doing that. Please do not do that, okay? Please do not talk amongst yourself. Listen. Okay? Yes. Go ahead. State Hardy-Weinberg principle. Listen, this is very, very simple. He said that, or basically Hardy-Weinberg, the law, it says that the allelic frequency is going to remain the same throughout in every single generation out there. Okay, for example, I say capital T allele frequency is this much and small t allele frequency is this much. According to Hardy-Weinberg, he is saying the frequency of these alleles is going to be the same. But do you think so that is possible? Obviously no, it is not at all possible. How can you think it is possible? Because as I said, these are the group of organisms which are there. Now, obviously, there are multiple factors that affect us. For example, I say mutation is happening. You do not have capital T, small t. Now, you have one more something else. One more thing that is something else. Another allele that is M, that is starting for medium height. You get it? Mutation is happening. New allele is getting added up. So don't you think allelic frequency is going to change? Yes. Now suppose, if not that, suppose that some more taller organisms are coming from somewhere else to this particular area. Again, you will see that the allelic frequency is going to change. Capital T will be more amount over here as well. This is known as gene flow. Now suppose that, leave that also. Second thing that we have over here is what? Suppose, okay, just think. something came up and basically all the tall people died because of that. It was occurred by a chance. All the tall people died and the dwarf people, they survived. It is occurring by a chance, but is it affecting the allelic frequency? Obviously, yes. Is the frequency remaining the same? Obviously, no. Right? So according to what Hardy-Weinberg said, he said that it is going to remain the same, but there are higher chances it will not be. Now, in sexual reproduction, you see genetic recombination, right? The chromosomes which are there, they lie close to each other, they cross over, they give new combinations out there. These new combinations are nothing but the new genes. Again, they're changing the frequencies. Again, they are going to change the frequency out there. The next thing that we have is genetic recombination is done, mutation is done, genetic drift is done, gene flow is done. The last thing is natural selection. You know, the white one, the black moth and the theory and things like that. The black ones, they were not surviving before, but they survived later on. Why? Because the nature selected it. Initially, their frequency went low, but they came back again. So isn't it changing? Yes, it is. Right? So make sure, listen to it very carefully. Okay. Ma'am, important questions. Dear, I am doing important questions. Okay. Okay. So, let's go ahead. This principle says that allele frequency in a population are stable and is constant from generation to generation. From every single generation, it is going to remain the same itself. And he denoted it by P plus Q whole square is equals to 1. This was the basic formula. Okay, it is going to remain the same. It is going to remain the constant. That is what is being put over here. Fine. Now, just a second. I think there is a need of an interference over here. Everyone who is watching this video, there is a request from my end. Please, amongst you, do not talk to each other. I am here for a reason. I am standing for a reason for a long time now. It's been almost, I think, 4 hours, 4.5 hours continuously. I have to take 2 more hours of class, 2 or 3. Yes, exactly. Yeah. And if you guys talk amongst each other, how would a teacher feel? Obviously not good. They feel demotivated. And I'm telling you honestly about that. How would you feel if at all someone comes into your face and talks just randomly and not talk to you? You'll feel bad, right? The same thing is happening over here. Please do not talk amongst each other. I will be putting you on timeout. Yes? Yes. Ma'am, we are asking doubts. I will be giving you time to ask doubts. If it is related to the concept, ask it. Once, okay, and I've given you the answer, okay? We are going chapter by chapter, and for those of you who are asking questions from other chapters, we are going chapter by chapter. We will be solving the questions from other chapters as well. Yeah? Good. We are good? We are done? Okay, whatever I had to tell you, I told you, that's all. Perfect? Yes. Okay, now, gene flow. Gene flow is basically a factor that affects the Hardy-Weinberg principle. I told you how. Basically, the transfer of genes between population of different gene pool which differ genetically but can interbreed is called as gene flow. Gene flow basically brings in new genes, okay, or probably will be changing the allele frequency of a proper population out there. Why? Because migration of organisms are happening up there, right? So this is super important. One thing, genetic drift is something that occurs by chance. It occurs by chance in a small population out there. Mutation are the spontaneous and permanent changes which leads to new genetic combinations out there. Similarly, what do you have? You have genetic recombination. It is the process of new combinations being formed because of what? Crossing over and things like that. Yeah, again the allelic frequency is going to change. Then you have natural selection. By the way, guys, if at all you know, there are 3 ways of natural selection. Please remember that also. That is also important. That is also important. One way is basically, suppose I am saying capital T and small t, capital T and small t, capital T and small t, that is tall and dwarf, right? When I am saying that both the characters are being selected, it will appear like this. This is known as disruptive natural selection. Disruptive natural selection, basically they have a break in between. Then you have 2 of them, right? Yes. Now the next thing that we have over here is what? Only the medium character is being selected. That is, probably neither tall nor dwarf is being selected. The middle character is being selected right over here, right? This is known as stabilizing selection. Now, finally, what do you have? You have one more thing. For example, only the dwarf, only one extreme is being selected. Only one extreme is being selected. This is known as directional selection. This is known as directional selection. Got it? Only one extreme and it is selected like that. Fine? Works? Yes? Cool. So this is what was there. Make sure you go through this as well. Also, if at all you get any frequency related question, please do that. Please numeric. Okay. So over here you get frequency related question, as in, for example, allele frequency of a dominant allele will be given to you, 0.4. Okay, that means that you have to take P is equals to 0.4. If at all they are mentioning that the allelic frequency of dominant allele is 0.4, that means you are saying that P is equals to 0.4. And now you have to find what is the frequency of the recessive allele. How do you find that? Listen to me. I gave you the formula, that is P plus Q square is equals to 1. Right, if at all we take the square root, it comes to P plus Q is equals to 1. I can just replace that in the formula. So over here I have P, that is 0.4, plus Q is equals to 1. So Q is equals to what? 1 minus 0.4, Q is going to be 0.6. This is going to be the frequency of Q allele. Now, if at all I am asked, what is the frequency of, okay, what is the frequency of homozygous dominant allele? That means I am being asked about P square. I am being asked about P square. Homozygous dominant allele. Dominant allele, that means I have to take 0.4's whole square. You get it? I am asked about frequency of homozygous recessive allele. That means I have to take Q square. Q square, because both two alleles will be there, right? Two recessive alleles will be there. So you have to talk about Q and Q square. Now, if at all you are asked about heterozygotes, the frequency of heterozygotes out there. Heterozygotes, what do you think the answer for this question will be? Guys, what do you think is the answer for this? If at all you are talking about heterozygotes. Okay, heterozygotes. Your P is 0.4, your Q is 0.6. What is the frequency of heterozygotes out there? Any idea? Any idea? Guys. Lena says 0.6 minus 0.4. No dear, it won't be like that. If you're asked about frequency of heterozygotes, you need to understand that there is a formula, P plus Q is equals to whole square is equals to 1, right? If at all we elaborate this formula, it comes out to be P square plus 2PQ plus Q square is equals to 1. Over here we got the homozygous dominant, this is the homozygous recessive, this is going to be the heterozygotes. So what do? 2 into P is equals to what? 0.4, Q is equals to what? 0.6. Your answer will be coming out to be 0.48. That is how you do it. Okay, I hope this is clear. Ma'am, for MCQs you made 150 plus that and model paper is enough. Okay, so for MCQs I have made a separate video altogether. It's a recorded video. If at all you want to watch it, go to the playlist in our channel. In the playlist you will see biology final. There you will be able to find all 1 markers. Yes? Yes dear, you can call me Akka. Ma'am, can you explain again this question or basically just the upper numerical part only? Okay, numerical part, right? Listen to me very carefully, okay? Over here, I am saying that the dominant allele which is there, it is, the frequency of it is 0.4. By that I mean, always remember, P is the one that denotes the dominant allele. Right? Now I got the value of P. How will I find the value of Q, that is the recessive allele? If that is a question, then I have to find the value of Q, that is the recessive allele. How will I find it? There's a formula. Okay, the formula goes like this. According to Hardy-Weinberg principle, he said P plus Q whole square is equals to 1. It remains the same. Frequency remains the same. If at all you take square root on both the sides, it comes out to be P plus Q is equals to 1. P is what? P is going to be 0.4 plus Q is equals to 1. What will be Q? 1 minus 0.4. This gets shifted over here, right? Q will be 0.6. You got it? The frequency of recessive allele is what? 0.6. Now you are being asked, suppose you are being asked about homozygous. Okay, homozygous dominant condition. Homozygous dominant condition. Suppose over here instead of dominant allele they have given capital A. Here they have given small a, and they have been asking you what is the frequency of homozygous A. That means you're asked about A and A, right? Or basically I can just say it's just P and P over here, right? How do you denote that? The formula which is there, you elaborate it. P square plus 2PQ plus Q square is equals to 1. This gives the frequency of what? Homozygous dominant allele. So P square is what? 0.4 whole square. It will be 0.16. If at all you are being asked about homozygous recessive allele, that is small a, how do you do that? Basically you are just talking about AA. That means Q square, right? AA means 1A is 0.6. AA means Q square only. So it will be 0.6 whole square. It comes out to be 0.36. Now you are being asked about heterozygous condition. You are being asked about heterozygous condition. In heterozygous condition, how do you do? Basically you write capital A, small a. That means 1P and 1Q. Right? 1P and 1Q. What is P over here? P is 0.4. What is Q over here? 0.6. And you have to multiply it with 2. This is what most of us miss. Okay, so over here we just multiply it with 2 and we get our answer. It comes to be 0.48. Okay, they can give you in this form point, otherwise they can ask you in percentage. If in percentage, just write 48%, 16%, 36%, whatever is there. Okay. Got it? Hi gaming legends. Hello, how are you? Children, got it? Can I go ahead with the next question which is there? Come on. Energy. Come on. We shall not go down. I am late too. It happens. Come on. Yes, ma'am. Perfect. Thank you. Ma'am, I'll take a screenshot. Yes, you can take a screenshot. I'm not sure if at all my handwriting would be that clear. Okay. Cool. Next thing. 33rd question. What were the different gases used by S.L. Miller in his experimental setup? Now listen, I told you that you can get a question based on S.L. Miller to make the diagram, or probably related questions to what exactly did he do, right? So what were the gases that were used by S.L. Miller during his experiment? What were the gases that were used by S.L. Miller? See, these were methane, ammonia, H2 as in the water vapor form, and also H2, hydrogen. So what do we have? Methane, CH4, ammonia, NH3, and we have H2O in water vapor form and also H2. These gases were being used. Yes? Yes. Okay. Now next thing that we have over here is what conditions of primitive earth was recreated in the flask. So what conditions were there that was recreated in the flask, my dear children? Listen to me carefully. Very simple. In the primitive earth, they did not have oxygen. There was a reducing environment. So in the flask also a reducing environment was being created. No oxygen was being put up. It was very much closed setup which was there. Second thing, in the primitive environment it was too much hot, too much hot. So for that over here, my dear children, electrodes were being put up, which is why the temperature was super high over here. How much high? 800 degrees Celsius high. That much high it was there. So these were the few features: a reducing atmosphere without free oxygen, and also presence of high temperature electric sparks out there. Okay, and the temperature was kept out to be 800 degrees Celsius. Cool. Now, the last one, write the conclusion drawn from his experiment. What was the conclusion which was drawn from his experiment? Very simple conclusions were drawn. What was it? Basically, he said that if at all these gases were there and if it was reducing environment and the temperature was super high, then probably from the inorganic things which are being mentioned over here, I am going to grab some organic components. I am going to get some organic components. And what were those organic components? It was right over here. Organic component that Stanley Miller got in this particular case was amino acids. It was what? Amino acids. This is something that they got. Okay? Yes? Done? Clear? It's done? So this was amino acids that you had to remember, and please remember in S.L. Miller experiment, nucleotides and nothing else was there. It was just amino acids. This is very, very, very important. Yes? And please make sure you know the drawing of this. And now we enter into the next chapter. Before I do that, I want you guys to just listen to me about for this thing that is there. Evolution of man. Ma'am, you're amazing. I really appreciate your efforts. Thank you, Slim Shady and Krishnamurthy. Okay. Alina, thank you, guys. Okay, perfect. Let's go. What do we have now? The evolution of man. With this, we'll end the chapter evolution and we'll go ahead with the next chapter which is there. Please go ahead with the, please know the importance of evidences, paleontological evidence, embryological evidence, comparative anatomy of all these things. Yeah? Yes. Gee, yeah, we just took a break before evolution started. We cannot afford a break as of now. Otherwise, I will not be able to complete the year. So please cooperate with me. I know you might be tired. Yes. Okay. So listen to me, evolution of man, you have two things, okay? You have two things that you have to go through. One is basically you had Dryopithecus, and then you had Ramapithecus. Listen to me, these were, Dryopithecus were more ape-like, they were like this, okay? They were more ape-like. What about Ramapithecus? They were more man-like. They were more man-like organisms out there. Okay, done? Yes. Now, over here, it happened 15 million years ago. These guys which were there, they evolved into so many things. But the things that we need to remember is just this much. First of all, we got Australopithecus over here. And basically, these were the people who started making weapons, as in stone weapons and stuff like that. And they started eating what? They started eating veggies, or basically not started eating veggies, they were vegetarians, okay? They were the ones who were strictly vegetarians, and basically they had the stored weapons for capturing the fruits and stuff like that. Okay, done. Next thing that we have is, by the way, this happened 2 million years ago. So directly from 15 million years ago, we get the data of 2 million years ago. Now, from them came most important things that you need to remember, and that is Homo habilis. Homo habilis. Okay, and Homo habilis, my dear children, are the first Habibi is there. Okay, is what? The first Habibi. Habibi as in the first hominid-like features or the actual human-like features were found in Homo habilis. So please remember, this is a very unique feature about Homo habilis. First hominid-like feature. Hominid-like feature. Ma'am, is there anything written about Homo habilis? Is there ear mentioned? No, ear is not mentioned, but their cranial capacity is mentioned. Okay, what was the cranial capacity? 500 cc? No. Think. Cranial capacity of them is what? Cranial capacity of them is not 500 cc for sure. That's something else. Come on, guys. Think. Yeah, brain capacity as in cranial capacity. Yes, that was 650 to 800 cc. 650 to 800 cc. And they were also eating what? They were also eating fruit only. Over here, please remember, ear is not been mentioned. Australopithecus 2 million years ago, it is clearly been mentioned. Now we come back again. From Homo habilis, we got Homo erectus. They started standing erect. And my dear children, The unique feature about them is that they were the organism who started eating meat. Okay, they were the ones who started eating meat, and my dear children, they came almost 1.5 million years ago. So 15 to 2 million years ago, then you put a point between 1 and 5, right? So 1.5 million years ago, you got Homo erectus, and their fossils were found in Java, Indonesia as well. Java, Indonesia as well, and they started eating what? Meat. They started eating meat also. Now what? Now what happened is that from Homo erectus, you got two things. First thing is the Neanderthal man. Neanderthal man. And Neanderthal man, my dear children, what's unique about them is that they were the first ones to have some emotions. They started covering their body, and my dear children, they started burying their dead also. So this is very unique feature, covering their body, and they started burying their dead. They buried their dead. By the way, I missed one more thing over here. The cranial capacity from 800 cc, it grew to 900 cc here in Homo erectus. Now, when you got Neanderthal man, they basically had a lot of cranial capacity, which is why they started doing all of this thing. Their cranial capacity was up to 1400 cc, and that was absolutely crazy, right? Yes, it was absolutely crazy. And they came around. Now, we were in million years ago, we came around, they came around in lakhs, okay, 1 lakh to 45,000 years ago. That's when these guys came up. That is Neanderthal man came up. And now what do you have? You have the next group of organisms out there, and finally that is us. So from Homo erectus only, Neanderthal man came up, and after some point of time we also came up, and that is Homo sapiens. About us, nothing is being mentioned, just that we basically went ahead with a lot of farming, this and that and stuff like that. Okay, so do not miss out on this. I hope I was able to explain you what is there. Do not miss any information which is there. Clear? Yes, dear, you can expect something like that coming up. Yes, Samir, I saw, and yes, you're right. And the year which is there of Homo sapiens, that has also been mentioned, 70 to 10,000 years ago. Okay. Clear? So it is 1400 cc, okay, not 1600 cc. Make sure you go through that. Is it new boards? Paper checking rule is there? Is true? I have to check with that. I have not heard about any new rules up now. Okay, guys. Clear. Hello, Bhanu Prakash. Hi, Hassan. You're back. Ma'am, KCB question bank model is enough to score 65 plus mark. It should be enough. Okay. Guys. The class will be up to 11 o'clock. Yes, done? Clear? Thank you. Let's go ahead then. Actually, my understanding about this concept is now clear. Thank you, JJ ma'am. You're most welcome, Zainab. Okay, perfect. Let's go ahead with the next chapter, that is human health and diseases. You have a 3-marker question and also a 5-marker question. In 3-marker, you can expect any infectious disease related question out there, the symptoms and causative agents and things like that. Apart from it, make sure you go to the other question also, and please make sure you practice the structure of antibody. Okay, practice the structure of antibody as well. Okay, done. Computer class will be there tomorrow morning, my champ. Start solving this question. The drug that is being abused by sports person is. The drug that is being abused by sports person is what? Opioid, barbiturates, cannabinoids, LSD. What do you think is the answer for this question? Hassan, my favorite chapter. Yes, it's your favorite chapter as well. It's mine also. Okay, guys, come on. What do you think is the answer for this question? The drug that is abused by sports people. That means they take it very often, and yeah, it's not good for them, obviously. Any idea? Opioids, barbiturates, cannabinoids, and LSD is the thing that you are having right over here. Mansi says the answer is C. Mansi says, another Mansi says C. Hassan says B. Okay. And C, B, and okay. Ravi says C. Pushpa Ja says cannabinoids. Zainab says C. Krishnamurthy also says C. Ayu says, ma'am, yes, dear, tell me. Class is there until 11 o'clock. Okay, so over here the answer for this question is actually C, cannabinoids. This is the one that is being abused, and under cannabinoids come charas, ganja, marijuana, hashish, everything that is there. Okay, done? So this is the answer. Please do not get messed up. This is the only thing that is being abused by sports people. Okay, Next question. Antivenom against snake poison contains what? Antivenom against snake poison contains what? Come on, guys. Preetham and Ravi also said C. By the way, I hope you're solving question number 35 now. So please give your answer like 35. What about computer class? It is going to be there tomorrow morning sharp at 8 a.m. and V. Okay. Darshan Grammar also says it is C. I think for the previous question you're answering. Yeah? Or is it the new question that you're answering? Kritika, I hope we'll reach up there. Yes. Preetham says B is the answer for this question. Okay. Manikanta says C. Manish says A. Vinayak says A. Zainab says A. Jia says C. Mansi A. Antigen. Pushpa. Okay. Guys, the answer for this question is going to be C. No. Preformed antibodies. Listen. Someone bit me, a snake bit me. Okay, not someone bit me, snake bit me. I went to the doctor. Will the doctor say, hey Jasmine, you must wait, let the antibody release antibody by itself, you will be safe. Wait, does the doctor do like that? No, Baba, the doctor will be giving me preformed antibodies because my body has never come across a snake venom, obviously, which is why we'll need preformed antibodies. This is an example of passive immunization. Your body is not actively participating. You are basically given antibodies. Okay, so your answer for this question is going to be C. So to those of you who have a doubt between this and this, don't get doubted. Don't have the doubt. Okay, it is preformed antibodies. The same goes with antitoxin of tetanus. Okay, in the case of antitoxin of tetanus also, you get preformed antibodies itself. I hope this is clear. Yes. Kids Zone C. Nitin also says C. Tara says B. Kids, guys, Gaming Legends say C. I hope it's sorted, right? It is sorted. Krishnamurti A. Okay, it happens. Yeah, perfect. Let's go ahead with the next question. Name the causative organisms of the following disease. Filariasis. Filariasis, the causative organisms of filariasis is what? Who causes filariasis? Who causes filariasis? Now, this is given in your MQP so many times, so which is why I thought I'll put it up here also. Filariasis is caused by which guy? Hello. Any idea? Filariasis is caused by Wuchereria bancrofti and Wuchereria malai. Okay, Wuchereria bancrofti and Wuchereria malai. So I'm writing like this: bancrofti. And what do you have? Wuchereria malai. Done, ma'am. Okay. What about, yes, perfect, Gia, perfect. Ringva macha. Okay. Vinayak, Mansi, Gia, Alena, and Rajat, Priya, Satvik. Hello, dear Satvik. Pushpaja, everyone is correct. Okay, next one that we have is ringworm. Ringworm disease is not caused by a worm, it's caused by a fungus. Everyone, I need the answer for this because I am pretty sure in ringworm most of you do the mistakes. Ringworm is not caused by any of the worms out there. It is caused by what? Trichoderma. Again, Hassan, remember, I still remember the mistake that you did in that particular live. And I told you, no. It's trichophyton. It's not derma. Trichoderma polysporum is elsewhere. Okay, trichophyton. Guys, yes. Alena, yes. So always remember, I'll telling you a way to remember it. There are two guys, trichophyton, epidermophyton. They are fighting. Trichophyton, epidermophyton. They are fighting. Fighting for what? Fighting for a small spore out there that is known as microsporum. So microsporum, trichophyton, epidermophyton, they are the one that causes ringworm, which is actually a fungal disease. It's what? A fungal disease out there. Yes, okay. And perfect, Harish, perfect. Everyone, okay. Krishnamurti, Ravi, Mansi. I am, yes, Nitin and Manikanta, nice name. Okay, Kritika, everyone, perfect. Okay, going ahead, malaria. Malaria is caused by what? Malaria is caused by three guys out there: Plasmodium vivax, Plasmodium falciparum, Plasmodium malariae. Okay, Plasmodium vivax, Plasmodium falciparum, and Plasmodium falciparum and Plasmodium malariae. Listen to me very carefully. The one that causes the malignant, the deadly form of this particular malaria is P. falciparum. Okay, the malignant or deadly form of malaria is caused by P. falciparum. Always remember that. Yes, perfect. Deepthi, this time we cannot predict which chapters HOTS question are going to come. Yes, but mostly molecular basis of inheritance and all. Can give you something. Okay. Got it? Perfect. Okay, now guys, I really want you guys to solve it, and I am showing you the picture right over here. Take your time and put the answers. A is ascariasis. Here the causative agent is ascaris. What are the symptoms of ascariasis? A. What are the symptoms of ascariasis? If someone is suffering from ascariasis, what are going to be its symptoms? Now this is a 5-marker question. Okay, ma'am had dinner. No, I didn't have dinner, dear. I am here. Okay, so basically this is a 5-marker question. The reason I am putting it over here is that I told you in 3-marker you will get a question something similar to this. Okay, probably a causative organism, symptoms and everything. So guys, I'm asking you ascariasis. Come on, come on, come on, come on. I cannot give you a trick, but I'll make sure that you remember it for a longer time. Dehydration, internal bleeding, muscular pain, fever, anemia. Exactly. Pushpa Jason. Yes, sorry. Okay, and Alaina, internal bleeding, muscular pain. Exactly. So listen to me very carefully. Ascariasis is caused by Ascaris lumbricoides. That is roundworm. Imagine the roundworm is inside the intestine. It is inside the intestine. Since it is inside the intestine, it causes a blockage in intestine. We call this intestinal blockage. First symptom. Okay. Second thing. Basically, since it is blocking the intestine, there will be some bleeding here and there. Okay, there is an internal bleeding here and there. So lots of blood is going to go. Since blood is going to go, what is going to happen? You will be having anemia. You will be having anemia. And obviously with all this thing happening, do you think you will have no pain? You will obviously have lots of pain as well. So these are the few features that you have to remember about it. And fever and all is definitely will be there. Got it? Simple. Okay. This is very, very simple. Computer science, please do not spam me. I teach biology. Computer science will be there tomorrow morning at 8 o'clock. Elephanta. Okay. Done, guys, done. Okay, perfect, clear. If it's done, can you give me a thumbs up, everyone who feels that ascariasis part is being done? Okay. Yashashwini Gowda, please, make sure you try to go ahead and draw the same diagram itself. Yeah, but yeah, you can, similar diagram also works. Perfect. Now the ring, okay, now someone gave me the correct answer again. So over here the name of the disease is being asked, and trichophyton is the causative organism. And what do you get? Appearance of scaly lesions on the various parts of the body, especially your skin and your hair. This is usually found in what? Guys, let's go one by one. This will be known as what? Ringworm disease. This is basically ringworm disease. Next thing, that is very simple, right? Now next thing that you have is typhoid. Typhoid is caused by what? Typhoid is going to be caused by what? See, Hassan says salmonella typhi. What about other people out there? Hassan says salmonella typhi. Yes, tomorrow morning at 10 a.m. there will be a video that is being released for diagram. Make sure you watch it. That I am doing it. I'm not Picasso, I'm telling you. Yeah. Salmonella typhi. So over here, salmonella typhi is the one that causes typhoid. And what are the symptoms? Again, this is also super important. Your small intestine is going to be affected. Yeah. And small intestine, since it is being affected, you will have constipation. Now, when you have constipation, you have stomach pain, right? When stomach pain is there, you feel irritated. And obviously, you have an infection, you will have high fever. Yeah. And you will have always weakness because you're not able to eat food because your stomach is already full. It's constipated, right? Nothing is going out. So there is a loss of appetite as well. There will be, what do you call, headache also because of all this thing happening, and there will be fever. These are the symptoms of typhoid. The ultra level symptoms of typhoid which is there is that intestinal perforation will be there. Small pores will be going to be seen, and there will be high level of fever also, 42 degrees Celsius and stuff like that. Yeah. Now pneumonia is caused by Streptococcus pneumoniae. Again, you have to give me the symptoms of pneumonia. Symptoms of pneumonia, anyone? Anyone? Symptoms of pneumonia. Symptoms of pneumonia, this is very much important. Dear Farhan, I cannot give you the answer for that because it will be wrong to say. I cannot guarantee how many questions are going to come from MQPs. The concepts might be repeated, so make sure you solve some extra questions as well. Okay, lower respiratory tract infection. Manish says lower respiratory tract infection. Yes. Common cold. Common cold is there for common cold. This is pneumonia, right? Okay, lips and nails turn blue to grey in color. Hassan. Okay. Cold, fever, finger. Okay, listen to me very carefully. I'll tell you a way. In pneumonia, your lower respiratory tract is being infected, as Manish said, and which is why you're talking about your basic things. Breathlessness. Okay, breathing issues will be there. There will be, definitely you will be having coughing. Okay, there will be coughing out there. You will have fever. Since fever is there, obviously, and cough is there, obviously you will be having headache also. All these things are related. Now, the high symptoms which are there are your lips and nails turning blue to grey in color and things like that. Okay. Please understand, common cold is going to affect your upper respiratory tract. That means in common cold, you will be having sore throat, you will be having cold, you will be having cough, you will be having lot of things that is around this particular area. In pneumonia, it's mostly related to the lower area out there. You get it? So please make sure you know that. And now, finally, dash. Over here, you have dash. Dash causes, and you have chills also. Fever, chills, cough, headache, severe cases. Lips and fingernails turn bluish green. Exactly, Pushpa Jason. Okay. Okay, come over here, guys. Over here, rhinovirus is causing a disease, and basically it causes nasal congestion and discharge, sore throat and cough and headache. I told you, nasal congestion. Nasal passage is there, discharge is there, there's mucus coming out, there's sore throat, there's cough and headache. This is the feature of what? This is the feature of what? Common cold. So over here, when I say rhinoviruses cause common cold. Yes, perfect. Sanjana, perfect. Everyone out there. Aditya, Darli, Kidzone, Shobha, Alaina, Zainab, Madhuri, Mansi. Perfect. Everyone. Hassan, everyone. Kidzone, yeah. Got it? Clear? Now we go ahead with the next thing that is up there. Name the infective stages of Plasmodium. Okay, guys, now this is something that is important. Might even make you think for a second. So please make sure you go through this question really well. Name the infective stages of Plasmodium which Anopheles mosquito takes in along with the blood meal from an infected person. Why does the infection cause chill and fever in humans? At what stage does Plasmodium enter into the human body? These are the few questions which are there. Can you give me the answer? Why don't you take my name? I'm so sorry, dear. I might have missed. Forgive me. Ma'am, can you make a video of 5-marker HOTS question? So I've made a whole video on important 5-marker questions. If at all you can, please watch it, because I don't think I would be able to do a separate video on that now. Yes? Okay. So first thing is, name the infected stage of Plasmodium which Anopheles mosquito takes in along with the blood meal from the infected person. From the infected person, it is going to take not the sporozoite, it will be gametocyte. Exactly, Hassan. Exactly, Sanjana. Okay, it will be gametocyte that is being taken up. Listen to me. If you feel that this particular portion is difficult for you, I can explain to you, because I know this particular question might be coming related to Plasmodium. So listen to me very carefully, everyone who is watching this video. And I know a lot of you know the answer already. So yes, there is mosquito. Okay, there is mosquito. Inside the salivary glands of the mosquito, what does it have? Basically, it has one very important thing that is known as sporozoite. Sporozoite. This is a stage of Plasmodium. Now, when the mosquito bites us, okay, now when the mosquito bites us, it is going to pass the sporozoite stage into us. Because why? The saliva is being passed. The saliva has anti-clotting enzymes such that it can drink the blood very peacefully, right? Now, sporozoite, when it enters the body, there are two camps. First camp, and there is second camp. By camp, I mean they're terrorists, and they have two camps. The first camp is basically, just a second. Why God, why? Why God, why? Why God, why? I'll explain it over here because there are high chances it will go. Okay. You have the mosquito. You have the sporozoites. And we have got it. And we have two camps. First camp is basically liver. In the liver, you have hepatocytes, that is your liver cells, right? So in each of the liver cells, my dear children, they are going to undergo asexual reproduction, asexual reproduction. Yes, they undergoes asexual reproduction, okay? Now what happens? Basically, their number increases way too much over here, such that they are going to burst open. They will be burst open and they are going to be released outside. Children, listen to me. They are going to be released outside. Now when they are released, where do they go? They mix up with the blood. This is the second camp. And in the blood, which is the cell that is affected most? It is going to be RBC. It is going to be RBC, that is red blood cell. Okay, red blood cell is something that is going to be affected. Now what happens in the RBC, my dear children? Very simple thing which is there. In the RBC, suppose this is an RBC, okay? Listen to me very carefully. It needs the blood meal. Plasmodium needs some blood meal. It goes and feeds the hemoglobin. It breaks it down into heme and globin. This is what actually the plasmodium wants. But heme, which is released over here, it is toxic. It is what? It is toxic for the organism, or basically, yes, the plasmodium out there. So what happens? The heme over here is being converted to something else that is known as hemozoin. It is being converted to hemozoin. Now, hemozoin is something that is less toxic for this guy, but you know what? It is toxic for us. So when the RBC, which is there, RBC, will it rupture? Yes, because why? Again, over here also it is undergoing asexual reproduction. When it ruptures, there is lots of hemozoin which is being released in our blood, and that is why we get fevers and chills. Why do we get fevers and chills? Because we feel that hemozoin is toxic for us. That is why, in a response to that, we get fevers and chills in the alternative days out there. Now, one very important thing that you should not be missing is that I told that they undergo asexual reproduction over here also. If at all you notice, they are undergoing just asexual reproduction in our body. They do not undergo sexual reproduction. Now listen to me. When they undergo asexual reproduction, which stage are they forming over here? They are forming a stage that is known as gametocyte stage. This is a stage which later forms the gametes in their body. But listen, it is ruptured, gametocytes are released, but the gametocytes does not form gametes in our body. Rather, this is there in our blood. Suppose I am the mosquito, I come to drink the blood. What happens? I now have the gametocytes. In my gut, the gametocytes are going to divide, form the gametes. They are going to fuse, do the sexual reproduction in the mosquito's body. And later form a state that is sporozoite that moves into the salivary gland. So always remember, blood meal is taken. When it is taken, it carries the gametocyte. When the mosquito gives something, it is basically the sporozoite. I hope this is clear. Okay? Clear? Cool. Now over here, if at all I see this question now, will I be able to answer this question? Let's see. Name the infective stages of Plasmodium which Anopheles mosquito takes in along the blood meal from an infected person. So what does the organism take in along with the blood meal? Any idea? Any idea? It is going to be the gametocytes out there. Yes? Perfect. Now the next thing that is there, why does the infection causes fevers and chills in the human beings? Fevers and chills in the human beings because they are releasing what? In the Plasmodium affected cells, they are releasing hemozoin, which is toxic for us, which is going to be toxic for us. Exactly, Hassan, Jia and everyone out there. Manish, perfect, perfect, perfect. Sanjana, all of you doing great. Okay, so it is going to be taking in, it is having hemozoin which is going to be toxic for us. Now what do we have? Alana, Manish, Satvik, perfect. Now guys, the next thing, what stage does Plasmodium enter into the human body? At what stage does the Plasmodium enter into the human body? So yes, rupture of the RBC is the correct answer for the previous one, but yeah, you have to also mention about the hemozoin, okay? Now at what stage does the Plasmodium enter the human body, guys? Any idea? Sporozoite stage. Exactly. Do not mess up with the spelling. It will be this. Cool. Sumit, Valian, tell me, what dear? Okay, so do not mess around here. And since we are anyway doing this question, listen to me carefully. It's a 3-marker question that I showed to you. Now, there are chances in a 5-marker section of human health and diseases, there is a chances of missing data of either of them, HIV virus or Plasmodium. So as in basically life cycle of HIV virus or Plasmodium can be coming in. So make sure you are pro at both of these things. Now, the next thing that you should be sure about is cancer and immunity related questions. They also have more chances. Ma'am, teach us with fun. Okay. So I usually teach with fun, but at this particular point of time, I think it's because of the pressure. The same pressure that you are having for yourself, I also have it, because I promised I will be covering by 11. Guys, are you getting bored? Anyone out there getting bored? Yes, Deepti, tell me. Ma'am, please tell the symptoms of hemozoin. When hemozoin is being released, you get fevers and chills. Tomorrow also we have live classes, yes, towards 3 p.m. I think so. Okay, so no is more. Yes, ma'am, bored. Okay, a little bit. I get it. I'll try my best to make it a bit more interesting. Okay? Yes. Thank you, Satvik. And okay, going ahead, guys. Okay, question number 38. Stages in the life cycle of Plasmodium is given below. Identify A, B, and C. So over here, machas, listen to me carefully. So you have to find what is A standing for, what is B standing for, what is C standing for, and what is D standing for. So we'll go one by one. Okay, we'll go one by one and then we'll go ahead. Okay, so first one is salivary glands. Salivary glands, they are basically releasing some things and those things are going into the human body. What do you think is that particular thing? Arjuna Chupuki, ma'am, thank you. Ma'am, I'm getting bored. Thank you. Guys, I cannot do much about it. Okay, I'm trying my best. I definitely know studying for long hours is a boring task. I definitely agree with that. But how to produce too much amount of energy after long hours standing, it's a difficult task, yeah? Yes. So, yes. Guys, tell me the answer for this particular question. Sporozoite, yes, the formation of sporozoite or basically transfer of sporozoite from the salivary glands to basically the human body. A is that. You guys got it correct and I'm super proud of you guys. That means you have understood the concepts even though the class is boring. Yes, yes. Okay. Now the next thing that we have is from the liver. Okay, in the liver something is happening over here. In the human host liver, something is happening. What is that something that is happening in the liver? Any idea? So if at all I zoom in, there are cells being formed up there and there is a rupture being seen. What exactly is this particular thing? Ma'am, my question, biological oncogene, that is a virus, dear. We'll talk about it. I have a question like that. Appreciate efforts. Thank you so much. Sure. Asexual reproduction in liver cells. Exactly, Manish. Okay, so over here you have asexual reproduction in the case of liver cells. That is what is happening over here. Now what happens? Finally it goes to the RBC. In the RBC, C happens. C is what? It is going to be asexual reproduction in the case of RBC and also bursting open of the cells. That is C. That is C. Right? And also we say that basically hemozoin is being released at this particular point of time. And now we have D. What is D over here? Any idea? What is D over here? If at all you have seen, the gametocytes are being formed. The mosquito takes up that capital gametocytes. Now what happens? What is this? What is D standing for? Liver cells is bursting. Yes, yes, yes. You guys are all correct. What is D standing for? Any idea? Fertilization in the gut. Exactly. So over here in this particular case, fertilization in the gut of the mosquito happens, my dear children. That is what you mean by the D. It looks like a sperm, right? Yes. Okay, perfect. Yes. So over here, this is a picture that is given almost in your NCERT. So if at all you want to take a look at it, you can take a look at it as well. So A is basically mature infective stages. Sporozoite escape from the intestine and migrate to the mosquito's salivary glands. When the mosquito bites another human, sporozoites are injected with the bite. This is your A. Okay, sporozoites are injected with the bite. Now, parasite reproduces asexually in the liver cells, bursting the cell and releasing it to the blood. Fine. That is B. Now C is what? Parasite reproduces asexually in the RBC, bursting the RBC and also releasing the hemozoin up there. Now what happens basically over here? Fertilization and development takes place in the mosquito's intestine. That is the thing. Ma'am is an angel. Thank you for saying that. Okay, now we have the schematic representation of our beloved HIV, that is human immunodeficiency virus. Are you ready for that? If yes, give me a thumbs up. Come on, quickly. If you're ready for that, quickly, give me a thumbs up. Ayyo, wait, something went on. Ma'am, I have a question. Yes. Tell me, Ayu, what is the question? Karthika, ma'am, if you have teached maths, we would have definitely topped. Ankit sir is the best at that, I'll tell you. Okay, perfect. Let's go ahead with HIV virus. Now, I'll try to bring out the energy in me. Okay, let's see. Okay. Listen to me very carefully. Over here, the virus which is there, its name is retrovirus. Okay, HIV is human immunodeficiency virus. It's a retrovirus. Now, over here, it has two RNA strands. We have what? We have DNA. Papa guy have over here has RNA, right? Done. Now, this virus, the only motive of this virus is basically to enter our cell, use our machinery and basically increase their number. But do you think that's easy? Obviously, that is not going to be an easy task, right? So, the virus comes over here along with the RNA. You call this single-stranded RNA, but there are two of them, okay? Single-stranded RNA are there. And along with some important proteins as well, and it is coded by some viral proteins as well, that is known as capsid. It gets entry into the cells. Probably it can be any cell, that is a macrophage. Macrophage is usually known as what? It is usually known as the, what do you call this as? The factory of HIV. The factory of HIV. Why? Because this is a place where the HIV viruses, they duplicate a lot of time. Okay? Now, listen to me carefully. At this particular point, okay, the viral RNA is introduced into the R cell. Yes, it is done. Now, we want to get into, suppose I am a virus. I want to get into the machinery. I want to use the machinery of the host cell. That means that I have to transform my RNA into DNA. And that is not something that is very common. Usually from the DNA, you get the RNA. Right? DNA, RNA, that is known as transcription. Now you're doing ulta of it, that is known as reverse transcription. Now for that, obviously you need some tools. And I have that tool. I have what? Reverse transcriptase. With the help of reverse transcriptase, I do ulta of it. From RNA, I get the DNA. Now I have got the DNA. I slowly have to get inside the cell. I have to integrate into the cell, into the nucleus, into the DNA of the host. I got it. But why did I do that? I wanted to use the machinery. Now, this guy does not know that it's my DNA out there. It will replicate, it will transcribe, it will translate also. I will get my viral protein, I'll get my viral RNA as well. And that is exactly what I wanted. I got the viral RNA, I got the viral protein, I can make so many baby viruses out there and finally I can move out of the cell. You get it? Okay, so this is super important. Fine? Yes. So now, in your MQP, you have, okay, first of all, you can get a question like this as in basically to draw schematic representation of HIV virus. That is a rare thing that could come, but yes, we never know. Okay, now the next thing that we have over here is this. You have retrovirus. retrovirus is going to affect, there are things that is there, you have to fill in the, what do you say, you have to fill in the blanks, okay. Virus infects the normal cell, viral RNA is introduced into the host cell. What happens now? Reverse transcription happens. After reverse transcription, what is the thing that is happening over here? Viral RNA is going to be formed, macha. Yes, viral RNA is going to be formed. Then what? Viral RNA which is going to be formed over here, it is going to get introduced into the nucleus of the host cell. It incorporates into the host genome and does what? And produces viral proteins as well as viral RNA. That's all. So if you know the basics of it, there is no one in this world who can mess around with you in this. Okay. Ma'am, for how many marks? So yes, it's a 5-marker question that may or may not come, either this or probably the plasmodium. Either this or the plasmodium can come, so make sure you go through this really, really well. Just a second, okay? Okay, perfect. So now 40th question we have, ma'am, DNA not RNA. Viral DNA, right? No, viral RNA is going to be formed. When it is forming, it is going to form the viral RNA. No, it wants to form the viral RNA. It inserted the DNA out there. The DNA gives the information to form the RNA, right? And it needed RNA. Its genetic material is RNA. So viral RNA and the proteins are going to be formed. Okay? Gia? Here when you are saying new viral RNA is going to be produced. From the DNA, you transcribed it, you just incorporated. Why? Because you wanted to make the RNA out there. There's only one way to doing that. You have to be like a DNA. The DNA itself undergoes transcription to form the RNA. You know, mRNA formation, RNA. So here also the same thing happens. Okay? The Deepti, before starting cancer, please explain immunization. Yes, we'll be definitely doing that. There's one more question related to this. I'll just solve it up and then we'll go ahead with that. Okay? Okay. Fine. So, ma'am, viral DNA is formed by reverse transcriptase. Yes, Alena, that is true. Okay, so double fertilization, triple fusion, please explain me, dear. I have explained it previously. Please go to the initial part of the chapter. Okay? It will be there. Okay, fine, guys. So let's go ahead with the next question. Name the diagnostic test. Okay, so this is about HIV positive. Okay, this is about being HIV positive, and over here you have to talk about the diagnostic test which helps in finding HIV. The person that is the person can take up for finding HIV virus. It is going to be ELISA. Yes, Manish, it is going to be ELISA. Yes, you tell me. What is the question? ELISA stands for what? Enzyme-linked immunosorbent assay, and please remember it is dependent on one very important thing, that is antigen-antibody interaction. Antigen-antibody interaction. This is super important. Think out there. Ma'am, doubt. Yes, Mamshi, please ask it out to me. Preetham, yes, dear. Which type of cancer is caused by tobacco and smoke? Usually the lung cancer. Okay, come. Now, the next thing, write the full name of the pathogen involved over here. What is the full name? It is human immunodeficiency virus. All these are straight questions, so I'll pace it up a bit, okay? Now, name the genome of the pathogen. What is the genome, my dear children? It's talking about the genetic material. So it is going to be single-stranded RNA. ssRNA is what you call it, okay? Single-stranded RNA. So ssRNA would be the answer for this particular thing. Ma'am, okay, I'll come to that. Okay, next thing that we have is which particular cells of this person are likely to be destroyed. Which particular cells of this particular person is likely to be destroyed. Always remember, the first cell that is going to be affected is macrophages. You call this HIV factory because it is going to divide up there. The target, the actual target is T helper cells. Why? Because T helper cells is something that maintains our immunity level. And now when these T helper cell is not there to mediate our immunity, our immune system will go low, and basically this virus can thrive in our body. So its main target is going to be T helper cells, and obviously the macrophages, where it finds a way to divide itself. Okay? Fine. Next thing that we have is what? Suggest the partially effective treatment to him. So if someone is HIV positive, what is the partially effective treatment that we have for them? There is only one thing, and that is antiretroviral therapy. Do you have a permanent cure? Obviously no. But you have antiretroviral therapy, which is up there. Got it? Yes. So will the test be accessible that I can attempt them later? Yes, Jayashri, you can attempt it at whatever time you feel you have. Okay, Preetham, what are exactly antigens are called? Antigens are called as antigens? Pathogens? Yes, anyways. Okay, done guys? Yes. Now, someone was asking me about immunization and vaccination, so listen to me out carefully. I'll not write, I'll make you listen. Okay. सब monkey human क्यों नहीं बने? Nice question. सब में evolution नहीं हुआ इसलिए. Okay, guys, come on. Now listen to me very carefully. Immunization and vaccination. That is an important topic, right? Anyways, okay. Two things. Vaccination is basically, first of all, active and passive immunity. Active immunity is basically when you rely on your body to produce something and basically act on the antigen. Suppose I am getting an infection. When I get an infection, my body naturally produces what? Antibodies against it. My body is doing the part, right? Yes. Okay. Suppose I am getting vaccination. What happens in vaccination? I am getting weakened or dead part of the pathogen or the antigen out there. Why? It is put inside my body such that my body knows that this type of thing can come sometime, anytime, and you have to be prepared. So when they put this thing inside my body, what happens? My body still produces antibodies. Again, is this active immunity? Obviously yes. So in vaccination, active immunity is working. What are you doing in vaccination? Basically, you're giving weakened pathogens to provide to the body such that your body can produce antibodies. Next time the actual guy comes in, it has already made the antibodies. It will just produce more and will kill it. Now, the next thing that we have is passive immunity. Okay, passive immunity basically means what? Over here, my body is not going to do anything. No antibodies are going to be produced over here. Rather, someone else is going to give me antibodies. How, man? Naturally, this can happen. For example, when a baby is being born, from the mother to the child, you get antibodies, IgA, through the milk, right? Is the baby producing it? No, the mother is giving it, right? This is passive immunity. Now, it can occur artificially also, and that is known as immunization. Listen to me. When we take an anti-venom, or when we anti-venom against a snake bite, or probably antitoxin against tetanus injection, what is happening over there? Our doctor does not wait for our body to release antibodies. Rather, we are given preformed antibodies. Yes, aren't we given preformed antibodies? Why? Because we cannot wait for our body to produce antibodies at this particular point of time. This is known as immunization. Especially, you call this specifically as passive immunization. I hope this is clear. Ma'am, give a break. I'll give a break right away. Okay, after cancer, I am going to give a break. Even I need a break. My legs are paining like hell. Okay. Okay, got it? Now, one more very important thing is there. And that is cancer. And with that, I'll end up this particular chapter. We'll move ahead with the next chapter. Name the group of genes that have been identified in normal cells that could lead to cancer. What are the group of cells that are been identified in normal cells and that can lead to cancer? It is proto-oncogenes. Proto-oncogenes means what? In our cell, they are there. They're generally there. They do the normal function out there. But suppose some mutations happens in them, my dear children, it can turn the whole cell into cancerous cell. Similarly, apart from that, please understand that there is oncogenic viruses also. What do? These viruses affect us. When they affect us, what do they pass? They are going to pass some genes. You call them biological oncogenes. They pass the genes to us. And basically, when we have it, we die. Okay? So this is very, very, very important. Now, the next thing that is there is which techniques are useful in detecting cancers of internal organs? Now, this question is super tricky. So, you know, there are multiple ways of detecting cancer. There is biopsy where you cut out the tissue where it is being affected and you test it under the, microscope and stuff like that. You're talking about internal organs. So you have few things. First one is radiography. In radiography, what you are going to do, basically, you are just going to take the help of X-rays. With the help of X-ray, you get an idea of what is happening inside our body. Second thing is computer tomography. This is a bit advanced form of X-ray. Here also you are using X-ray only, but here you are using it for generating what? 3D images, okay? 3D images of what? Your internal organs. So that is one very important thing. So these unique features which are there, you need to remember. 3D images of internal organs. This is also X-ray that you are using over here. The last one is MRI, comparatively very safer. MRI stands for magnetic resonance imaging, where what do? You use strong magnetic fields out there and you use non-ionizing radiation. So until now you were using ionizing radiation, which has a potential to change your DNA, can be cancerous also. But here you are using non-ionizing radiations. And why are you using it? For finding the problems in the living tissues out there. You find the problems in the living tissues also. So make sure you write radiography and then CT scan and then MRI. These are the three methods that you have over here. Okay? Yes. Next question that you have is, why are cancer patients often given alpha interferons as a part of the treatment? Why? Why do you think they are given that? Because, you know, they undergo radiotherapy, they undergo chemotherapy. After doing all of this thing, their good cells, their bad cells, everything die. When the good cells die, you mean that immunological cells die. They are weak over here. Their immunity is what, Baba? Weak over here. And if they are weak, okay, if they are weak, obviously they can be attacked by any pathogen. Do you think that's okay? Obviously no. That is why we need some biological response modifier, something that can boost up, just like how I have you guys in my life, right? So whenever I go down, I see your comments, I come back again. So the same thing is happening over here also. They are biological response modifiers. What do you call them? You call them alpha interferons. So they are going to activate our immune system and obviously boost up our body's ability to destroy the cells. Does it work? Yes. PRG, how does immune system works after snake bite? They also produce some antibodies, but obviously not in too much amount. We might die. It needs some time. So how does immune system works after snake bite? I told you. Yes. Part B once again. Yes, Jia, I can. Ramesh, I will just a second, okay? Okay. Okay. Part B is what? Part B is which techniques are useful in detecting cancers of internal organs. So there's, as I said, radiography, CT scan and MRI. Listen, you're talking about internal organs, so you need some rays and things like that. First is radiography, where you plainly use X-rays, okay, for finding problems internally. Then you have CT scan. Again, you are using X-rays only. It gives you 3D image of your internal organs. That is the next thing. MRI, magnetic resonance imaging. Over here, you are not going to use X-rays. X-rays are ionizing radiation. You are rather going to use non-ionizing radiations, and also you are going to use strong magnetic fields. Why? Because you want to get an idea of internal organs, especially the living tissues out there. So that is the next thing that you are having. That's all. Okay. Now, can I explain hnRNA? What is precursor of mRNA? Yes. hnRNA, which is there, you get it directly from the DNA. Okay. And hnRNA will be having introns, exons, everything. Non-coding, coding sequences just like your RNA, sorry, your DNA. Now, when you form a mature RNA, that is mRNA, you do not need the introns out there. So what do? You cut them off. And that process is known as splicing. And basically, when you make the mRNA, you have to make sure this guy should be easily able to go out of the nucleus, because out of the nucleus is where translation is going to happen. So you do capping and tailing such that all the portions are safe out there and it is not being cleaved. This is the only thing. That's all. If the question about snake venom is being asked, how to write? So you can just write that is an example of passive immunization where preformed antibodies are given to the patient. That's all. As simple as that. Yeah. Oh. Okay. So yes, I got to understand it. Okay, children. So we'll take a break now of 10 minutes. If that works, my leg is paining so very much. So I will meet you at 9:50. Kit Kat break. And we'll start off with the next chapter. Be right back. So it's a 13 minutes break, but yes, I think we deserve it, right? So please take that break and yeah. I had lunch, ma'am. Luncha. Now it's dinner time, no. Do not skip any topics. Semi-conservative mode of replication is important. Ask ChatGPT when you're not able to simplify it. You guys live in a tech era, right? Yes. Thank you so much, Sampath. Okay, guys, let's go ahead with the next chapter that is up there. I think everyone is almost up over here. Okay. Hello, Nitya. Hello. Okay. Perfect. So, starting off with the chapter Microbes in Human Welfare, you have the 40-second question right in front of you. 1 extra minute. It's dinner, yes. So, you had dinner. I didn't have my dinner. I just had water. I'll go home and I'll have my dinner. So, actually, over here, there is no one. They have arranged the cab and stuff like that. So, yes, there is time limitation. Okay, guys. Which of the following statements regarding flock is incorrect? Over here you have the options. They are the masses of bacteria associated with fungal filaments produced in aeration tanks. They consume major part of the organic matter found by useful anaerobic microbes. What do you think is the answer for this particular question? Come on. Hello, Munaza. Hello. Fatima says the answer is D. Okay. Strix, hello dear. Praveen Rich, okay. Lloyd has B. Others B. Praveen A. Mansi B. Sorry, Mansi D. Ravi Gamer also says it is D. What do you think is the answer for this question? Ishant says it is D. Okay. Fatima says it's D. Okay. Nitya D. Okay. Perfect. So your answer for this question is D. Why D? Listen to me. Flocks are being made up over here. In the aeration tank, what do you have? You add the organic matter which you want to break down, right? And flocks are being made up over here. Why? Because you provide lots of oxygen, and with the help of this oxygen, the aerobic bacteria which is there, it is going to divide itself, and plus it comes up with some fungal filaments. They both become friends, and that's what we call as flocks. And these flocks, they act on the organic matter and break it down. But the only thing is that you need what do you call air or oxygen, and over here this is aerobic microbes, not anaerobic microbes. Okay, it is going to be aerobic microbes. So that is why this is going to be wrong. And you guys were absolutely correct. Next question. Which of the following is not an advantage provided by mycorrhiza to the plants? What do you think about this particular question? Which of the following is not an advantage provided by mycorrhiza to the plants? Hello, Janu. You Vaimo. Ram. What language is that? Okay, guys. Which of the following is not an advantage provided by mycorrhiza to the plants? Resistance to root-borne pathogens, increase in the yield quality, tolerance to salinity and drought, overall increase in plant growth and development. What do you think is the answer for the question? Dheeraj, ma'am, thank you for teaching us. You're most welcome. Thank you for giving me the opportunity to teach, by the way. Nitya says the answer for this question is B. Fatima says B. Hassan says B. Mansi says B. Okay. Vinayak says B. Everyone says B. What is there in B? Let me see. So, resistance to root-borne pathogens. First of all, what do you mean by mycorrhiza? Mycorrhiza is where you have your roots, and these roots, my dear children, are associated with what? Fungus. They are associated with fungus. These fungus can go deep into the soil. Okay, what do? They go deep into the soil and they trap phosphorus. What do? They trap phosphorus from within, my dear children. So that is one very important thing. Apart from trapping phosphorus, they provide resistance. Resistance to what? To drought. So when there is too much amount of water, still the plant will be able to survive. Why? Because this guy is there. It says that, "Bacha, do not worry, I'll take care of everything." Okay. Next thing, it provides tolerance. Tolerance to what? Suppose there is too much amount of salt in the soil. Okay. Still the plant will be able to survive. Why? Because of this particular feature out there. Okay, so these are very unique things out there. Do not miss them by any chance. Yes? Okay. Now, overall increase in plant growth and development, that is also something that is provided. So, overall plant growth and development, that is also something provided. Now, B is what? Increase in yield quality. That is not something that fungi provide. So, it says that, fungi says that, "Macha, please take me away from this. This I cannot provide. I can give everything, but not this." So, yield is not being increased. Okay? Okay, guys? Alena, I see all my classmates are here too, ma'am. Is it? That's a good news, right? That's actually a good news, Macha. Okay. Let's go ahead then. Okay, the only request is classmates and all, you be here, you guys talk to me, okay? I am most lonely person over here in the studio and also probably in this building also. Okay, so if at all you want to talk to someone, it should be me, and please ask me doubts and let's cover everything. CS, Dharma, please my dear children, tomorrow morning, the first thing that you will get is CS video at 8 AM, that will be live. Okay, microbes in human welfare. First of all, only 2 and 3 marker questions are going to come from this particular chapter. Say, and also listen to me very carefully, that this is a chapter that can give you easy 7 marks, so do not miss out on this chapter. Okay, perfect. Now here in 2 marker, most probably sewage treatment and biofertilizer related questions have more chances, but that does not mean that other things are not there, right? So other things could also be there, and over here I have put 2 questions which are something new that I have not discussed any time before, so I'm putting that over here. Okay, so yes, first is biocontrol of insects and pests is desirable and beneficial compared to the use of insecticides and pesticides. Justify this with 2 statements. So listen, we say that biocontrol agents are good. When we put pesticide, what is going to happen? Basically the chemicals are going to deep into the soil, right? When you put insecticide, insects are going to die, but we will also die someday because of the insecticide, because there's too much amount of chemicals, right? Now when you use biocontrol agents, these organisms are going to eat up the insects, so we do not need any pesticides and stuff like that. Now you have been asked about 2 good reasons for replacing them. The 2 good reasons for replacing is that first is eco-friendly, biological control uses natural predators, parasite, does not pollute the environment. Yes. Now the next thing that is there, safe for non-target organism. For example, the insecticides, when we put, it is going to kill lot of other organisms, but over here when we are going to use biocontrol agents, they are only going to kill those organisms which has to be killed. So it is safe for all the other non-target organisms. Okay, so it's only if there is chunnu, munnu and tunnu also, it's chunnu only that was going to be targeted, not munnu and tunnu. So yes, ma'am, will you do CS? If I do CS, you will not pass CS. That is why I will not do CS. Ma'am is there, ma'am will be doing CS. Okay, next thing that is there, and that is the next question that we have over here is, list any 2 bioactive molecules of fungal origin and explain how those molecules help restoring good health in human beings. You are being asked about what? You are being asked about fungal origin bioactive agents. So bioactive agents, ma'am, I know 3 of them. One is cyclosporin, one is statin, and the other one that I have is, ma'am, streptokinase. Ma'am, but I do not know which is fungal origin. That is where most of you lose your marks, so please know that. Okay, please know that what is the ones which are of fungal origin. Ma'am, you're the best teacher. Thank you, champ. Okay, thank you, Janu. Okay, listen to me. Cyclosporin A is produced by Trichoderma polysporum. Trichoderma polysporum is what? A fungus. So that is one. Okay. Now statin, which is there, it is produced by Monascus purpuris. Monascus purpuris is also a fungus. So these are the 2 things that you can write. Okay. So please write cyclosporin A produced by Trichoderma polysporum. It is an immunosuppressive agent, and basically it is what? Produced by a fungus. Similarly over here also you have this. Okay. Got it? Ma'am, you can do the class tomorrow. Now go home, ma'am. It's too... Okay. Okay, I will be going at 11 o'clock as sharp 11 I'll be going. Okay. Now coming back right to this particular thing. In 3 marker, there is something else that is like the unusual thing which are being there. I am talking about that over here. So please listen to me, okay? Substantiate the following. Methanogens are found in the rumen of the cattle. Why do you think, Papa, methanogens are there in the rumen of the cattle? What are they doing up there? So one very important thing that they are doing up there is basically they are going to help in the digestion of cellulose. You know, plants, cows which are there, they are going to eat plant. And plant which is there, it will be having so much amount of what, Baba? So much amount of what? Any idea? Cellulose in their cell wall. You need someone to break it down. There is someone to break it down. The thing is that you need some help, and that help is provided by the methanogens out there. Okay, so methanogens which are there, they do it. Manish Kavi, please ask me, dear. Okay, yes. Yes. So over here, first thing, methanogens are found in the rumen of the cattle. First thing, methanogens are anaerobic archaeobacteria. They are anaerobic in nature. I hope you guys know that. Yes. Now, they break down cellulose and produce methane, helping in digestion of the plant material. So basically, they are the ones that is going to break down cellulose, helping in the breakdown of cellulose. They do not directly break down the cellulose. Please know about that. Okay, okay. Why are you looking so pretty, ma'am? Thank you. Beauty is in the eye of the beholder, is what is said. Yes, yes. Next question. Nuclear polyhedroviruses are species specific. Some people might be thinking, where is this thing written? Nuclear polyhedroviruses, where is it written? Listen to me very carefully. In the biocontrol organism, you know, there is baculoviruses. Viruses can also act like biocontrol agents. They can also kill insects, right? Similarly over here, baculoviruses are coming under nuclear polyhedroviruses. And they're species specific, as in they kill only certain types of species. They will not kill around all the organisms that they see. So please make sure you know that as well. Okay? So over here, nuclear polyhedroviruses are species specific. NPV infect only specific insect species and do not harm the other organism. Therefore, they are safe biocontrol agents. Done, done, done. Okay, now one more thing. BOD is an index of water pollution. BOD is what, Baba? BOD is, Baba, Shobha, I will be giving you the answer. Ravi, I will be giving you the answer. Okay, just wait for a second. Okay, BOD is an indicator of water pollution. How can you say that, ma'am? Very simple thing. Everyone is listening, by the way. Guys, listen to me. Very, very important thing. Okay, you might lose your marks. Flux and BOD is super important thing over here. So BOD means this organic, suppose there is aerobic bacteria. Its name is Chunu. Okay, Chunu is the aerobic bacteria that we are having over here. And this aerobic bacteria, what does it do? It pairs up with some fungal filaments, I told you previously. And what do you call this now? You will call this as flux. Perfect. Why are they pairing up? Because it wants to break down something. What is that something? The organic matter. It wants to break down the organic matter. That is the base storyline, right? Yes. Now, when Chunu wants to do it, it says that I need oxygen. Without oxygen, I am not doing it. So, Chunu needs oxygen. Oxygen is given to it. Oxygen is given to it. Why? To break down organic matter. Now, you only think, if organic matter is way too much in amount, don't you think Chunu will need too much amount of oxygen? Because obviously, for breaking down, it needs oxygen, right? It's an aerobic bacteria. So, demand for oxygen is what? Leveling up. It is increasing. What do you call this demand as? This demand is known as biochemical oxygen demand. What do you call this? Biochemical oxygen demand. Very, very, very important. Right? Now you think, if organic matter is less, Chunu will not need too much amount of oxygen to break it down, right? That means in that particular case, when organic matter is less, that means pollution, when it is less, obviously the level of BOD, biochemical oxygen demand, also goes less. If pollution is more, biochemical oxygen demand automatically increases. Got it? Did you get it? Guys, if at all you have got it, just give me a quick thumbs up. Everyone out there. Ma'am, question bank in the yetto MCQ. So listen to me very carefully. We do not know if at all the questions will be repeated from the question bank or not, but we still have to practice it. Okay, for understanding the concepts. Okay, ma'am, solve our doubts. Okay, we are coming to that. Hi. Okay. Ravi was asking me a question. I'm not able to find Ravi's question because you guys were talking so much. Ravi, once more, macha. Give me the question. Okay. Perfect. Nice explanation, ma'am. Thank you, champ. Okay. Hello, Prithvi. Okay. So, my dear children, apart from these questions, there are new questions that I've added up. I've explained them as well. Now, yes, ask me the doubt, Fatima. Ma'am, fixed question are given in the channel. Go watch it. Ma'am, what is your name? My name is Jasmine. Okay. Okay. Macha is personal. Yes. Okay. Fine. Now, mention the uses of following bioactive agents. Now, this is super important question. Why? What is BOD? Biochemical oxygen demand. I just discussed that, right? Yes. Ma'am, I didn't understand molecular basis of inheritance. I'll request you to watch the masterstroke video. It will definitely help you out. It's a one-shot video, okay? It will help you out. Go watch it. Okay, now listen to me. Streptokinase, what is the use of that? You call this as bioactive agent, right? What does it do? So listen. Streptokinase is a clot buster. For example, someone is suffering from myocardial infarction that is leading to heart attack. At this particular point of time, there are clots being formed. And when you have clots being formed, obviously you need something to dissolve it. Otherwise, what is going to happen? Basically, these clots can travel through your head also, or else basically it does not allow the transfer of oxygen, everything out there, right? Listen to me carefully. So what do you need? You need clot busters. Streptokinase is going to be a clot buster, and over here you get it from what? You get it from Streptococcus, which is a bacteria. Streptococcus, which is a bacteria. The second thing that you are having over here, ma'am, tell me which organism that helps in nitrogen fixation. We'll coming to that only, Baba. We'll talk about nitrogen fixation completely. Okay? Yes. Next thing, cyclosporin A, which is there. Cyclosporin A, what does it do? It is an immunosuppressive agent. For example, I asked Ravi its kidney. Okay. Ravi gave me the kidney, but the problem is that my body rejected it. Why? Because it's a foreign thing. At this particular point of time, I needed kidney, but my body rejected it. So this, what will I do in this condition? I will take immunosuppressive agent to suppress my immune system such that the organ transplantation process is super clear. Okay, so cyclosporin A is an immunosuppressive agent, and basically this is very useful during organ transplantation. This is obtained from Trichoderma polysporum. Trichoderma polysporum. Super important. Do not forget it. Okay. Fine. Now, going ahead, what do we have? We have statin. What do we have? We have statin. So statin is what? Statin is something that is, it is a fungus, by the way, Trichoderma polysporum. And also, statin is something that stabilizes your blood cholesterol level. I remember it like that, but you can remember, statin is something that lowers the blood cholesterol level. Stabilizing, lowering, okay? How does it do that? Basically, the statin competes with the actual thing. For example, you have an enzyme over here, okay? And this enzyme is responsible for production of cholesterol, yeah. Usually when the substrate comes in, that's how cholesterol is being formed with the help of this enzyme. At the same binding position of the substrate, your statin will come and bind. What is going to happen? The substrate which is coming like, hey, Macha, you side, ma'am, I have to go and attach over there. So anyway, it won't be moving aside, which is exactly why how it is going to inhibit the action of, or basically synthesis of, cholesterol out there. So statin is what? Blood cholesterol lowering agent. Where do you get it from? That is important. You get it from Monascus purpureus. Monascus purpureus, which is also a fungi. It's a fungi. Okay? I have a gut feeling that this question will repeat again. Even I have that somehow, or probably something related to this. So make sure you practice it. Okay? Okay. Going ahead with the next thing that we have, mentioned the product and uses of the following. Children, if it is clear to you, just can you give me a thumbs up, quick thumbs up, will really work. Will be sweet of you to give me a quick thumbs up, because I really want to know if at all you're still connected, or are you guys just talking among themselves. Hey, what is happening over here? Janu, are you married? Not liking it. Children, Janu and the marriage and everything. Listen to me very carefully. We'll talk about it later on. Okay, this is not the right app for that. You should understand it. Aren't you Gen Z kids? Okay. Okay, perfect. Now let's go ahead with the next thing that is there. And it goes like that. Acid produced by Aspergillus niger. Aspergillus niger produces which acid? Aspergillus niger produces very important acid which is known as citric acid. Citric acid. Aspergillus niger is a fungus. Please do not forget it. It is producing what? Citric acid. Very important thing. The next thing that you have is enzyme produced by Streptococcus. Streptococcus produces which enzyme, Baba? Streptococcus produces only one enzyme, and that is what? Streptokinase. Okay, it is what? Streptokinase. Okay. Bioactive molecule produced by Trichoderma polysporum. We just studied it, Macha. What was it? Trichoderma polysporum produces what? It produces only one thing, cyclosporin A. Okay, it is going to be cyclosporin A. It's repetition of content, but yes, these type of questions can also be coming in. Okay, got it? Super important. Fine. Yes. Children, this question clear? If clear, give me a thumbs up. Fast. Come on, we are reaching up 11 o'clock. Before that, if at all we can. Some people are planning their meetup over here. Okay. What do you think I am? Scarecrow sitting in the field? Okay. Okay, macha. How many marks will I get from model question paper? Dear, I cannot deduce this data. Listen to me, no one knows how many marks you're going to get from model question paper, but you still have to practice it. Okay? Okay. Okay. Listen to me. Type of biofertilizers out there. This is something Ravi was asking me, okay, the fixing bacteria and stuff like that, nitrogen fixing bacteria. Here, a basic funda is being applied. Listen, bacteria, cyanobacteria, two different things are there. Bacteria, cyanobacteria, these are two different things. Very, very, very important thing. Okay. Now, over here, bacteria which is there, it is not going to prepare, not all bacteria are going to prepare its own food, but cyanobacteria are preparing its own food. That is why you call them as cyanobacteria. They are also known as blue-green algae. Fine? Yes. Now, under nitrogen fixing plain bacteria, you have three guys. One is rhizobium, the other one is azospirillum, the other one is azotobacter. Okay, the rhizobium which is there, it is there in the root nodules of the legumes. For example, you know, pea plant and stuff like that. So there are root nodules of the legumes. In that, you would be having rhizobium. What do they do there? They will be chilling and also they will be fixing up nitrogen. You know, nitrogen is found in the nature in the form of N2, that is nitrogen gas, right? And this has triple bond. It's very difficult for the plant to absorb it, which is why some organisms will take it, break it, make it something else and give it to the plant. You got it? Yes. So one such organism is what? Rhizobium, and that is known as fixing. Now the next thing that we have over here is what? Azospirillum and azotobacter. Listen, this guy was sitting or basically was staying in the root nodules of the legumes. But azospirillum, azotobacter, they are free living. They are not dependent on anything else like that. They are free living bacteria. They are chilling here and there, but they also enrich the soil by what? Fixing nitrogen out there. That is different thing about azospirillum and azotobacter. They are coming under plain bacteria only, but they are free living bacteria. They are not associated anywhere. Now what do you have? You have one more guy, cyanobacteria. This is important. In cyanobacteria, I always tell you, remember it like this. Uno, you might have played Uno. Similarly, over here, you have Anno. Anno stands for what? Annabena nostoc oscillatoria. Annabena nostoc oscillatoria. These are other things that is out there, right? Over here, what do? They prepare their own food because they are cyanobacteria. Apart from that, my dear children, they are the one that fixes nitrogen. Okay, obviously that is why we call them as biofertilizers. And they are very much useful in the paddy fields. They are very much useful in what? The paddy fields out there. Okay, done. That's all about cyanobacteria. Please remember the name Anno. That is super important. They come under cyanobacteria. Now we have one more guy, that is mycorrhizal fungi, and we spoke about it already, right? I told you that over here the roots of the plant as well as the fungus are grouped up together. The fungus can go deep into the soil and give what? Phosphorus. Do not forget it. Phosphorus, not potassium. It is phosphorus. Okay, it gets phosphorus. And also remember one thing, that this fungus which is there, it belongs to the genus Glomus. Okay, Glomus. And what does it do? It provides resistance, it provides tolerance to what? To all this drought and things like that condition. And finally, it helps in total growth and development of the plant. Fine. Apart from the phosphorus thingy, three other things are there. And I hope that's clear. So these are the type of biofertilizers that you need to remember. Store it in your brain right away. If this is clear, give me a thumbs up. If this is clear, give me a thumbs up. Nice name, ma'am. You're looking like a jasmine flower. Thank you, Macha. Okay. Guys, is it clear? If clear, can you just give me quickly a thumbs up? Ma'am, what is your name? My name is Jasmine. Ma'am, great applause for you, ma'am. Having such a day, standing for more than 6 hours for us. You're most welcome, champ. And thank you guys for giving me the thumbs up. I hope it makes sense. Let's go ahead. Okay. Next thing is explain the stages of sewage treatment. How many questions do we have then? Okay, sewage treatment. Fine. Now, Rohan, you didn't understand it? Do you have any doubts? You can ask me one. Mani, I guess she was named Jasmine because she's pretty as one. Okay, guys, listen to me. Sewage treatment. If at all you listen to me over here, any question related to sewage treatment will be like chuck in your brain. So listen to me carefully. First of all, we have to make one very important thing. But since I have the nature to basically mess around. Okay, so we'll start with this. First thing that we have is a settling tank. This name of the tank is also important. I didn't know that. Okay, settling tank. Now what happens in settling tank? This is going to be known as the primary treatment. This is going to be known as the primary treatment. What exactly happens over here? Things that are going to float around are going to be removed with the help of filtration. This is the first step of primary treatment, filtration. The next step that we have over here is what? Is sedimentation. So whatever is heavy and things like that, those things are going to be removed via sedimentation. That is the next process that is there. Always remember, primary treatment is a physical process. Okay, it is going to be a physical process. Ma'am, is azotobacter also autotrophic? No, it is not. That is why we put it in plain bacteria. Okay, coming back. Yes, where were we? Just a second. Yes, we were here. So whatever is left behind over here is what is known as the primary effluent. What do you call it? Primary effluent. And this is what we are going to treat. When I say primary effluent, that means lots of organic matter is over here. Okay, there will be lot of organic matter up over here. Now, how will you treat that? We are going to put it in the next tank. And this tank is a super important tank. And a very cutie tank out there. You call this as aerobic. Sorry. You call this as aeration tank. What do you call this? Aeration tank. Now, what happens in aeration tank, my dear children? The organic matter is being put, right? And over here, we are going to provide a lot of oxygen. Why? Because our main guys are going to increase up over here. What are our main guys over here? Aerobic bacteria. They literally increase way too much over here, and they group up with some fungal filaments out there, giving rise to what? They make up the flocks. And the flocks work on the organic matter utilizing the oxygen, and this is when we say that basically what is happening, the biochemical oxygen demand is going to go on really high. Why it is demand is high? Because over here there's too much amount of organic matter. So we will work until the demand goes up very less. If the demand is less, that means there is no too much amount of organic matter that is left to be broken down. Done? Done. Going ahead. Now what do we have? You have done everything. You take it to the next tank, which is known as, again, the settling tank. Again, the settling tank. What do you want to settle over here? You want to settle all the flocks down. So whatever flock content is there is being settled down. Now whatever is being cleared is being sent to what? To the river, lake and stuff like that. What is being settled over here? You said that this is basically what? The flocks. But here you have a different name altogether. You will now call this as what? The activated sludge. You will now call this as what? The activated sludge. This is super important. Okay, activated sludge. Now what happens? This activated sludge which is there, you take some portion of it, and you give it back over here. Why? It acts like an inoculum. So the next thing comes up for treatment, basically you get some amount of aerobic bacteria, which is why we say it acts like inoculum. This settling tank name is important, do not get it wrong. Now, going ahead, what do we do with the activated sludge? This portion is given to the river. No, what do we do with the activated sludge? Now comes the last one, and that is known as anaerobic sludge digester. Anaerobic sludge digester. That is the last portion up over here. What has been happening in this particular condition? You put the activated sludge. This is nothing but the aerobic bacteria as well as the fungal filaments, right? And now basically you are going to treat them with the last guys, that is anaerobic bacteria, the ones that does not need oxygen, and that is why you call this anaerobic sludge digester. Okay? Activated sludge question in the question bank is confusing. I shall pause the question. Yes, you can, please. Okay, go ahead. Okay, so basically anaerobic bacteria over here is going to act on the activated sludge and is going to produce what? It is going to produce biogas. biogas. And this biogas is what? It's a combination of what things? It is a combination of methane, that is known as CH4. It is a combination of H2S, hydrogen sulfide, and it's a combination of CO2, that is carbon dioxide. Biogas is not just methane, but all these three gases come up together. Done. Okay. Where is the question, dear? Ma'am, which all scientists' name should we remember? Most of the scientists are there in Principles of Inheritance and Variation, their name, and also Molecular Basis of Inheritance. All the important names, very few names are there, right? All of their names are important, yes. What is happening up here? Guys, is, okay. Totally confident by biology. Thank you, Manjunath. Love you, ma'am. Love you too, guys. Is harmful for the river? No, dear, it is not. Okay. Guys. Okay, fine. Then I go ahead then. Ma'am, are we done? We are not done. We are done with this chapter though, is what I believe. But please go ahead, children, with all the other questions as well from the same chapter itself. It's super important for you to do that. Shobha, thank you so much, dear. As in biocontrol agents, all the examples of biocontrol agents, the things that you use in house distillation and everything. Okay? Cool. Now, just a second, I've got a quick message out there. Okay. Ma'am, model, Tarun Reddy, ma'am, briefly explain the reason for conserving biodiversity from narrowly utilitarian point of view. I have a question on that. I can explain to you directly. Okay. Okay. Since, children, we are ending up this chapter. Okay, fine. I have a question to you guys. Shall we, since anyway I will not be able to cover the entire thing, that is 2 chapters, shall we directly go to what do you call organisms and population, if at all you do not mind that, and the last chapters, if at all it's fine. Activated slug in settlement tanks of STP is a rich source of aerobic bacteria. This statement is correct. No, settlement tanks. Okay. Of STP is rich source of aerobic bacteria. If it is aerobic bacteria, then it should be correct, and it's settling tank actually. Okay, it is settling tank. Ma'am, I'm getting neglected. No, you're not. You're not, dear. Because no, there's a question on that. Yes. So, perfectly. So, we'll go ahead with organism and population. And I think before organism and population, we should go ahead with one very important concept that Tarun has been asking. I'll just give him the explanation, okay? Just give me a second. Where is that? I gave it. Narrowly utilitarian point of view. I have written it somewhere, it seems. Okay, fine. I'll just tell then if it is not there. I do not want to waste your time as well. Okay. By the way, guys, please go ahead with this particular question. And Tarun, listen to me carefully, okay? You can write that whenever you conserve organisms, I remember where I have put it. Just a second. In MCQs. No? In MCQs, didn't I put it? No, I didn't. Where are the MCQs though? Here. Yes, I did it. Okay, so listen to me carefully. Narrowly utilitarian, you can write this. Humans obtain many direct economic benefits. Okay, economic benefits from the biodiversity such as food, fiber, fuel, medicines, industrial products, etc. Many modern drugs and traditional medicines are also delivered. So basically, narrowly utilitarian point of view means you are getting direct benefits in the terms of products, and that is why you are safeguarding the organisms out there, or probably the region out there. Then you have what broadly utilitarian point of view, according to which it says that we should be conserving the nature for the services that the nature is providing. For example, oxygen production, For example, the aesthetic value, when you go sit somewhere else, you feel so nice, right? So this is the ecological service that is providing you. Okay, obviously you cannot get direct benefits from it, or probably you can, but not exactly in the form of products. Okay, not exactly in the form of products. Now, ethical arguments is that basically there's an intrinsic value. It has a right for everyone to live on this particular planet. That is why we should be basically safeguarding all the organisms out there. Got it? Done? Yes. Fine. Now, yes, now we'll go back to organism population. The reason I want you guys to do that as well is that there is a 5-marker question that you can get from this chapter, and you should not be missing out on it. Yeah. So chuma I'll just go ahead fast, because you know what, ma'am, I'm coming. Are you coming live tomorrow too? Yes, I'll be coming, but yes, so there can be changes in the time. Biotech, please listen, Vinay. The thing about biotech is that it could take a lot of your time. Okay, so I'll cover these chapters. Okay, and it can give you an option for the 5-marker. Yeah, let's do it. And if at all it's possible, I'll take biotech also. So there's a cab that is being booked by the office. So if it comes fast, I would have to go. Okay. Yes. Perfect. Okay. Fill in the tabular column with appropriate describing biological interaction among species. My dear children, over here, you have phytophagous insects and species B. This shows name of the interaction is predation. What is this particular species B? Any idea, guys? This is important. It can give you a 5-marker question. Do not miss on it. Any idea? Any idea? Any idea? Guys, Nitya, Janu and all, please talk to me. Do not talk among themselves. Give me the answer. What do you think is the answer for this particular question? Yeah, from biotech it's also there, but it's easier to grab those marks over here. Okay, zooplankton. Listen, frog. My God, dear children, listen. Phytophagous insects are there. Phytophagous means plant-eating insects are there. So over here, these insects feed on what? Which acts like predation. They are going to feed on the plants. No, they're going to feed on the plants. Yes, phytophagous, that is known as predation. This is bichara the prey, and this is the predator. Okay, very, very important thing. Fine. Now going ahead. Ma'am, can you take biotech in tomorrow's class? I shall. I'll definitely do that. I'll not leave the chapter by any chance. Okay? Cool? Works? Fine. Let's go ahead then. Balanus and thalamus. What do they show? Balanus and thalamus, if at all you remember, they are there on the seashore, right? First of all, you had thalamus over here. Balanus was the competitive barnacle, okay, which was competitive in nature, which is why thalamus has to leave the intertidal zone and has to move up. Had to move up. Okay, so this is an example of competitive release where competition is happening, which is why the guy moved up, and over here it has an entire region for the thalamus to sustain. Sorry, the balanus to sustain. What do you call this? Is competition. Exactly. This is going to be competition. Okay, Vinayak, and Vinayak, please do not be sad. I'll try. Okay. I'll definitely cover it up tomorrow, if not today. Now, marine fish and copypods are there. Name of the interaction which is there. Marine fish on the copypods. Listen, copypods are something that acts like the lice on our hair or a head, or basically ticks on the dog's body. Over here, the copypods are parasites on marine fishes. So this is a parasitic adaptation that is there. Okay, this is what? A very small aquatic. Yes, Mashali Khan, exactly. Okay, done. So this is what? Parasitism. So we'll write parasitism. Now, next thing, dash orchid commensalism. Dash is there, orchid is there, commensalism there. If at all you have studied on a mango tree, orchid grows. Why? Because it wants any benefit from it? No, the only thing is that orchid wants enough amount of sunlight. Orchid wants enough amount of sunlight. That is why orchid is basically growing on the mango tree. It is not there to harm the mango tree out there. Mango tree is neither getting harmed nor getting basically benefited. So I will say over here that is commensalism. Okay, so this is mango tree. Who is getting the benefit? Only one guy, and that is orchid. Now fig is there, dash is there, and over here you have mutualism. This is the last one, and give me the answer for this particular question from this particular thing. Epiphyte on mango, exactly, Harish. Okay, exactly. Now last one, fig and dash and mutualism. What do you think is the answer for this? Guys, Syed Amin, Vinayak, everyone is doing super awesome, okay? Isham, dear, monarch butterfly, I think. Which one are you answering? Wasp. Rihan says wasp. Exactly. Shashank also says wasp. Exactly. So fig and wasp, my dear children, they show mutualistic relationship. Both of them are going to help each other. So this is absolutely correct. Sayada also, Uma Maheshwari also show, yeah, bee is also an insect, but wasp is the actual answer. They show mutualism, okay? Wasp is the one that is the actual answer. Perfect. Jia, Harish, GM, Veena, Mansi, Sanjan. I see some people from the starting onwards. Awesome. You guys are awesome. Okay. And Manjunath and everyone who are talking amongst each other, please, my dear children, stop that. Okay, fine. Now write the equation to calculate the population density at time t. How do you write it? Population density at time t is denoted by NT. Okay, NT is equals to what? N naught. N naught means initial population. Plus what? Plus birth plus immigration. Birth increases the population. Immigration also increases the population. Exactly. Right? Yes. So over here, just a second. Guys, just a second, okay? Okay, perfect. Do not worry, it's not your generation, it's some spammer. Okay, we know how to deal with the spammer, especially being a biology teacher. Yes. So going ahead, what do we have over here? Okay, B and I, that is birth rate as well as immigration, is going to increase the population. Now you have minus. Minus means it is going to delete some population also. How? By death and emigration. So death and emigration are limiting factors. So we have N naught plus B plus I minus D plus E. That is one very important thing out there. And yes, you have all the other information up over here. Now what are the very important things? Okay, the question is, what are the what you call basic processes which are there that is going to affect the population density? Natality. Natality means what? The birth rate. Okay, and then what do you have? Mortality, that is death rate. Immigration, that means entry, number of individuals getting entry into the population. Immigration means number of individuals leaving a population. These are the few things that you have to go ahead with. Yes, works. Fine. Now, what is mutualism? Explain any 4 examples of mutualism. So, mutualism related questions can be asked. And so, yes. What do you think is the answer for this particular question? Examples for mutualism, you have lichen, you have mycorrhiza, you have rhizobium, and fig tree and wasp. Fig tree and wasp we saw. Listen, fig tree basically wants pollination from the wasp. Wasp is going to lay its egg inside the ovary of the fig tree. So that is how they do. That is an example of what? Mutualism. Right? Yes. Now the next number that we have over here is rhizobium bacteria and leguminous plants. Again, rhizobium which is there, it is going to be in the root nodules of the legumes. Rhizobium, what does it do? It fixes nitrogen and gives it to the plant. Okay, what about the plant? Plant will provide, obviously plant is going to do photosynthesis and will provide organic material out there. Okay, so these are something that is out there. And the plant basically provides nutrients and the shelter. Yes. Okay. Niche means specific what do you call, area, or the specific characters out there. That is known as niche. Okay? Yes. Now we go ahead, we have okay, mycorrhiza. Mycorrhiza, I told you, it's fungus as well as the root of the plants. The fungus over here provides what? Provides the phosphorus and overall growth and development and everything out there. Whereas the root of the plant, again, it's the root of the plant. Plant can always do photosynthesis. So it does that and provides everything out there. Yes. And finally, you have lichen. Lichen is basically a combination of algae and a fungus. So instead of having a plant over here, you have an algae, and that's the only difference between lichen as well as mycorrhiza. So over here, again, the algae provides what? Food with the help of photosynthesis, and fungus basically provides protection out there. Okay? Guys, done? Clear, clear, clear. If it's clear, just can you give me a thumbs up? Will be really, really good if at all you do that. Thanks for the help, Chetan. You're most welcome, champ. Tarun Reddy, ma'am, are you still in DVLC? How do you know that I'm in DVLC? Yes, I am indeed, we'll see. Okay, so yes. Okay, now, yes, this is one very important question and I want you guys to solve it. So listen to me carefully. There are two types of graphs. You have to listen to me carefully. This is super important. Soundarya ma'am, it's 6 hours already. Thank you so much. You're most welcome. So over here, one graph is for, over here, what do you have? You have population density on this particular side and you have time at this particular side, right? Yes. So what do we have over here is that there are some cases where you do not have any limiting factors, okay? You do not have any limiting factors and they grow like this. So there is no capacity over here, no carrying capacity, it just grows. What do you call this type of growth as? You call this growth as exponential growth. What do you call this? Exponential growth, and over here you get a J-shaped curve. Done, then. Okay, now listen to me. Ankit sir is there, going strong, ma'am. Thank you, sir. Yes. Perfect. Now listen to me. The next thing that we have over here is the next one, which is known as logistic growth. Basically, logical sort of growth. Logically, if at all you think, you have an area. Do you think the everything grows exponentially? No. If at all we have a breadcrumb and probably from tomorrow, if we do not have any food, we are going to fight like animals for that, right? Exactly. So there is a limiting factor being involved in natural cases, and that is known as the carrying capacity. It is known as what? The carrying capacity, and it is denoted by the term K over here. Now, when I say carrying capacity, you need to understand that organisms which are growing, they will be growing like this. They can grow only until here, and after that, they will reach a phase of stability. Yes, so you see a S-shaped curve over here, and this is known as logistic growth. This is known as logistic growth. So in nature, what do you find usually? Logistic growth. Now, there's a formula that you need to remember. When it goes like this, exponential growth, when you have something like this, you have one very important thing out there. The formula for this is denoted by dN by dt, that is, the change in the population density is denoted by is equals to Rn. R stands for reproductive capacity actually. R is basically B minus D, birth minus death. So R, according to Darwin, he says that it is reproductive fitness. The organisms who produce more, basically they will be having more value for R. Now, apart from that, you also have, as I said, in logistic growth, the formula is going to be kind of different. Why? Because you are including carrying capacity value K over here, right? And how do you put that up? dN by dt is equals to Rn remains the same. You just have to put up something like this, K minus N divided by K. K minus N divided by K. And done. Now, when I say that you have to solve this question, okay, the Deepthi have given me the answer, B logistic, realistic, D exponential. Oh my God. Okay, powerful. Just by model papers, you will not get out, dear. Okay. Perfect. Now listen to me carefully, okay? Over here, curve A represents which type of growth? You guys tell me, curve A represents which type of growth? Everyone who is out there, guys, probably I'll be ending up the live really quick at what do you call, just a second, okay? Messages. Okay. R is intrinsic rate of natural increase. Yes, yeah. It, and according to Darwin, you call it as what do you call it? Reproductive success or fitness. Okay. Okay. A is what? A is what? Guys. A is what? Exponential growth. A is exponential growth. Yes, A is exponential growth. Fine. What is curve B? What does curve B signify over here? Curve B is what? Curve B is what? Curve B is also exponential growth, is it? A is exponential, yes, Alaina Saida, Mansi also. What about B? B is logistic. Yes, B is going to be the logistic growth out there because you see a S-shaped curve. Clearly you see an S-shaped curve. Perfect. Now coming over here, which growth model is considered more realistic in nature? Which one? A, B, you answer in C, okay? You answer in C. Which one is going to be considered more realistic in nature? I'm talking about C. C. Hello, Kelly Sethi. Hello. Logistic is going to be considered as more realistic form, right? Because you have carrying capacity every single time. Now, when bacteria are cultured under controlled optimum environmental condition, which curve is going to be obtained? Ma'am, A is J-shaped, right? Yes, it is J-shaped curve and the type of growth is exponential growth. Yes. C is logistic. Okay, fine, guys. I'm talking about this one. D. When bacteria are cultured under controlled optimum environmental condition, which curve is going to be obtained? Controlled optimum environmental conditions. D is exponential. Yes, D is actually exponential, Alana. Perfect. D is actually exponential. Why exponential? Listen to me carefully. Here it is written controlled. When you hear the term control, you feel it is logistic. But here it is also mentioned optimum. Optimum means everything necessary, everything important is already provided. So controlled optimum environment means, okay, so basically some culture is happening over here. Whatever is not required is being removed. Whatever is required, it is being given also. That is what we mean by controlled optimum condition. So in this case, it is going to show exponential growth. It is actually going to show exponential growth. And I remember in preparatory, you got one particular question where what was happening is that it was mentioned ideal environmental conditions are being provided. If ideal environmental condition is being provided, please understand it is going to be exponential growth. If they are mentioning ideal environmental condition is provided but with limiting resources, the term limiting comes, that means it is there is carrying capacity involved over there. In that case, it will turn into logistic. If there is no limiting term, okay, nothing is being limiting over here, please understand it will be what you call exponential. Now the interactions such as predation, competition, parasitism can be represented by which type of curve? The interaction such as predation, competition and parasitism can be represented by which type of curve? Any idea up there? Any idea for this particular thing? E? Logistic? Okay, Akhil says logistics. Okay. Logistic as resources are limited. Exactly. Hello, Kali and Deepti, Elena, Shobha, Mansi. Yes, it is going to be logistic. Why? Predation, competition, everything is going to make sure that everything is going to be limited. That's the reason. Perfect. By the way, everyone who is watching this video, I hope you have subscribed to the channel. If not, subscribe to it right now and also click on the like button of this particular video. Yes. And with that, I think we have completed this particular thing also from organisms and population. Obviously, you have to go ahead, look at for all the important things. This is something that I could brief and put it up. Okay. Now, my dear children. Okay. It's Janu. Janu, dear. I remember you, Janu. Let's not talk to each other there. Very precious time. Literally very precious. Okay. Guys, identify the decomposers among the following in the pond ecosystem. In the pond ecosystem, which are the decomposers over here? Guys, come on. Bacteria, fungi, flagellates, phytoplankton. Who are the decomposers in the pond ecosystem? Man, quickly tell what's competitive release. Competitive release basically means that when the competing organism is going to move away, the inferior one, when it moves away, the superior one gets more space to get, all the resources out there. That is known as competitive release. Okay. 16C is the answer. C is what? A, B and C. A, B and C as in bacteria, fungi and flagellates. Are they the decomposers in the pond ecosystem? Obviously, yes. They are the decomposers in the pond ecosystem. I remember fungi and bacteria like FB, Facebook. And apart from that, now you have flagellates also in the case of a pond ecosystem. Okay, done? Done. The next thing that we have is productivity available as biomass for consumption of heterotrophs is. The productivity which is available as biomass for the consumption of heterotroph is what? Guys, you are giving the correct answer. I am seeing that. Okay, perfect. And those of you getting things wrong, make sure you're getting it corrected as well. Ma'am, how to score out of out? Make sure you watch this video. Make sure you solve the questions which I have shown over here as well. Yeah, that's the only way we can get out of out. B and A is what you're saying. Some people are saying B. Okay, Fark Anudin, okay, is saying B. Akhil says A. Nagaraj says A. Okay. 67A, GPP. Guys, how can it be A? You only think, how can it be A? Available as biomass for consumption of heterotrophs. Listen to me carefully. Suppose I have over here the producers. Then you have the primary consumer. Then you have what? The secondary consumer, right? The productivity of all the producers put together, listen to me carefully, very important. The productivity of all the producers put together is known as GPP. When I say about one producer, I call this as primary productivity. When I talk about lots of producers together, I call it gross primary productivity. Done? Done. Now, do you think everything is available for the heterotrophs? Heterotrophs means the organisms that feed on other organisms for food. Primary consumer is a heterotroph. Right? Do you think every entire GPP is available for them? No. GPP minus R. R means respiratory losses. All the loss for the metabolic activities is done, removed. And now what do you get? Then you get NPP, net primary productivity. Net primary productivity is something that is available for the next group of organisms. Got it? So your answer for this question is going to be NPP. Do not go and write GPP and come. Is this understood to everyone out there? Okay. Is this understood? B is the answer. Okay, it is NPP. I hope you have understood the concept. Do not mug it up. Now, if at all they are asking you what is the productivity of a producer, it is primary productivity. Okay, if they are asking you what is the total gross productivity of the entire producers, it is GPP out there. You get it? Yes. Thank you, thank you. Okay, the annual net primary productivity of oceans is about. The annual net primary productivity of oceans is about what? Bharat, thank you so much, dear. Okay, annual net primary productivity of the oceans is about BIA. 170. Yash says 170 billion tons. Exactly. It is 170 billion tons. And out of it, how much is taken by the oceans? Any idea? You know, oceans net primary productivity is less. Whereas when you talk about the land area, their net primary productivity is more. Okay, so please have that in your head. Oceans, it is less. Rest, everything. The terrestrial ecosystem, their net primary productivity is more than oceans. Even though oceans take up lots of a space, but it has less productivity out there. Okay, so it is 170 billion. Please remember it is billion, not million. Fine, perfect. Ayyo, why it is written 55? Sorry, it is this. Okay. Next one. Which of the following step in decomposition does not occur on the detritus? Which ones, okay, which of the following steps in decomposition does not occur on the detritus? Detritus means what? The dead and the decaying matter out there. And you have been asked which step is not directly happening on the detritus. Janu is gone, dear. Yes, 55 is for the ocean, exactly. Yeah, okay, fine. So, ma'am, do fast. Oh, I'm doing fast, dear. How fast can I go? See. Listen, Humification, is it humification? So some people are saying it does not directly occur on the detritus. Humification is a process, yes. So listen to me carefully. There is a question that is being asked, and just a second. And the question was like this. Yeah, no, where is it? Yeah. Describe the steps of decomposition that operate simultaneously on the detritus. So always remember, on the detritus it is fragmentation, it is leaching and catabolism. These are the direct things on the detritus. Now, other things happens mainly in the soil. It is not on the detritus, and that is humification and mineralization. Very, very, very important thing. Okay, very, very important thing. Make sure you do that. Ma'am, but you marked 170 for ocean. No, I marked, what's the question for oceans? Was the question for ocean? Oh, sorry. Sorry. I didn't read the question. Yes. 55 billion tons is for the ocean. Yeah. Total is 170 billion tons. Okay. So over here, yes. Guys, I'm so sorry. I read the question wrong. I said the correct thing, but yes. That ocean NPP question. Yes. Got it correct now. I hope it's clear. Yes, ma'am, can you get the PDF? Yes, you can. After the live is done, I'll be giving you the PDF. Okay, fine, guys. Fine, we'll come back to this only. The following step in decomposition that does not occur directly on the detritus. It is humification and also mineralization. Always remember that. Fine? Yep. Perfect. Now, 70th question. Dash is the amount of living biomass present in an ecosystem or the trophic levels at a given time, usually expressed as dry weight per unit area. Dash is the amount of living biomass. What do you call this term as? What do you call this term as? Dash is the amount of living biomass in an ecosystem or trophic level at a given time, usually expressed as dry weight and things like that. You might be asked to define it. That is why I'm telling it again and again. Okay? Any idea? Any idea what do you call this as? If at all we want to find the biomass of anything that is there, in the living thing that is there, at a given time. Standing crop. Exactly, Mansi. It is known as what? Standing crop. And everyone else also who gave me the answer. Please remember, this is an important term. You should be knowing this. Okay? Tarun, Yash, everyone. H.T. Raja. Okay? Now, the pyramid of C is generally inverted. Is that correct? Statement number 1. Pyramid of C is generally inverted. Statement number 2. The biomass of fishes far exceeds that of phytoplankton. So, in the case of pyramid of C, my dear children, it is totally ulta. Why? Why? Because there, the biomass of the producers which is there, that is phytoplanktons, it is very less. They are chunno organisms. They are very small organisms. Now onto them feeds what? Onto them feeds your zooplankton, okay, which have more biomass than phytoplanktons. Now over them you have small fishes which obviously have more biomass than zooplanktons, and then you have larger fishes, which is why you have an inverted pyramid of biomass. So pyramid of biomass in C is generally inverted. Obviously yes, this is correct statement. Everyone is saying exactly. Now the next thing, the biomass of fishes far exceeds that of phytoplankton. That is also super correct out there. Okay, done. Now ecosystem, you can get all these type of questions. I have a question over here which you need to practice extra. Schematically represent pyramid of biomass with appropriate units. So you have to make a pyramid of biomass with the units. So you should know how to make the pyramid of biomass. Write the units: dry weight, kilogram, m raised to the power -2. All the small things are super important. Okay, all the super important things are there. Ma'am, make a video for MCQs. I have done it. Listen to me. In the YouTube, go to the playlist section of this particular channel. You will see biology final examination. There you have all the MCQs in one video, all the 2 and 3 marker in one video, all the 5 marker in one video, all thing merged in complete biology video also. Watch it. Okay. Now, Okay, one more question is there. Describe the components of aquatic ecosystem, taking pond as an example. So what are the components of aquatic ecosystem? Abiotic components is water with dissolved inorganic and organic things, soil at the bottom, and factors like sunlight, temperature, everything. They are the abiotic components. Now biotic components, producers are what? Phytoplankton, algae and aquatic plants. These three things are there. Phytoplanktons, algae and aquatic plants, all of them are being there. Consumers are what? Zooplankton, free swimming and bottom dwelling organisms can be there depending on what are the producers. Decomposers of bacteria, fungi and flagellates as usual. And always remember, in the case of energy transfer, it is always going to be unidirectional. The flow is always going to be unidirectional. Okay, ma'am, it's energy, not dry weight, right? It's the basically the dry weight you're talking about, right? What's the question? I didn't understand, sorry. And guys, whenever you make something like this, please mention what all trophic levels are there and give an example of a trophic level is possible. These small things are super important, okay? Make sure you do that. Now, one very important thing that I quickly remembered, tell me, in the case of aquatic ecosystem, which food chain is active, grazing food chain or detritus food chain? Ma'am, please repeat. Dia Maria, which one, dear? Tell me. MQPs are not enough, you need to practice extra questions. Guys, I was asking you something. Yes. In the case of aquatic ecosystem, detritus food chain or basically this food chain, which one is active? Grazing food chain. Grazing one, exactly. Listen to me carefully. Grazing means producers and then consumers and all of this thing. That is grazing food chain, okay, where you have all the levels being signified. Detritus food chain basically means where the decomposer is the one that plays a pivotal role. In aquatic ecosystem, when you go deep into the water, the temperature is too cold. Okay, it's too cold. In cold temperature, decomposers cannot do decomposition. They need a warm environment, which is why grazing food chain is dominant in aquatic ecosystem. Please remember that. Okay, whereas in terrestrial ecosystem, yes, you have the warm environment and everything that is needed, which is why it will be dominant over here. Okay, detritus food chain is dominant where? In this particular condition. Okay, in the terrestrial ecosystem. In aquatic, it is grazing. Have that in your head. Perfect. Okay, just a second. Children, I might have to go. Okay. Ecological pyramids have limitation. What are the limitations of ecological pyramids, my dear children? Any idea? Listen, when you talk about ecological pyramids, they do not consider decomposers, but decomposers are very important things in our nature, right? That is not being considered. And here they assume a simple food chain, but do we have an actual simple food chain? No, we have food web. Next thing, basically over here, my dear children, same organism cannot be considered in two different levels, right? If at all you see over here, for example, human beings can eat plants, human beings can eat animals such as chicken and things like that also. But are we put in different things? No. Right? So please know about that. Now one more very important thing which is there, please know how to make an inverted pyramid in the pyramid of number also. Any pyramid of number things, anything can be asked. Pyramid of number usually looks like this. You have to put down all the names, then you put the number of individuals and you put down a number. This is upright. Inverted one is seen in the case of one tree. If at all you talk about one tree, number is 1. Okay, of the tree. But what of the organisms who is there? Obviously, there will be more amount. So in that case, you will be having inverted pyramid out there. Okay. So yes, that is there. Now the detritus, important conditions for decomposition. Always remember, decomposition is done by aerobic bacteria, not anaerobic bacteria. Who does that? Aerobic bacteria does that, not anaerobic bacteria. So this is something super important. And also, it needs oxygen for that. So without oxygen, it cannot do it. Second thing, there should be a warm environment and moist environment as well. And the content which is going to be broken down, the detritus which is there, it should be having one very important thing. That is what? It should be rich in nitrogen and carbon. If at all it is made up of nitrogen and carbon, it is easy to digest or basically break down. Whereas if it is rich in lignin and chitin, it is difficult. So it will take more time. Okay, so always remember that, and always remember aerobic bacteria does that. So 3 processes that occur simultaneously is fragmentation, leaching and catabolism, and the things that occurs mainly in the soil is humification and mineralization. And my dear children, pyramid of energy is always upright. Okay. So I literally wanted to go ahead with the, so I believe that 3 chapters are left, right? 3 chapters are left, and I'll be coming live, and the last chapter is important, very, very important. All the 3 chapters are important. I don't want to miss out on anything, but still I'm telling you. So just give me a second, a quick second. I'll just give a call and I'll be right back and see if at all I can go ahead with some more time. Just a second, okay? Okay. I have 5 more minutes. So listen to me carefully. Ma'am, put a short for biodiversity and conservation numbers to remember. So many things are there, but yeah, that's a good idea. I'll try it out. Okay, I'll try it out. And over here, okay, please go through decomposition. Very, very important question can come from here. That's why I'm telling you again and again. And yes, so I'll quickly listen. You listen to me. You just listen to me. You do not do anything. Okay. Should we put the same numbers? No, you can put different numbers depending on you. Okay. Statement number 1. There are 32 biodiversity hotspots in the world. Wrong. There are 34 biodiversity hotspots in the world, not 32. Second thing, high level of species richness and high degree of endemism are criteria for declaring a region as biodiversity hotspots. So when I say that this region is biodiversity hotspot, you need to understand that yes, species richness is definitely needed to be there. Apart from that, high degree of endemism. Endemism basically means these organisms are not found elsewhere. That is correct. So these 2 conditions are correct, but the statement is wrong because over here 32 is being mentioned. So I'll say statement 1 is basically incorrect and statement 2 is correct. I'm so sorry children, I am going a bit fast, but it's for you guys, yeah? And thank you so much, ma'am. Oh, that's so sweet. Okay. The Earth Summit was held in Rio de Janeiro so there was 2 summits. One was Earth and one was World Summit. The Earth Summit was done first. It was done in 1992. Now, 2 you remember, because in 2002, the World Summit was also being done. In 1992, the Earth Summit, which was done in Rio de Janeiro, was for biodiversity conservation and also for sustainable development. World Summit which was done, it is also for conservation only, but they have not explained much about it. It happened in Johannesburg. Just go through it once. Okay. So Earth Summit first, 1992, then World Summit 2002. Now, what do we have next? And narrowly utilitarian arguments for conserving biodiversity are: so cereals and fruits, ecosystem services, intrinsic value, aesthetic pleasure. Cereals and fruits are products, and products are something that are the reasons why you should be conserving nature according to narrowly utilitarian point of view, which is why your answer for this question becomes A. It becomes A. Okay, very, very important thing. Next question that is out there. Robert May places global species diversity at about. So global species diversity which is there, it is about 7 million. How many? It is going to be 7 million in nature. The answer for this question is going to be D. Now, do not forget it. Global species diversity, I'm not talking about India, Robert May, global species diversity, 7 million. Cool? Yes. Now, biodiversity and conservation, few things are there which I have not discussed anywhere before, so I'm just going to discuss that. Mention David Tillman's observation with respect to species richness in a community. So always remember, David Tillman was the one who was talking about species richness, not diversity, richness, okay? And over here, this guy said that if at all you have too much amount of species, basically they will be doing too much amount of product, they will be having too much amount of productivity as well, right? If there's too much amount of productivity, other organisms can can survive because of them? Yes. So David Tillman suggested that greater species richness leaded to what? Greater productivity. Yeah, and communities with more species are more stable. Why more stable? Because you have food, you have all the resources and everything out there. Please go through David Tillman's paragraph. That is super important. Okay, it is going to be super important. Do not miss it out by any chance. Now the next thing which is there, name any 4 recent extinct organism as per IUCN Red List. So you have few organisms, dodo, okay, dodo, quagga, thylacine and stellar sea cow. Stellar sea cow is from Russia. Stellar sea cow is from Russia. Dodo is from Mauritius. Okay, all these unique features which are there, you have to remember. We have 4 things, dodo, quagga, thylacine and stellar sea cow. 4 things are there. Make sure you go through their places also. So 2 things I have told you the place, 2 things you have to find it out yourself. Okay, dodo is from Mauritius, stellar sea cow is from Russia. Done. Now we come to 2 different terms. Define the term endemism and bioprospecting. Endemism basically means the ability, it refers that species are confined to one particular geographical area. They are not found everywhere. They are very uniquely found at certain geographical area. So endemism refers to species that are restricted to particular geographical area and are found nowhere else in the world. This is the basic thing of biodiversity hotspots. How many hotspots are there? There are 34 hotspots out there now in the world. Bioprospecting means what? It is the exploration of molecular, genetic and species level diversity for commercially valuable products such as medicines and chemicals. What? It is something that you have to rectify. Okay. Bioprospecting is what? Exploration of the molecular, genetic, species level diversity. Molecular, genetic, species level diversity of commercially valuable products such as medicines and the chemicals out there. Cool? Yes. Now, biodiversity hotspots, there are 34 of them. In India, we have 2, Western Ghats and also Himalayas. Western Ghats and Himalayas. Please remember that it is there in India. Western Ghats, Himalayas. Apart from that, you have Indo-Burma and stuff like that. Indo-Burma, Sri Lanka, Western Ghats and Himalayas are given in your NCERT. 2 of them you remember, that is from India. Okay. If you refer to this video, you should be getting full and full, Baba. But yes, I have not completed it yet. Please make sure you go through the, this thing at least. Okay. The PDF at least. Now next thing, India is rich in genetic diversity. Justify the statement by giving 2 examples. Genetic diversity, when I give, these examples are super important. So I hope you're just listening to me. So genetic diversity is being shown by Rauwolfia vomitoria. This is a plant and this plant is going to produce a thing that is known as reserpine. What do you call it? Reserpine. Now when I say genetic diversity, I mean that there are multiple plants of Rauwolfia vomitoria and they are different in a way of how much potency of the chemical are being produced. So the chemical is same, reserpine, but what is their potency? Some might produce with less potency, some might produce with more potency. So this is an example of genetic diversity in India. That is example is Rauwolfia vomitoria. Apart from that, 50,000 genetically different species of what? Rice is there. This is also an important thing, 50,000. Apart from that, 1,000 varieties of mango is also there. What is there? 1,000 varieties of mango is also there. These are the 3 examples of genetic diversity. It is basically diversity at genetic level, that's all. Now species diversity, the example is basically Western Ghats as well as the Eastern Ghats. The Western Ghats, my dear children, they are biodiversity hotspots, right? So obviously they will have more species, you can think like that. It has greater amphibian species, you're talking about species level. So they have greater amphibian species, but in Eastern Ghats, they have less amphibian species out there. Done. Next thing, ecological diversity. Ecological diversity means, for example, in India, we have too much diversity. There is mangroves, coral reefs, westland, estuaries, so many things. But if at all you talk about a Scandinavian country such as Norway, do you think you will have so many things? No. You do not have so many things out there. So ecologically, who is diverse? India is diverse. Done. Next, write any 2 advanced techniques of ex-situ conservation. Ex-situ means conservation outside their natural habitat. Zoo, botanical gardens, wildlife safari parks, not sanctuaries, wildlife safari park. Park term is there so you can understand it is ex-situ conservation. Now you have been asked about one very important thing, that is advanced techniques which are there. One is cryopreservation, other one is tissue culture. Cryopreservation means you are preserving something in minus 196 degrees Celsius liquid nitrogen. Why you are preserving it? Because you want to make sure the species is there. right? Isn't it ex-situ conservation? Are you conserving it in a natural habitat? No, you are not, right? Then similarly, tissue culture, you take the plants and you grow them. Over here, you're not culturing the plant in the natural habitat. It's done in vitro. So that is one reason. The next one we have is IVF. IVF is also the advanced method. The next one we have is seed bank. Seed bank is also an advanced method. So 4 advanced methods are there. Please have this in your head. Very, very important. Hello, Chandra. Okay. Now, explain species-area relationship. This is something that I wanted to really explain you guys. So if I can, I will. But before that, this thing, evil quartet. Evil quartet means there are 4 reasons for species diversity loss. One is habitat loss. Okay, if you're taking an area, you are basically putting it apart. What is going to happen? The organisms which are staying up there, they are going to get killed. They do not have space to live. The second thing that over here you have is what? Overexploitation. Overexploitation basically means what? Human beings which are there. Okay, they're exploiting, they're killing organisms out there. And that is how passenger pigeon and Steller sea cow got extincted. Passenger pigeon and Steller sea cow, they got extincted because of what? Overexploitation, habitat loss, overexploitation. The third one is invasive species. Invasive species basically means that from outside, you brought something inside, but now the indigenous organism, that means the native organism, they're suffering. Example? Easy examples I'll tell first, okay? Water hyacinth, which you call as Eichhornia, you have studied with before also. Lantana, Parthenium. These 3 things are there. Parthenium is carrot grass. You look at the picture, it will be easy for you to remember. Lantana, Parthenium and water hyacinth, basically 3 of them are plant invasive species out there which came in and destroyed the normal organisms which were up there, okay? Now, apart from it, you have 2 examples. Nile perch, it is a very big fish. It was introduced into Lake Victoria. Okay, Lake Victoria. What happened? There were 200 species of cichlid fishes that got extinct. Why? Because of this big guy, that is the Nile perch. It's not a bird, it is basically a fish which was being introduced in Lake Victoria. Please remember, 200 species of cichlid fishes were lost. Now what? You have African catfish, Clarius garipinus. What do you call it? Clarius garipinus. This Clarius garipinus which was there, again it led to what? It threatened. It was introduced for aquaculture and things like that because it looked so pretty. But the native catfishes which were there, they got extinct. So we bought what? African catfish, but the native catfishes got extinct because of this particular guy. What is the name of this guy? Clarius garipinus. Apart from that, Nile perch is there. It was introduced into Lake Victoria. 200 species of cichlid fishes went away. Done? Done. Now what do you have? Co-extinction. Co-extinction means when one species get extincted, the other organism which is dependent on it will also become extinct. For example, if wasp goes, probably the fig will also not survive for too long. Otherwise, they would need to have some of the new features out there. Okay, so please remember habitat loss and overexploitation, invasive species as well as co-extinction. These are very important things that you need to remember. Okay, done? Yes, and now one more thing, although India has only 2.4% of the world's large land area, its share of global species diversity is 8.1. So according to Robert May, the global species diversity is what? 7 billion. Okay. Over here it says that even though India has less space, but it has diversity. It contributes to 8.1% diversity out there, and it makes us one of the 12 mega diversity countries of the world. 12 mega diversity countries of the world. Slow, ma'am. Okay. Sorry. Okay. We'll complete this, guys. We're up. Okay. One more thing, if at all you talk about organisms as in animals or plants, who dominates? Animals are in more amount. In animals, who? Insects. 70% of the organisms which you see are animals. 70% of the animals are what? Insects. Always remember that. You can be given any of this graph. I'll tell you an easy way of remembering this. Farm. F-A-R-M. Farm. Okay, fishes, amphibians, reptiles and mammals. In between you have to insert the birds. Fishes are the ones that are the most invertebrates in the case of vertebrates. Put them together, it will be insects that are more in number. Now when you talk about plants, fungi is being introduced here itself in the case of plants, and we say that fungi is basically a combination of fishes, amphibians, reptiles and mammals. So listen to me carefully. In total, 70% of them are going to be what? Animals. They are going to be animals. In animals, 70% of them are going to be what? Insects. Done? Yes. Now what about the plants out there? In plants you have included a fungi, and fungi, we say the total combination of fungi is basically a combination of FARM, that is fishes, amphibians, reptiles together, mammals all put together, they give the number of fungi which is there. Okay, are these important questions? Yes dear, what do you think? Okay, yes, got it. 70% animals, out of that 70% is insect. And when I talk about fungi which are there put under plants, fungus is basically a combination of fishes, amphibians, reptiles, mammals, all of them put together. You do not include birds over there. Now they can give you A, B, C, D over here and tell you what is this. You would have to fill in this diagram. So please make sure you look at this. Okay? Fine. Did you understand? Children, you have doubt? Gia, I know I went a bit fast, not a bit, too much fast. At the end, if you have doubt, please ask me. Okay? Ma'am, thank you for your efforts. You're teaching for 7 hours. Thank you so much, guys. And I think I got the call. Just a second. Hello? अच्छा आप पहुँच गए? Okay. Sir, मैं आती हूँ 5 minutes. Okay? I'll just log in. Yeah, thank you. Okay, guys. I wanted to teach you that probably tomorrow. Okay? I will be teaching you this very important thing. And then we just have 2 chapters left. So thank you so much. You will get the PDF right after this. Join the WhatsApp channel. I hope you know how to join the WhatsApp channel. Please refer to the PDF. The chapters which I am not able to complete it from your end. And one more very important thing, I'll show it to you as well. And that is this. Just give me a second, okay? Okay. Yes, over here, if at all you see, you go to playlist, okay? In playlist, what do you have? You would be seeing biology final revision updated today. Click on this particular playlist. You have basically all the things which are important for you. Now, I'll tell you one thing importantly. If you're someone who is losing marks in your 1 marker, I'll ask her to put it over here. She has not put it. I'll ask her, okay? So 1 marker, 2 marker, 3 marker, everything which is there, 5 marker, everything which is there, it will be put over here. Make sure you go through it. And in tomorrow's live, if you have time, you come and join, okay? So, yes. So those are the things which is there. Complete biology. This particular live is of 10. This is not live, actually recorded video is merged. Okay, you can watch this. It's 10 hours long. If you feel stranded, you do not have any knowledge about biology, this class might help you. Fine. So, thank you, ma'am, for giving good time for else. Thank you so much, Vinayak, and my dear children, everyone who have been here. Slim Shade Best, ma'am. Thank you so much. Jia, thank you. Mansi, thank you so much. Shobha, thank you. Good night. Thanks, Alena. Everyone, okay? Samira, every one of you. Lots of love. Please take care of yourself. Jyotsna, thank you so much, dear. Please take care of yourself, and I'll be giving you the PDF, and I'll be updating you in the WhatsApp channel. So be there in the WhatsApp channel for getting all the things. The time and everything will be given to you, okay? Bye-bye. See you. Lots of love. Wishing you the best.